beautiful morning. On the behalf of Organizing Committee, NIPER PHARMACON-2022, I cordially welcome you all for the third and last day of this international conference. Today we are going to have two technical sessions going to happen in this venue before the lunch. Now we are here to listen to the first technical session on the very important area, therapeutic approaches for the CNS diseases. This session is chaired by Dr. Sasibala Singh. She is an eminent scientist, Director, NIPER, Hyderabad, and co-chaired by Professor S.A.S. Sharma from the Department of Pharmacology and Toxicology, NIPER S.A.S. Nagar. May I now invite both chairpersons, Dr. Sasibala Singh, Professor Sharma, to come on the dais, please, and do the needful. Thank you.
Check.
So, good morning and namaskar to everyone present here. I'm delighted to be part of this conference and especially this symposium on the third day of the conference on an important topic, therapeutics approaches for CNS diseases. I think we have realized that health is wealth, at least post-COVID and during COVID. And mental health are gaining a lot of importance. They're occupying a lot of space in healthcare system now for the reasons well known to us. You know, I always look at the system as, it's a computer. The software is the brain and the hardware is your body parts. So we need to take care of the software. If software is not in order, hardware will not function. So this is really good that we are talking on this issue today. And we know India also have, statistics also tells that neurodegenerative diseases are increasing. Even now, the epilepsy occupies the most of the space. Stroke, as I was talking to Gulati, has been from a conference on stroke in Singapore. India, sir, about 37% of the cases in the neurology is from stroke and then followed by headache, epilepsy, and other diseases. So we are looking forward to listening to very eminent speakers. We have 4 speakers who have done a lot of work in their areas. And our first speaker is Professor Anil Gulati. He is in Pharma's company in USA, and I think he holds a very important and distinction in his field. He is MD, PhD, and is chair and chief executive of Pharma's in Illinois. He's a professor emeritus in the University of Illinois. He has done a lot of work. I will not sort of come between him and my introducing him. I think we will listen to him, and his biodata is available in the Google. You can always browse and see. He's an outstanding researcher and an outstanding clinician also. Thank you, sir. Please.
Thank you very much for the kind introduction. It's always a pleasure to be back in NIPER Mohali. In India, this is one of the places where whenever I'm in the country, I do visit NIPER Mohali. So it's like my home. So what I'll do is, because I know that in the audience there will be few clinicians and many basics scientists, so I will go through a little bit slowly so that the clinical aspects of the the results which we got, they are also clarified and understandable. So I was professor and dean at Midwestern University, and based on my inventions, this company was started in 2010, and gradually it progressed and became bigger. Then I resigned from my deanship and professorship, became emeritus, but I still have an office at Midwestern, and still I'm a professor at UIC. But my salary comes from the company now. Next slide, please. I have the control. So, yeah. So I'm presenting results on behalf of many investigators. There were 18 centers in India who conducted the trial. And conflict is that I am the inventor and I am employee of the company which funded these trials. Okay. So slides are moving from there, or I have the control here? Yeah. Okay. Thank you. So this is the structure of the compound, which is initially it was called as IRL 1620. So this was made in 1988, late 88, 89 by Siba Gaigi from Japan. So when endothelin was discovered, first publication came out in Nature in 88, and the industry started working on endothelin A antagonist because it was described in that paper by Yanagisawa that it is the most potent vasoconstrictor substance known till date. So obviously the interest was, let us synthesize antagonist and see if we can treat hypertension. So that was a simple approach. So industry jumped on it. Thousands and hundreds of thousands of compounds were synthesized. And then ultimately in arterial hypertension it failed, but in pulmonary hypertension it succeeded. So today there are several drugs based on endothelin A antagonist in treatment of pulmonary hypertension. But endothelin B receptors were completely ignored. And we started working in 89 on endothelin B receptors, and we recognized, and others recognized, that there are very, very high concentration of endothelin B receptors in the brain. So our strategy was also to see whether endothelin B receptors in the brain are controlling blood pressure. And we conducted a lot of studies and found negative results. We gave IRL 1620, which is a highly selective endothelin B agonist in the brain. Nothing happened. So peripheral, there was some dilatation, and which was transient, and then after that, so not much of cardiovascular effects were observed. So then after that, many years of research of failure, one idea struck that these endothelin B receptors are present on the endothelial cells and they are dilators. So we thought, when the tumor is growing, why not give this substance, and when the fastest part of the growing tumor will be lacking smooth muscles which are constrictor, but will be having more of endothelial cells which are dilator here by ETB. So we gave, and we found that solid tumors, there was increase in blood flow. So we discovered and patented the technology that we can use 1620 to dilate the tumor and give chemotherapy 15 minutes later and enhance the delivery of anti-cancer drugs to the tumor. So that technology was of interest when I presented at one of the cancer meeting, and Spectrum Pharmaceuticals, they came, they did their due diligence and licensed the product. And so they named it as SPIS 1620. So they did phase 1 clinical trial, 32 patients in cancer patients, and then they did 2 phase 2 trials. One was in non-small cell lung carcinoma, and the second was in cholangiocarcinoma. Both were very tough cancers, so the trial failed. So it was back to the bench again. So then we kept on our research on endothelin B with focus that there is very high concentration in the brain, So roughly about 2002-3, there were some publications which started coming out that there are hidden stem cells in the brain, and there are these hidden stem cells can, when the brain gets damaged, they tend to form new neurons. So that was the idea which came, and ultimately we and there is a scientist Koyama from Japan, they found that endothelin B receptors, if they are knocked out in the brain does not develop and the animal dies. So the idea came to us that, and in collaboration with Advocate Lutheran Children's Hospital, neonatology, I gave them the project, their fellows, the project that let us see the characterization of endothelin B receptors during growth. So in a rat, newborn rat, till 1 month of age, we kept on every week. And we found that when the rat is born, number of neurons are very high, and then by 28 days, it goes down to almost half. And it correlated perfectly well with endothelin B receptor concentration. So our interpretation was that endothelin B receptors are integral part of development of central nervous system. So then we thought that if they are that important, why not give endothelin B agonist and see what happens to the brain. So in a normal animal, nothing happened. But in a damaged brain, we found that endothelin B receptor expression increased. So we thought, let us target stroke or cerebral ischemic stroke. And 2008, I was in NIPER that time, and Dr. Shyam Sundar Sharma was having the model of middle cerebral artery occlusion. So we tried some experiments, we got some, but I was about to leave, so we continued those experiments in US and ultimately found that there was very significant reduction in infarct volume. You can see that middle cerebral, almost 90-95% reduction in infarct volume. And tunnel cells also. So we did a lot of studies. They have been published in Brain Research, scientific reports, and many other journals. There are almost like 15-16 papers, basic indicating that endothelin B receptor stimulation helps in the animal model of stroke. So then we thought that let us find the mechanism, how it works. So one mechanism which was very conclusive is anti-apoptotic phenomena. So prevention of cell death was taking place, and the second aspect which was of interest was multiplication of neurons or formation of new neurons. So these are some of the results which are indicating that double cortin, neuro-D, HUC, HUD with sovateltide. Now let me introduce the name sovateltide. Since the drug IRL-620, there was no proper name. So once it came to a stage that the drug has to go to clinic, you have to get a proper name. Now the procedure to get a proper name is that you have to get approval from WHO. There is a committee in WHO which gives the classification of the name, and that committee meets 2 times in a year. And then there is a committee in US, USAN. So we applied to WHO and got the name sovateltide approved, and then we applied to United States AN, nomenclature committee, and the name was approved as sovateltide. So now official word worldwide is sovateltide. That is that. So now you can see these figures that there is highly significant increase in neuro-D, HUC, indicating that these are the markers for neural progenitor cells which are improving. So stem cells, endothelin B agonist sovateltide is not acting on stem cells in the brain, but is acting on one step after the stem cells, that is neural progenitor cells. Now, these findings are more on mitochondrial fission and fusion, and once again, you will find that apoptotic phenomena, it is acting through prevention of cell death and mitochondrial preservation. So all findings are indicating that mitochondrial preservation is quite significant at day 1 and day 7 of post middle cerebral artery occlusion with sorbitol. So these are some pictures indicating mitochondrial DNA activity. So you can see the green bar, significant improvement in mitochondrial DNA activity in presence of sorbitol. Then these are some electron microscopic pictures, once again indicating that mitochondrial preservation is quite intact, and mitochondria is the seat of activity that is very important in the brain. So cross-sectional area, so all these findings are indicating that the mechanism of action is twofold. One is that it is protection, and the second is neuroregeneration. So protection, we published results that it is anti-inflammatory, antioxidative, apoptosis is decreased, mitochondrial biogenesis, so it will preserve neurons and functional recovery. The second part, which is a little bit delayed, is the angiogenesis, which correlates with VEGF factor, DXN, NeuroN, and the mechanism is through neural progenitor cells. And we also found that presynaptic, postsynaptic markers are also increased. So these findings also have been published in scientific reports. So these are some of the publications, key publications which you can look at. So in nutshell, what is happening is that besides prevention of apoptosis or anti-apoptotic activity, there is stimulation of neural progenitor cells and formation of mature neurons. So we quantitated, it is close to 300% increase in formation of mature neurons by sorbitol. So we did phase 1 study. We got the approval from DCGI here to conduct phase 1, and phase 1 came out to be okay, although we had done 32 patients plus close to 200 patients in cancer, but the committee decided that you have not done in normal individuals, so do it again. So we did phase 1, came out okay, and then we started on phase 2. We got the permission to do phase 2. These results have been published in CNS Drugs, and that came out to be quite promising in the sense that the neurological outcome was improved. And we all know that stroke has faced so many failures that almost every drug which has been developed since TPA has failed. So nothing has come out. So we were well aware of all that fact, but the results were so promising, we decided that let us move forward and spend the money to get phase 3 permission. So after phase 2 results were published, we embarked on phase 3. We got the permission to do phase 3 trial, which was done in 18 hospitals across the country. And this was the design of the trial. So the design of the trial is such that NIH S.A.S is the assessment of the extent of stroke. So we took moderate stroke, that is above 5, so 6 and above NIH S.A.S was kept those patients, and we excluded patients wherever any surgical intervention was there. For example, thrombectomy and those kind of things, they were excluded. So intracranial hemorrhage was also excluded. So only ischemic patients. And the reason is, although we know that the drug is neuroregenerative and all that, so it can act, but to do a trial, you have to be, better it is to keep it clean and not too many interferences. So that was the main reason why we wanted to have only ischemic stroke. And so those patients were included, randomized standard treatment, whether they were blood thinners or TPA, they were allowed to be given. So 2 groups, saline and PMZ 1620 or sorbitol, and visit 1, 2, 3, 4, 5. So 90-day assessment, that is the gold standard which all the regulatory agencies across the world look for. And so primary endpoint they look for is 90-day, and nowadays US FDA and Japanese agencies, they are looking for longer term also. But primary endpoint still is 90 days. So we did that trial. There were 760 patients who were assessed, and we did not want any recurrent stroke. So All India Institute Delhi, they got a lot of patients with recurrent stroke, they were excluded. So only fresh stroke patients were included, and they 158 patients, 78 in control, 80 in sovatiltide, and 90-day follow-up completion was done by 70 patients in control and 67 in sovatiltide. So the mean age, body weight, male-female ratio, they are very similar. Now very important is NIH, S.A.S at baseline, 9 and 10 median, they are very similar to each other. Median aspects is by radiologically, you can find out the volume of infarct by three-dimensional CTs and MRIs. So you can see 8 and 8, exactly similar volume of infarct was there. Thrombolytic therapy, which is TPA, which is the only drug which is approved, has to be given within 4.5 hours, because the later you give, the chances of intracranial bleed is very high. So recommended is 4.5-hour limit. So you will see that 25% of the patient in control got thrombolytics and only 11% in sovatiltide got thrombolytics. And that is statistically different. But I want to highlight that in spite of the fact that more patients got in control thrombolytics, but you will see the results are in favor of sovatiltide. Similarly, large artery atherosclerosis is the most devastating, and high mortality is there with large artery atherosclerosis and more disability. So more 46% patients in sovatiltide compared to 37, although statistically not different, but still 10% difference. The patient's outcome in sovatiltide was better. Median hour interval is about 18-19 hours after the onset of stroke. So that also is very promising in the sense that instead of 4.5-hour window, we kept a 24-hour window based on our animal experimentation data. So in the trial, it is roughly about 18-19 hours. So the subtypes of stroke, that is also important, because we do not want that the tilt is more towards devastating in one, but here it is opposite, that large artery atherosclerosis, there are more patients in sovatiltide than in control. But other small vessel occlusion is more or less is the same. Now comes the result. So the results are neurological assessments, and the gold standard which US FDA and other regulatory agencies look for is head-to-head comparison of modified ranking scale. So these are neurologists, they rank according to the neurological tests, and modified ranking scale at day 90, head-to-head comparison, p-value is 0.007. So then we thought that let us analyze from baseline how many patients are having more improvement in modified ranking scale. So modified ranking scale of 2 or more improvement from baseline to 90-day, once again is statistically significant, 0.0045. Another thing, with our initial correspondence with US FDA, they said that baseline modified ranking scales are not reliable. So can you assess patients with 0 to 2 modified ranking scale? 0 to 2 means that 0 is normal, 1 is clinically, the disability is not even obvious clinically, and 2 is very mild clinical disability, and then 3 is that you need assistance in walking and all that. So 0 to 2 is something which is very important. So you can see that patients at 90-day with 0 to 2 modified ranking scale are 54 compared to 39, and p-value is reaching at 0.001. Then another way to test is NIH-SS. So although this is not a primary endpoint with US FDA, a modified ranking scale is the primary endpoint, but NIH-SS also at 90-day head-to-head comparison is coming out to be significant, p-value 0.002. Then number of patients having 6 or more improvement in NIH-SS is also significant, 0.1. Barthel index is another way to assess, and in Barthel index there is a component of cognitive also within. So the head-to-head comparison is 0.1, statistically significant. Now change in Barthel index of 40 or more missed the level of significance, 0.6, but still there is a from 61% to 76% number of patients in control and sorbitol-tide. Then we thought that let us see how many patients are having full recovery. So full recovery with all 3 parameters, that is modified ranking scale, NIH-SS, Barthel index. So modified ranking scale 0 is full recovery, NIH-SS 0 is full recovery, Barthel index 100 is full recovery. And at the bottom you can see the percentages from 15 to 26, 25 to 37, and so on. So at least 10% more patients are having full recovery in the sorbitol-tide group in any way you measure. Then quality of life index, although these are exploratory endpoints, they are also coming out to be statistically significant. Montreal cognitive score did not come out to be significant, and the reason for this is for cognitive, in Alzheimer's disease we are doing a trial, but there the drug has to be given every month. It's a longer term, so the dosing has to be different. So in stroke, the dosing was 6 days. On day 1, patient got 3 doses of sorbitol-tide. On day 3, patient got 3 doses, and on day 6, 3 doses, and that is the end of treatment. But in cognitive, we have to give every month. Survival mortality log ranking Cox shows no statistical significance, and there were 4 deaths in sorbitol-tide and 2 in control. And once again, if you notice large artery atherosclerosis, 3 patients who died were having large artery atherosclerosis in the sorbitol-tide group. These are the adverse events which have been recorded. So 33 adverse events in 24 patients and 27 adverse events in 15 patients, and most of the adverse events have not been correlated with the drug. So basically safety was established in phase 1, phase 2, and now in phase 3 also. Now it is a lot of interest that how many patients got intracranial hemorrhage, because that is one of the major adverse events which happens with TPA. So 7 patients out of 80 in sorbitol-tide group and 7 patients in 70 in the control group got intracranial hemorrhage, which is similar, and percentage-wise also about 8.7, 8.9% is equal to the conventional reports which have been published across the world. So there is no incidence of intracranial hemorrhage, and we also did in vitro clot lysis and all those studies, absolutely no interaction with TPA, and it can be safely given with TPA. So the summary, you can see that any way, with any parameter we analyze, this compound sorbitol-tide is showing highly significant improvement in neurological outcome. So p-value of, you can see that on the other side. Neurologists are always concerned with what is the shift which is taking place. That is clinically meaningful improvement in neurological outcome. And you can see the shift of modified ranking scale from 0 to 6. 6 is death, and 5 is severe disability. So the shift, that means from control 15 to 25% shift is taking place with patients which are completely normal, that is no disability. And with modified ranking scale 2, from 54 to 76%, that is close to 20% improvement in the shift which is taking place. So this is of lot of interest to neurologists. So we thought that such a shift has never been reported with any compound after TPA. So let us compare with the only gold standard which is there, which is TPA, and we compared that. So in 1995, New England Journal of Medicine, they reported 0 to 1, 2 to 3, 4 to 5, and death. So we could, head-to-head comparison, we could do at 3, that is shift of modified ranking scale from 0 to 3, and that is coming out to be 9% with TPA. With sovorteltide, it comes out to be 12%. And if we take 2 modified ranking scale 0 to 2, which is even better, the favorable outcome is coming to about 24-25% with sovorteltide. So primary endpoint with modified ranking scale 0 to 2, it has met p-value 0.002. Another way to analyze is number needed to treat. That also is sovorteltide 5. So to have the excellent outcome, you need 5 patients with sovorteltide. With TPA, you need 10 patients with 3-hour window. And if you go 4.5-hour window, it jumps to 19. So in summary, it's a platform drug, although we are focused on acute cerebral ischemic stroke, and we have applied for marketing authorization. We got the minutes that IND committee has approved that this drug can be allowed to be manufactured and marketed in India. So now the formalities are going on, audits are happening. DCGI is conducting audits at clinical sites. So once all those are complete, manufacturing audit will take place, and after that, most likely by February, we should be having this drug in market in India. And we have also applied for the application for phase 3 directly in US is ready, and we are having good feedback from US FDA that we'll get straight phase 3 permission in US with the US FDA. So thank you very much, and I'll be happy to answer questions.
So congratulations, sir,
Thank
for bringing this molecule to this level, and hope in future we'll be able to, some of us will be able to sort of be part of this molecule. Really great. Congratulations. Any question? One or two questions? Dr. Chauhan. Yeah.
Oh, I can. So I have a couple of questions. Very nice talk,
Thank you.
So my first question is, did you try the interaction of this drug with the TPA in vivo?
Can I have the, yeah.
So did you try the interaction of this drug with the TPA?
With TPA?
Yeah.
Yes. So as we indicated that there were 25% of the patients in the control group got TPA along with the drug, and 11% of the patients in sovorteltide group got TPA along with the drug. And we have analyzed further. There is extensive due diligence going on with one of the pharma companies in Japan. So they wanted the timing between the two drugs. So TPA, in the patients who got TPA, TPA was the first drug and sovorteltide was the second drug which was given.
Okay. Thank you. And second question is, this all looks nice, but have you tried this in an embolic model of stroke where you can give it emboli, because most of the strokes are like embolic, right? 85% stroke. And then give a TPA to lyse the clot and give you a drug to see whether you have a longer outcome to prove in vivo that the TPA is not cutting this drug. And I also saw that in your listings, you only choose patients who were given TPA, but no one with a mechanical thrombectomy. So you do not take patients with a mechanical thrombectomy?
Mechanical thrombectomy we could have taken, but that would have confounded the results. Then the trial would have been designed, would have been completely different.
Okay.
So we are now doing patients, we have made the protocol for thrombectomy separately. I'll very strictly, this is another very important thing. When you are designing a clinical trial, you have to design, the key thing to design the clinical trial is to keep it clean and not confound too many factors. Otherwise, you will be getting results all over, and then you will have to do a 1,000-patient trial, which will take 10 years to do it. So that becomes more of an academic exercise. So we have to keep it a little bit, for example, when I was doing the trial on artificial blood, we kept blood loss only, not the fluid loss. So these are some things which have to be decided early on, that what is the protocol. And regulatory agencies also want a clean thing. They do not want things to be included. Later on, academic exercises can be done. And right now, for TPA, it is even easier for us because there is a trial which we are starting in Singapore where thrombectomy, because the catheter will be there in the artery, the thrombus is taken out by the stent, and we can inject the drug right in the arterial. So that trial will be starting soon.
Yeah, thank you. One more question. Yeah.
So, my name is Bharat from University of Wisconsin. So, phenomenal talk. So my only question is, if you need a clean data, you selected and excluded some patients, but the patients are not clean. They are come up with a lot of complications. So what is your next plan? How you are going to take this molecule to the complex patients?
Yeah, so TPA, already we know that only 5% or 6% of the patients are able to get. Even in the United States, only 10%. Japan is the only country where almost 50% of the patients are going to getting TPA because they are so efficient emergency management system. So the best part is that we decided to keep TPA patients in the trial, and we found that whether you give TPA or you don't give TPA, the results are, they are independently acting. TPA is to dissolve the clot. This is to protect the brain, regenerate the brain. So it is completely different. So it can be given, and 24-hour window also is very good, that even in remote areas, 24-hour window, a patient comes, they can be treated with something which is not available up till now.
Yeah, interesting. Thank you.
Thank you. Thank you very much.
Thank you. Thank you, sir, and thank you, everyone. Now I hand over to my colleague, Dr. Sharma, for further deliberation.
Yeah. And thank you, Professor Gulati. I think as a pharmacologist, always you are an inspiration for everyone because you are putting your molecules to the clinical stage and not to the market. So it's a great success. Congratulations for that. So now we are going to have the second speaker, Dr. Bharat Chaliboinia from the University of Wisconsin, Madison, USA. And I'm happy to tell, like, he's from the University of Illinois, Chicago. He did his postdoc, and I think from the same place, also University of Illinois and Chicago, I also did my postdoc, actually. And Dr. Bharat published, actually, around 50 papers in the area of stroke, and now I think, without wasting much time, actually, I would like to invite Dr. Bharat to share his findings, actually, on a very nice topic, actually. I think is continuation to the ischemic stroke, impact of comorbidities in developing neurotherapeutics for ischemic stroke. And Dr. Bharat, please.
Thank you.
And I would appreciate, actually, if you can restrict 25 minutes of your talk and 5 minutes for discussion. Thank you very much.
Yeah, sure. I have a habit of finishing 10 minutes or 1 hour. Thank you. So thank you for your kind introduction. Good morning, everyone. It's my pleasure to be here today. And today I would like to talk about ischemic stroke, one of the subtype of stroke. So, stroke is a very common, most common leading cause of disability in the world. today I want to share some of the factors that contribute to more disabling and more fatal strokes. So in particular, we and fellow researchers found that a lot of comorbidities such as hypertension, diabetes, atherosclerosis, or renal dysfunction contribute to these disabling outcomes. And in our lab, we found that some molecules like extracellular matrix molecules, subclass, namely tenascins, play a crucial role in connecting the organs between the periphery and brain, and it complicates the stroke pathogenesis. So my talk involves a brief introduction about the stroke and comorbidities and inter-organ mechanisms, and what are the challenges we are facing in developing or in studying stroke outcomes in presence of comorbid conditions. And the question comes whether developing a model is tough or identifying a common target is tough. And I want to link some of my recent findings with these things. So if you see, stroke is incredibly common. It is getting more common in younger population because of various life changes, particularly physical inability and atherosclerosis, high cholesterol, atrial fibrillation, and blood pressure. So here, since I'm talking about ischemic stroke, I stress more on atrial fibrillation rather than hypertension, because hypertension mostly leads to fatal strokes, hemorrhagic strokes, that I'm not going to talk about today. So ischemic stroke happens when there is a clot within the brain, but it is not necessary to form a clot within the brain. The clot forms somewhere in the periphery, and it travels all the way to the brain, and it blocks somewhere. So in clinical scenario, the symptoms vary between the men and women and depends upon the location of the blockage. For example, I'll show these are the common symptoms people suggest, especially American Heart suggests. If you have any of these symptoms, go to the hospital. And there is a chance, there is a time-based prognosis of the disease where you see the red color, the blockage happened, the tissue died. There is a green color, you are going to rescue the tissue by means of thrombolysis or thrombectomy if you go to the clinic within a 4.5 hour window in case of thrombolysis, or in case of thrombectomy, you can go up to 24 hours. But if you have a comorbidities, any of the comorbidities, it creates a pre-environment in the brain which compromises the neurovasculature, and it compromises various factors, including the transient attacks. Just like a heart attack, you don't even realize sometimes you get a stroke and the clot immediately dissolves. You don't even feel any neurological dysfunction. So in comorbid conditions, you don't really know whether the stroke is a first-time stroke or a recurrent stroke. So when there is a disability or comorbid conditions, the same like ischemic damage happens within the 10 minutes. In case of normal people, it happens in 3 hours. So here, in case of comorbid conditions, we have a narrow chance to rescue the tissue so that they get more disabled strokes and also more fatal strokes. And treating these patients is really, really tough. And I like the talk from Dr. Gulati, and where I interested more in exclusion criteria. So you recruited 700 patients, 760, and you excluded 600, where there is the patients with a lot of complex disorders. So my goal is how to treat a stroke if somebody come to the emergency room with a lot of comorbid conditions, and how to design the therapeutics. So we have a very well-established mechanisms in terms of stroke. We all know the ischemic stroke happens because of the rupture of the plaque somewhere in the periphery. It travels and blocks the brain, or inflammation or thrombosis within the brain, and which leads to gut dysbiosis, inflammation. Those are the common mechanisms recently demonstrated very well in humans and experimental stroke models. And if you don't treat properly, it ends up with some cognitive dysfunction and also chance of, again, recurrent fatal strokes. So if somebody have a comorbid conditions, the inflammation and sympathetic nervous system overflow starts just before the stroke. So your brain is already delicate to these changes. So the thrombosis happened in the comorbid conditions is more tough to break by the endogenous TPA. It needs more endogenous TPA, or you have to give more TPA. If you give more TPA, there is a chance of hemorrhagic bleeding within the brain. So these patients are not eligible for the frontline medications at the acute phase of the stroke. And also, Here I want to, don't touch on, I don't want to touch with the inflammation because everybody knows about inflammation. I want to touch base with gut dysbiosis. So everybody knows stroke happens, stroke induces gut dysbiosis, but also comorbid conditions produce gut dysbiosis. But here, my focus is not about the gut dysbiosis happening because of the diseases, even not either because they involve in the pathogenesis. So here my worry is how, if you give a drug to these patients, how it is going to be activate or produce the therapeutic effect. Because a lot of antihypertensive agents or antidiabetic agents, their metabolism or activity depends upon the gut bacteria. Not only that, once they degrade into the product or bacterial products, if they have comorbid conditions, they can easily translocate into the brain and they cause undesirable side effects. So that is a very serious concern. Nobody looked at those things. And remaining like a gray matter and white matter is very common in kidney disease patients. It reduces the white matter and gray matter. So these are the complications. And also, you have a cell death mechanism which is very specific to the sex. Males have a different cell death mechanism, and females have a different sex inflammatory mechanisms and cell death mechanisms. So altogether, it affects the diagnosis process. Whenever the patient comes to the clinic, most of them, they are not eligible to certain contrast agents if they have a renal dysfunction or a compromised kidney, or they are not eligible to take other contrast agents. And also, it interferes with the vascularization, either thrombectomy or a thrombolysis. And also, most importantly, all the ischemic patients are going to survive for a relatively long period if you provide a proper post-stroke management. But in this case, even if you use a new generation oral anticoagulants or certain antihypertensives or a multi-drug therapy, they have a narrow opportunity to treat with the regular antihypertensive or regular medications. So here where my research comes. So I'm very particular about developing a preclinical model. Although we use a conventional techniques, the problem with the conventional technique in the stroke is there is a huge variability. If you show brain damage in one animal, and if you show behavior in other animal, or if you show physiology in other animal, there is no proper rationale to track down the prognosis. So here my animal design or experimental design is, I'll track a single animal from the zero. I induce the prognosis of comorbid conditions, whether it is a CKD or a diabetes or anything. Then I induce a stroke, and I'll track the same animal up to the survival and end point. So we use various techniques, you name it, whatever the techniques we use for human clinical trials, we use in the lab. So we use ultraset and like a microspect with Neurolite, which is a clinical tracer for the CBF. And my study design is long-term. So we use various MRI techniques with the contrast agent to determine the BBB disruption and brain damage. And we also do some regular behavioral motor function tests, trotter rod and beam walk. And we do Morris water maze, and we do survival things. So this is a plan. And so these are the things published recently by our group in stroke. So here we identified the aging and time of treatment play a crucial role. Although I don't want to discuss all the things, I'll show that there are few things we have to observe in the cresal things. The young brain, if you induce a stroke ischemia for 60 minutes, it produces the damage which is equivalent to if you induce a stroke in an age group in 30 minutes. That means basically it is producing a 50% more in case of age groups. And also if you treat acutely, it gives protection. And the same drug, if you treat a delayed time point, it is not going to work. So those are things we have to consider. So meaning that therapies in the stroke, especially in the different age groups and male and female, are time dependent. So this is an interesting and very latest technology. We used nanostring, genomic spatial profiling for proteins. So what is happening in the comorbid conditions? So comorbid condition itself produce some proteomic changes in the brain. So what are the blue color shown here? The proteins, we profiled 57 proteins, and we identified some proteins induced after only comorbid condition in the normal brain without any stroke. Then we induced a stroke, and those molecules are further induced in a comorbid condition plus stroke group when compared to the stroke group. Then we compared what are the common targets. Is there any common targets in this study? We identified several molecules. The molecules of my interest is the cleaved caspase-3, a well-known cell death marker, and fibronectin. So fibronectin is a very good molecule to interact with the different organs and cells. So we want to pursue like what happens, what causes these things. So fibronectin, we know from the literature that certain molecules like tenascin C activates or induce the mechanisms through the fibronectin, and also like other inflammatory mechanisms. So I will introduce the tenascin C. So which is a very good protein in the developing brain. It helps to develop the organs, and it disappears as soon as the brain development is over. But in adults, if it induces again, that indicates that person is having some problem in the brain, or overall he has some disease. This is like a condition of inflammatory marker. It's a biomarker. So it's currently using as a biomarker for several diseases. Not only biomarker, this level, CSF levels of tenascin C is an indicator. We can predict whether the hemorrhagic stroke patient is going to die or survive based on the tenascin C levels. Not only that, this is a time-dependent activity. It also have a very good activity at the delayed phase of the stroke. In case of hemorrhagic stroke, the problem is if you have a more tenascin C in the delayed phase, there is a chance of wound healing followed by the scar formation. So you all know if the scar formed in the brain, then it produces the convulsions. And again, the post-stroke convulsions is a detrimental factor. In most of the stroke patients, they are going to die because of the seizures. Post-stroke seizure is a detrimental thing. So not only this, it also participates in the thrombotic plaque stability. If like a healthy person have a stable plaque, he's not going to get any blockade. But once there is a high levels of tenascin C, it destabilizes the plaque, and that detaches from the blood vessel. It travels somewhere and blocks somewhere in the brain or some other parts of the body. And also we have a direct role on the TPA. And also there is like a disparity in using a knockouts as well as like inhibition of the tenascin C. Because knockout, since this tenascin C is very good for the brain development and organ development, knockouts are not an option for here. So we decided to block this tenascin C by using a siRNA technique, and also we are developing some inhibitors. And we came to know that tenascin C upregulates after strokes for several, like 42 days. And also stroke pathology peaks at the third day of stroke. So we want to study what happens to the mechanisms within the 1 day and 3 day, and also outcomes up to 1 month and survival up to 180 days. And we identified this tenascin C induces immediately after stroke. We tested up to like as soon as 3 hours after reperfusion. So we tested the efficiency as well as the in vivo delivery of the siRNA. So the spectral imaging shows there is no differences in the blood flow dynamics whenever you after administering the siRNA. And also we checked whether it reached the brain or periphery. So we decided like we injected the fluorescent dye along with the siRNA to the retroorbital sinus, and it entered into the brain. Then we tested the protein efficiency, and it knocked down sufficient amount of tenascin C after stroke. Then we tested for the infiltrated cells as well as the apoptosis. The siRNA administration at the 5 minutes of reperfusion reduced the neutrophil infiltration and also the apoptosis. There is an interesting mechanism. This tenascin C attracts the inflammatory cells from the periphery by means of a chemotaxic entry, and it's promoted into the brain. And also like a cell death mechanisms. And also it works with closely with the myeloperoxidase, and it recruits the cells to the brain. Then next, we are interested to see, is it because of the chemotaxic entry alone, or is there any brain disruption or BBB disruption after stroke, and what tenascin C is doing for the brain disruption? And I'm very interested in non-invasive imaging. I don't go for like a conventional techniques. That's what I want to say to all the students. So look outside the boundary. So I induced a stroke, and I treated with the tenascin C, and I injected a gadolinium, which is a very, very famous contrast in the clinic. It don't enter into the intact brain. If it enter into the brain, that indicates the BBB disruption. And we identified there is a huge uptake of the gadolinium into the untreated group when compared to the treated group. And we further confirmed with several proteins by testing for the tight junction proteins. And next, we want to see what happening to the brain damage. We did a T2 MRI, and we identified the brain damage reduced with the tenacity knockdown. And we also do like a T2 star and DWI images to see iron content that indicates the hemorrhagic transformation in the brain. And we also see neurodegeneration by using amyloid tracers, by using PET scan. We have all the facilities. And also, like these are the direct effects because there is no changes in the fluid dynamics. And next, we tested the motor and cognitive functions by using water maze and this rotor rod and beam walk. We identified a significant good recovery after a stroke and treatment. Then we identified what kind of mechanisms playing a role, and we identified there is a we screened only few like well-known pro-inflammatory cytokines, and we identified some sex differences here, but I don't want to talk here, but I will talk in the next slide. So here, it is very interesting that males showed IL-1 beta very high, almost like 24-fold, and females showed only 6, because like young females have more protection after stroke when compared to the males. So then, we know like more in-depth mechanisms. We know tenacity activate different functional groups. Then we want to see one of the endogenous ligand for tenacity is TLR4, toll-like receptor 4, and we profiled all the signaling, total signaling of TLR, toll-like receptors, and we identified few molecules changed in a differential manner in the male and female. So if you identify TLR4, it reduced with a tenacity knockdown in both males and females. If you take the TLR3, it reduced only in females, meaning that TLR4 males are following the mid-88 pathway, and TLR3 females, they are following mid-88 independent pathway. So this is really interesting. This prompted us to check what happens exactly in the toll-like receptor 4 knockout mice. Then we purchased recently mice, and we induced the tenacity siRNA, and we identified it further reduced. We know TLR4 is a protective after stroke, but it further reduced with the tenacity knockdown, meaning that tenacity is partially acting through the TLR4, and also it has its own mechanisms with the other functional domains. Then, if you want to take any drug to the clinic, we know there are several like limitations to siRNAs and adenovirus and lentiviral. So recently we screened for the druggable targets. So we identified tenacity TLR4 interactive sites and also tenacity TLR4 independent site, and we are currently screening some 10 structural molecules we purchased from the Sigma. And once we get an inhibitor, we want to check in the animals, and we want to start a clinical trials. So this is a story of tenacity in young mice, male and female. Now we are working on various comorbid conditions to see whether those complex mechanisms I discussed earlier are playing a role here. So this is about my story. And these are the conclusions. So testing post-stroke therapeutics in all the comorbid conditions is really complex, and also it produces less conclusive results if you use a conventional technique. And also common targets like tenacity are the best way to go. And non-invasive imagings give a very good rigorous information about the disease ongoing pathogenesis. You can test the magnitude of the damage. And with this, I'll thank my team, and my Professor Vemuganti is one of the mentor for me, and also all my team. And I'm happy that I have my own funding, and also I borrow funds from my mentor, Professor Vemuganti. So basically, we have a very rich lab. We can do whatever the imaging we want, and we can do like plenty of stuff. Thank you. Thank you for your kind attention. I'm happy to take any questions.
Thank you, Dr. Bharat, for, I think, finishing within the time limit, actually. Thank you very much, actually. I think one of the issues always comes with the stroke is the translational failure, because most of the drugs which have been discovered, they could not go to the market. And that we can see actually maybe in future the success of Dr. Gulati drug, how it's going to perform. Like I would like to know, like, because when we do a lot of experimental studies, always we look into the S.A.S guidelines actually. In your study, like how you're following those S.A.S guidelines for these studies?
So my study is, we always follow state criteria, but like earlier we used to do, like I have all the struggles of failure when I worked with the diabetic mice and hypertensive mice and other mice. And then one day I talked to Dr. Wim Ganti and I told, I don't want to do any of the conventional techniques because it is giving less conclusive results. And also it is really tough to track a mechanism if you use a conventional techniques. And also at the same time, all the other labs, they don't have those facilities. So luckily we have facilities to the animal facility. It's like a walkable distance. We can use whatever imaging. And also I want to share to the students, if anybody interested to screen any of your stuff, we are ready. We are happy to help you. Even our industries also, even Dr. Gulati also. If you are interested, we are always ready to screen any of the molecules.
Actually, I mean, one thing which I'll admire is a nice presentation, of course, and it's not easy to build that kind of infrastructure and the facilities which you have built in. So I commend you for that. Regarding comorbidities, hypertension, diabetes, and there are other situations. Out of these, particularly from India, which one you think will be the best target to handle it first? And the reason I'm asking is that there is a group of neurologists in around Hyderabad area. What they did was they selected few villages and just monitored blood pressure and controlled blood pressure, and the incidence of stroke has gone down in the last 3 years. They could monitor. So your thoughts on which one is the most important?
So in clinical setting, we have seen a lot of patients. They recover more if they are under comprehensive antihypertensive agents and anti-diabetic agents. Most surprising thing is, I don't know whether it is there is no mechanism, but like a post-stroke recovery is very good if somebody quits smoking. Nobody so far discussed about it or demonstrated, but if you see the clinical data, if you quit smoking, the chance of post-stroke survival after TPA is more than 50%. And it is only 20% with the hypertensive management and diabetic management. When it comes to India, there is a regional disease prevalence. So if you go to south, it is more like renal problems. And if you go to urban areas, more so recently I came across one article when I want to give a talk in India. I checked out what are the stroke structure in India. So what I identified is all the urban people living in the urban have more strokes, and all the rural area people having more cardiovascular, heart attacks and all those things. And also the problem is stroke you can easily prevent by changing lifestyle and everything. So I think in India, I think it's better to start, India needs a drug like yours because it is kind of more economical. Because if you suggest a thrombectomy, you need a lot of people in comprehensive unit. You don't get a lot of specialists here. You don't get a neurosurgeon, neurologist, and an interventional neurologist here. So I think we need a proper drug candidate. But targeting a drug candidate with diabetes is more better in India when compared to the hypertension. The reason I'm saying diabetes is, if the person is diabetic, they also have problem with the drug clearance because they end up with getting nephropathy, diabetic nephropathy. I think that is one thing. And another thing, there is a new class of drugs now prescribing SGLT2 inhibitors. They are well known to control the hypertension and diabetes. Yeah.
Okay, so I have a question for you and a comment what you asked, Dr. Klatia. So I think that diet-induced obesity is a very nice model to, because a lot of people, we have fat. When we become old, we like to eat, all junk food, obese conditions, you have increase in diabetes, all DBDB models, you are making knockout, but in human, we don't do any knockouts. So I think diet-induced obesity is a very nice model to understand the comorbid condition. and I'm sure his boss is he's working on that. So my question is, okay, you are working on TNSNC, so you show some of the mechanisms, very nice talk.
Thank you.
One of the mechanisms you show is like TNSNC is promoting thrombosis,
Yes.
and that's my field, and you also show that this is also promoting inflammation.
Yes.
But TNSNC is an extracellular matrix protein, right? It interacts with so many targets,
Yes.
not only TR4.
Yes.
Do
Yeah.
you think that TR4 is the only target, or it also interacts with some integrins which are present on the neutrophils, because you show some slides on neutrophils?
So, yeah.
And second question is, why you think targeting TNSNC is the best approach if it's going through TR4? Why TR4 antagonist has not been successful in clinical trials?
So, yeah, very good question. So, the first question, why TNSNC is having a role is TNSNC already is a marker for atherosclerosis in the humans. And it is also identified in the stroke patients, and it correlates with the TLR4 levels in the humans. So there is a clinical evidence show that TNSNC have a role by interacting with TLR4 in atherosclerotic plaque and also in the large artery atherosclerotic strokes. And at the same time, it also have a role in hypertension because it induces cardiac fibrosis. Coming to the next question, TNSNC have several functional domains. We tested only TLR4 now. So whatever I'm talking about the last slide, which have independent of TLR4 activities, that might be through the other functional domain. So now we are checking the fibronectin, and EGFR is another functional domain, and also integrins. And also we are checking all the things, but we have to start somewhere. So we started here.
Thank you.
Yeah, thank you.
Thank you.
If you don't have any
any
questions,
question, just last question. Yeah, just please be brief actually, because we are running late.
Good morning, sir. Thank you for delivering nice talk. I am Ziaur Rahman from NIPER, Hyderabad. Sir, actually you showed one slide like you are also working on stroke-induced gut dysbiosis.
Yes.
So we know that stroke will cause gut dysbiosis. So how will target both together, gut TLR4 and brain TLR4?
So if you are working on stroke, you always try to protect the brain. So the first thing, I'll protect brain, then I'll come to the gut microbiome or gut bacteria. And also gut bacteria is very old now. We recently published a paper in the gut virome. So you have more virome in your body when compared to the bacteria. That tell the bacteria what to do. So we are manipulating the virome now. So gut bacteria is almost 2015 and 2020. So it's almost done. But the question for you is, we have a special prophages and all the probiotics with a very specific phages and very specific bacteria. If you maintain the balance between the bacteria, I think it helps a lot. So the probiotics is a key. But the problem is what probiotics if you take and how much quantity you take, there is no proper investigations. The problem with the stroke and the use of probiotics is that some of the bacteria can travel, or metabolites can travel to the brain. And we don't have that information how they are going to affect the brain. But overall, clinically, if you use probiotics, it stabilizes the gut.
Thank you.
Thank you.
Thank you, Dr. Bharat, for nice talk. Thank you very much. Can you, madam, introduce next?
Next speaker is Dr. Anil Kumar Chauhan. He is a professor and director of thrombosis and vascular biology in the Division of Hematology, Oncology in the Department of Internal Medicine, University of Iowa. Dr. Chauhan has a lot of honors and credit. He has been awarded many honors, he has 75 publications, and he has made numerous contributions onto how adenosine molecule contribute towards thromboinflammation. He has been awarded a lot of grants, which includes R35 Outstanding Investigator Award. He also has been awarded from NIH and prestigious Established Investigator Award from American Heart Association. So he has a lot of distinctions and honors to his credit. And I now invite Dr. Chauhan to give his presentation.
I can stay here, sir. So, thank you very much for the kind introduction, and I would like to thank the organizing committee and the chair and the co-chair
and especially all the students. So when we came at 9, we thought that it's only speakers. We had to give a talk to the speakers. Now we can see all young faces. So let's change the gear. Yeah, okay. So last 2 talks, you heard about ischemic stroke. And 85% of the ischemic stroke are caused by thrombosis. So I am going to talk, so I'm a basic scientist. So what I'm going to show is that I'm not going to change the world, by this thing. And the topic, what I'm going to tell you is a nice story, which is by accident. It's not I discovered something. We were trying to do something and just it came out. So, and most of the discoveries are made by accident. So what you see here is this is an aerial view of the center of the University of Iowa. This is a building and my lab is somewhere here. So this is our University of Iowa campus. So in this slide, I would like to give an overview of the research program in my lab. So since you know that thromboinflammatory process contribute to coronary artery disease and stroke, so the major focus of my lab is to study the interface between thrombosis, inflammation, and the vascular biology in context of stroke and atherosclerosis. And a couple of years back, NIH started a SPAN network. It's new, it's only 3 years old. And the main aim of this network is to bring together basic and translation scientists to systematically address all the prior methodological limitations of cellular protections. You know that 95% of the neurological protections, I think there were 1,600 which were successful in the preclinical trials, but nothing, when they came to clinics, all failed. So they have developed this network, and this is the site. If you want to go into detail, you can see these things. And there are 6 centers which was funded by NIH for the first span, and one was Carver College of Medicine, where I'm the PI on that, with Andy Cleara, who is a contact PI, Yale University, Johns Hopkins, MGH, Medical College of Georgia, and UT Austin. So what we do is, suppose you have a drug, and if you want to go, if you can put your drug in this trial, and if your drug works in this thing, so these are 6 centers, and we test this drug in different models, comorbid models, and if the drug passes the efficacy, you can tell the NIH it can go for the clinical trials, fast phase. And if the drug does not pass, then we just cut it. So recently, like 3 years or 4 years back, so I tried to focus on a different pathway. And that pathway is how to understand the metabolic plasticity contributes to platelet function thrombosis. So what you see here is the platelet receptors, and the signaling pathways that contributes to platelet function thrombosis. And all the green what you see are the current antiplatelet targets. And in the white are all the antiplatelet targets which are under clinical trials and could be potential targets. So for example, we have a abeximab which targets alpha-2b beta-3, one of the major receptors on the platelet, and then clopidogrel, Plavix, which targets the ATP. And then we use aspirin. People get stroke, then they are put on the aspirin, or combination of aspirin and clopidogrel so that you inhibit platelet function. And this aspirin inhibits COX. So when we have so many drugs, why we want to test more drugs? Why we want to do, either we want to do a basic science, or there's some limitation with every drug. So I think that, so these are some of the studies which you can go and look at the literature that this aspirin and P2Y inhibitors do not translate into clinical efficacy in 1/3 of the patient at risk for coronary syndromes and ischemic stroke. And they are like more potent antiplatelet agents, which includes like abeximab. Yeah, I cannot see. Anyway, so are associated with bleeding complications. So one of the things is that if you have a very strong drug, which is antiplatelet, then it leads to a bleeding complication, and you don't want that. So can you fine-tune between thrombosis and hemostasis? So now I come to topic of pyruvate kinase. It is heavily studied for last several years, and it just came by accident. I started going into this field. So this is a basic biochemistry. You know that there's a glycolytic pathway, and the glucose is converted to phosphoenol pyruvate to pyruvate and ATP. This is the last step of glycolysis, and this is controlled by pyruvate kinase. And then pyruvate then goes to the oxygenate phosphate cycle. It's a TCA cycle, and by LDH is converted to lactate dehydrogenase. under anaerobic condition. So this PK pyruvate kinase exists in 4 different isoforms. PKR, which is present on the erythrocytes, which is RBCs. PKL, which is in liver and kidney. PKM1 is present in the heart, brain, and muscle, and the PKM2, early fetal tissues and most adult tissues. And this PKR and PKL are encoded by PKLR gene, whereas PKM and PKM2 are encoded by PKM gene. So it's very important to note that unlike other PK that exist as tetramers, PKM2 exists as tetramer dimer. So these are made of similar monomers, but the dimer has different activity and the tetramers have different activity. So why PKM2 and not other PK isoforms? So as you know that most of the processes associated with platelet activation response to injury, including whether it's a platelet aggregation, alpha-2 beta-3 activation, granule secretion, spreading of clot detection, you need energy. So like normal cells in the resting platelets, the platelets depend on both the oxyphos and the aerobic glycolysis for the ATP generation. And this is happening in presence of oxygen. Whereas in the activated platelets, they exhibit a high level of aerobic glycolysis, which is shown in here. So, and dimeric PKM2, as you can see, is a tetrameric, and this dimeric PKM2. This tetrameric PKM2 and the PKM1 drives the oxyphos, whereas dimeric PKM2 drives the aerobic glycolysis. And why is it known as aerobic? Because this is happening in presence of oxygen, and this was first observed by scientist Warburg. It's also known as Warburg effect in tumor cells. And this molecule is very interesting because in addition to having its glycolytic activity, it also possesses protein kinase activity that might regulate platelet signaling and pathways. And this dimeric PKM2 also interacts with transcription factor STAT3 and promotes inflammation by regulating IL-1 beta and IL-6. So as I told in the first slide that most of the processes involves thrombosis and inflammation, including stroke, heart attack. So I started thinking, so I was in a seminar, I started thinking, oh, this molecule is regulating inflammation. And if it regulates thrombosis, then it could be a very good target because you have to use one drug to inhibit both things, which happens after reperfusion when you give a TPA or a mechanical thrombectomy. So if there are a lot of biochemists here, so people will ask, okay, this OXPHOS pathway, it generates 32 ATP of molecule of glucose, whereas this lactate pathway generates only 2 ATP molecules of glucose. So why platelets prefer activated this aerobic glycolysis for ATP generation? So I looked at the literature and I found that despite low efficiency, the rate of ATP generation is 100 times faster in the aerobic glycolysis compared to the OXPHOS pathway. So this could be one of the reasons, because when platelets get activated, it's a very fast process. You get an injury, you start having clots. So it's a very fast process. You require instant source of energy. Maybe that's a platelets are activating that. So as we know that because glucose is sufficient and if inefficient but accelerated rate of ATP generation might be well suited to fulfill the short-term and high energy demand of the activated platelet in order to form blood clot in a shortest possible time. And another reason could be that aerobic glycolysis regenerates NAD plus a cofactor which is necessary to maintain high glycolytic flux from top to bottom, which may facilitate faster glucose metabolism to meet the short but high energy demands of the activated platelets. So based on this rationale, we build up a hypothesis that limiting PKM2 dimerization or deletion of PKM2 will inhibit aerobic glycolysis and thereby platelet function thrombosis. So approach was first to use a inhibitor, which is a small molecule amylase 5. It's available from the companies make it. So we wanted to just limit the PKM2 dimerization, and this molecule binds between the dimers and stabilizes the tetramers. So now the PKM, when you give this molecule, very specific for the stabilization of parameter. It does not interact with other molecules. This has been already shown. My lab was not the first to show that. And then we used the platelets, the genetic model where we just delete the PKM2 from the platelets to show that this is indeed a phenotype of the platelets and it's not an off-target effect of a drug. So first important thing is that what is the expression of PKM2? So we take the human platelets. So these are Western blots. 1, 2, 3 are three different individuals of 3 different mouse. And you can see a nice PKM2 expression both in the human and mouse platelets. and we use beta 18 as a control. So which question was, so this is all hypothesis. So we wanted to understand in platelets whether there is indeed there is an increase in the dimerization of PKM2 on activated platelets. So what we do is that these are resting platelets, and these are platelets which are treated with conluxin and thrombin. These are, I showed you, conluxin activates the GP6 pathway and thrombin the PAR receptors. And these are different concentrations. As you can see here, between this resting and activate, you can see there's a nice increase in the dimer tetrameric ratio, which is quantified here, and each point is a single sample from a human sample. And these are all healthy donors. And if you treat ML2 you can see there's a decrease in the PKM to dimeric tetrameric ratio, as you can see here. So this shows that dimeric PKM2 is upregulated in activated platelets, and if you use ML2 which stabilizes tetramers by limiting PKM2 dimerations, you can limit PKM2 dimerization in the platelets. So this cartoons, so we shows that we want to test if you limit PKM2 dimerization, whether there is a decrease in the glucose uptake, and also there is a decrease in the lactate production. These are the results. As you can see here, this is a resting platelet, and this is the platelet activated conluxin and thrombin. 2 different doses. Each point is a single human sample. As you can see that if you ML2 it emits glucose uptake in a dose-dependent fashion. Also, there is a decrease in the lactate production in the activated platelets when you activate with conluxin or thrombin, suggesting that ML2 by limiting PKM2 dimerizations inhibits glucose uptake as well as there is a decrease in lactate production. So, obvious question is, if you stimulate platelets with agonist, and then you pre-treat platelets with this ML2, whether you see any inhibition. So what we do is you take you isolate platelets or platelet-rich plasma, pre-treat with the inhibitor at different doses, and do a platelet aggregations with a agonist, which is GP6 agonist like conluxin collagen, and GPCR agonist, TRAP and ADP. On the top, it shows the aggregation profile. On the left-hand side is shown the aggregation. Each point is again from a single human donors. As you can see, that percent of platelet aggregation is inhibited with ML2 in a dose-dependent fashion, both with the GP agonist, conluxin collagen, and GPCR agonist, suggesting that limiting PKM2 dimerizations inhibits integrin inside outside signaling. These are some of the markers on the platelets. So we want to look at whether inhibiting PKM2 dimerization also inhibits the P-selectin, which is alpha-2 P-theta activation, or alpha granules or dense glucose secretion, which is P-selectin and alpha granules. As you can see here, that we use 2 different doses, and it's also dose-dependent fashion. If you inhibit, the human platelets which are treated with ML2 they have a less PEC binding, which is decrease in alpha-2 B3 activation. They also have less CD60P fluorescence, which is the P-selectin secretion as well as the ATP generation, suggesting that ML2 inhibits alpha-2 B3 activation, alpha and dense glucose secretion in human platelets. Next question is that whether the ML2 is also inhibiting the integrin outside in signaling. So we went for a clot retraction assay, and you can see this is a vehicle. This is ML treated with 50 micromolar, this is with 100 micromolar, and this is on the left-hand side from the quantification. This is with the vehicle, black is with the 50 micromolar, and this is with the 100 micromolar. As you can see here that the clot retraction was significantly inhibited in the human platelets which are treated with ML2 after thrombin stimulation, and this was dose dependent. So this is all good because we are using in vitro situation, in vitro because, and we were using platelets, purified platelets or PRP, which is platelet-rich plasma. But in real situation, you have whole blood and the other cell types. So we went for another assay, which is a microfluidic chamber assay, where you coat with collagen and then you pass the whole blood. And the beauty of this technique is that you have parallel channels, and one channel you can put a vehicle-controlled pre-treated whole blood, and this is ML2 pre-treated whole blood. And then you can quantify over time. So as you say, this is you can quantify the thrombus growth, and this is a percent of area covered by the platelets. As you can see here, again that amyloid-2 inhibited percent of area covered by the activated platelets, and also there was decrease in thrombus growth size, suggesting that by limiting PKM2 dimerization, you can inhibit platelet aggregation in vitro in whole blood. Then we went to look at the mechanism, which is because I told you that PKM2 also has a protein kinase activity. So we look at the mechanism. So here you show this is the resting platelets, and these are the platelets treated with convulsion and thrombin. As you can see here, the amyloid inhibits phospho-AKT as phospho-GSK3 beta. And here is a quantification. You can see that at 2 different concentrations, amyloid inhibits both the phospho-AKT and the phospho-GSK. And we use total AKT and total GSK3 beta as the loading control. This is very new, it's not published. So we went and looked at the metabolomics on the global snapshot. And we just used 3 samples because it's run on triplicates, and 1 sample costs $500. So we look at, so we don't have so much fun, so I just wanted to start. Now I got some money, so we are going to continue this study. So this is a global snapshot of metabolites. This is with the resting platelets, this is with convulsin, which is agonist for collagen, and this is inhibitor. You can see all these things. But then you have to use a tool like gene analysis, where we found that inhibiting PKM2 dimerization with amyloid inhibits glycolysis, glycerol phosphorylate metabolism, as well as arachidonic acid metabolism, which is the formation of thromboxane. That's one of the mechanisms how platelets get activated. And this is the quantification from 3 different individuals. Of course, this is all preliminary data because this is just 3 individuals, though it's significant, but we are going to repeat this thing with more than 20 samples, and we are in the process of working with the core facility to generate more data. So until now, what I have shown is that if you limit PKM2 dimerization by amyloid it reduces glucose uptake, it results in reduced lactate formation, somehow indirectly or directly, we don't know the mechanism. It inhibits the phosphorylation of PI3 kinase. It also inhibits the phosphorylation of AKT and upregulates the GSK3 beta, leading to reduced platelet activation, aggregation, and thrombosis in human platelets. So thrombosis is not a correct word to use here because what we are doing is all in vitro, because it's not possible to do in vivo. So once these studies were confirmed with the human platelets, then we did same study with the mouse platelets, and we found the similar results with the mouse platelets. So because of less time, I'm not going to show the data with the paper, but it's all published, so you can go and see the papers. So obvious question is when you are using inhibitors, the first question people ask whether it has an off-target effect. So we went and made we crossed the PKM2 flox mice with a PF crow mice, and this is the PKM2 deficient mouse, which is a platelet-specific PKM2 deficient mouse, and this is the wild-type controls. It's important to know that all the studies in my lab, we use literate controls. So this is a Western blot to confirm that there is no PKM2 expressed in the platelets of a PKM2 deficient mice. And this is an additional control to show that when you delete PKM2 only in the platelets, there is no deletion in the macrophages. So this is a cartoon showing that here now, because it's not possible because PKM2 had 2 different isoforms, and at this point, I did not have the tools to just remove the dimeric PKM2, so we have to just remove both the isoforms of PKM2, which is dimeric and tetrameric. So whole idea was that if we are deleting both these things, whether we can see a decrease in the glucose or lactate production or not. If not, then this is not a nice model. So we went and looked at the lactate production, and also glucose is already published. I just put this thing. So this is the resting platelets. You can see there's no difference. But if you delete PKM2, there's a reduction in the lactate concentration. Though that is significant, but it was not so strong effect with what we see with the human platelet. However, this suggests that this mouse model can be used to test the effect of PKM2 on platelet function thrombosis. So then we did the platelet aggregation studies. And I would like you, so on the top are aggregation profile curves from the wild type and platelet specific PKM2 deficient mouse. And I would like you to concentrate on this red things. So when you use a higher concentration of convection, we did not see any difference. So this was surprising. But when we use suboptimal concentration of agonist, there was a very strong effect. Suggesting that if you inhibit PKM2 dimerization or PKM2 with a suboptimal concentration, so if you use a suboptimal concentration of agonist, then you can see a defect in platelet aggregation in the PKM2 deficient mouse. Again, this is the same assay where we look at the alpha 2B3 activation, alpha granulosecrase and dense granules. You can see here that with there's a reduced alpha 2B3 activation as well as P-selectin and ATP secretion in the platelet specific PKM2 deficient mice compared to the wild type control. Then we look at the clot retraction, which is this is a assay to understand the integrin outside insulin signaling, because then fibrinogen binds to integrin and leads to outside integrin signaling. So these are the wild type platelets. These are different time points, and this is the platelet specific PKM2 deficient mice. And you can see that the clot retraction was inhibited in the murine platelets lacking PKM2 after thrombin stimulation. And again, this is the same mechanism what you show in the mouse plate. With the human platelets, we want to confine the mouse platelets because these are specific mouse which are deficient platelets. You can see here that there is a decrease in the phosphorus AKT, as well as there is a increase in the PSK3 beta by total G squared beta, as you can see here. So this is all good. So this is all in vitro. So the question is, can we reproduce this in vivo? So in my lab, we do a lot of intravital imaging, which is real-time imaging. So in this assay, can you see this movie? Yes. Okay. So here is a procedure where you level the platelets with calcium green and you infuse in the retroorbital plexus, and then you make an injury on the artery using a ferric chloride, and then you can see the thrombus formation in real time. And then you can record, and then you can evaluate all the videos. So what you saw here, this is initially you saw there was some thrombus formation, and this is 18 minutes after 5% ferric chloride injury. So very powerful technique. If somebody wants to test a drug, you can give the mouse. If the drug interacts directly with the platelet receptors and inhibit, you can see the assay in 1 hour. It gives a direct evidence whether the drug is working, is inhibits platelet or not. You can have all this effect, but here you can show the in vivo effect in a real in vivo model. You can also test other drugs. You can give the mouse, you can take out the platelets and then see the effect, but this is a direct evidence, and you can record everything. But these are carotid arteries, because my lab is interested in stroke, and most of plaques get into carotid artery and it goes up, so this is a model. But these arteries are very big, and it's not easy to image. So we have another assay which is a micro vessels. These are like 150 micrometer vessels. This is a vein, this is artery. You can see a nice vessel. There's no thrombus formation. So this is a baseline. And these are single platelet adhesion, which is after 2 minutes after ferric chloride injury. Now, because what this does is this ferric chloride injures the endothelium, exposes the subendothelium, then collagen exposed, and the platelets start binding to the collagen receptors. This is 9 minutes after injury, so if there is a drug or if the molecules playing a role, you can see inhibition in this thing. So this is just 9 minutes after. You can see a nice thrombus formation on the injured vessel. And this vessel is occluded, so you can just, this is after, I think it's after around 15 minutes, and you can see. So by this technique, using micro vessels, you can quantify single platelet adhesion, which is the first step of thrombus formation. You can look at the emboli, you can look at the embolization, you can look at the mean occlusion time. So you can use a lot of parameters. So we use this assay, and then what we did is that we use a platelet-specific knockout mice. On the left-hand side is shown the microphotographs, and this is shown the quantification. And this is the thrombus growth. So what we found that if the mouse which has deficient of PKM2 in the platelets, the time to occlusion was prolonged, as well as the thrombus growth fold was decreased in the platelet-specific PKM2 deficient mouse. And similar results were obtained with the female mice. So these are all with knockout. Then we'll want to look whether, because we are affecting the thrombosis, so the important question is whether it also affects hemostasis. And if the drug affects hemostasis, then it's like other drugs. So why people are interested in alternate drug? So as you can see here, that the bleeding time was not different, but they were compared between the wild type mouse and the platelet specific came to different mouse, both in male and female. So now we want to test the efficacy of the drug. So we use the same wild type mouse, and we use 2 different concentrations, and we have some people who are like MTech in pharmacology, and they came from industry, so they calculate the dose which mouse can go. So you give oral gavas, and then you can see the time of occlusion. This is the control with the vehicle control, and these are 2 different concentrations. You can see a very good effect. This effect is better than the knockout mice. And the reason is, I'm sure somebody is going to ask the question, but I can give the answer. The reason is because there we are using a platelet-specific knockout. Here we are giving an inhibitor which also affects the function neutrophils and monocytes, which also contribute to thrombosis. I'd like to conclude my talk. Hopefully, what I've convinced you is that platelet activation increases PKM2 dimer formation, which regulates PI3 kinase mediated AKT and GSK3 signaling, and thereby modulates platelet function arterial thrombosis. Targeting dimeric PKM2 inhibits arterial thrombosis without altering hemostasis in vivo. So what I showed you is all healthy donors, but healthy human does not get disease. So right now what we are doing is, we are doing some studies where we are trying to target dimeric PKM2 and looking at whether it affects platelet function in the platelets from the coronary artery disease patients or the obese patients. We are recruiting some donors and controls. It's very difficult to have diet-induced obese patients and they match the same control with the same ethnicity, with the same age. So we are doing and working with some doctors. I have some couple of MD PhDs in my lab, so they are working on that. And this is a new area we are targeting. So what is the effect of targeting PKM2 dimeric on metabolic pathway platelets? And looking at the metabolic data, we have found multiple targets. And that's the whole area of, you will see in a couple of years, a couple of papers coming from my lab, and nobody's working on this area. This is a very hot area, and this was by accident. This is all published in Blood, and we have filed a patent, and we are looking for that. So, this is my lab. I used to have like 10, 12 people. Now this is the lab people. Most of the work, what I showed you, is done by Manas, and the other people who left my lab have become research professors in different universities. And my lab is funded from NIH R35, NIH R01 from NINDS, and the NIH SPAN, where we do multiple testing. And I am also the actual investigator of the American Heart Association. And recently, I was given a Distinguished Scholar Award and funds for developing this into a novel therapy. Thank you.
Yes, thank you. Thank you, Dr. Chauhan, for a very excellent presentation and very basic research, yet very important and had a lot of translation. How PKM2 inhibits platelet aggregation. It's ML265, I think. You know, I remember when we were in Leh, we used to have a lot of venous thrombosis patient, and they used to die within few days, and without, clinician diagnosing it, it is because of the venous thrombosis. So we took a very big project analyzing it, and mainly the clotting factors were responsible. Some of them were getting activated, which was leading to platelet aggregation. So very important study. It had a lot of clinical implications. Any quick question, one or two?
So before somebody asks a question, so I will tell because...
Good morning, sir. I'm also working on the same area. As you show the plot of dimer and tetramer, as we know, the dimer and monomer also, they have nuclear translocations. So it is better to check the monomer also.
Yes, but these are different type of gels, not it's a non-native gel.
Sir, if you should run a native gel also, we get the 3 bands, monomer, dimer, tetramer, in the same level. So it's better to check the monomer also, because monomer and dimer, they have the nuclear translocation function. And one question, if you knock out the PKM2, there is a feedback loop. So it's better to check the PKM1 level as well.
Yeah, we have checked. There's no difference. But we have, so you can follow the papers, you will find all the data there.
Okay.
So I just, because in half an hour I cannot come, so you can. So we have all type of mouse. We have neutrophil specific knockout PKM2. We have a myeloid specific knockout of PKM2. We have the endothelial specific PKM2. We have the smooth muscle specific PKM2. So we have multiple projects. We work on multiple things. You can find all the data. You follow my papers. Some have been
Sir,
published.
You use one activator, one DASA is also there. It's better to check the another activator. And if you check the inhibitor also, it will it will increase the research area. Like you test the activator, you test whatever the effect of inhibitor as well on that simultaneously. So it will be more clear picture.
You are right. So DASA,
He will be available during lunch. You can discuss. Maybe we can have 1 or 2 more questions from others.
Thank you, sir.
so I just like to comment on what she said. So I was several years back, I think they had a paper from Lay about DVT, and I think it was published in Blood. I have not shown the data on DVT, but we have so one of my junior faculty got a grant. We have data on DVT. It works very well.
Thank you. Dr. Chan, just one simple question actually. Yeah, what about I think this PAR antagonists like ergotroban and all, actually they are available for the similar kind of indication. Whether this PKM2 has any effect on the PAR pathway also, protease activity receptors pathway?
Yes, I showed the data on the thumbnail.
So is going to inhibit actually that molecule actually, what you make a PKM2 molecule?
So this inhibits the pathway. So only the point is here. So if you see that data, there's no bleeding.
Okay.
If you use a high concentration of any agonist, this is not going to work.
Okay.
If you use suboptimal concentration, it's going to work. So that's what we do with aspirin and other things, right?
Okay.
So if you use aspirin and if you use a high concentration of this thing, aspirin doesn't work. But that's what we want. We want to inhibit the hyperactivation of platelets so that person does not die. But if you inhibit completely platelets, so it's like putting more gas, so more in US we say gas, so putting fast accelerator. You can go at faster rate, right?
Okay.
So if you want to go from here to there, right? So if you put more gas, so you are faster going there. But if you put less gas, you will still reach, but more time. That's what we want. That you inhibit hyperactivation of platelets, which affects in the type into obesity or other conditions, and then prevent platelet and make platelets calm. Then inhibiting the whole pathway, because same pathway controls thrombosis and the same pathway controls hemostasis. So there's a fine balance.
Okay.
So if you repeat the experiment with a high dose, it's no more.
Okay, thank you very much. Thank you very much for your talk. Now I would like to invite Dr. Surajit actually Kamakar from INST Mohali for his talk. Actually, he is going to talk in the topic which is the preclinical therapeutic implication of nano melatonin for neurodegeneration and inflammatory disease. He is one of our collaborators also. Now, Dr. Surajit, please.
Good morning. Thank you for the invitation and the kind introduction. So I am Surajit from the Institute of Nanoscience and Technology. So today I'm going to talk on preclinical therapeutic implication of nanomelatonin for neurodegenerative and inflammatory disease. Basically, last few years we are working with this molecule, melatonin, which is neurohormone and released from the pineal gland and has control over the sleep-wake cycle and the circadian rhythm. So basically, the melatonin synthesis and release is controlled by the suprachiasmatic nucleus in the brain. Suprachiasmatic nucleus in the brain, and when light falls on the retina, so it got stimulated and causes the inhibition of the synthesis and release from the pineal gland. So, but when the light got diminished...
Surajit, can you use please mic actually, because, thank you.
So when the light got diminished during the late evening, so then the suprachiasmatic, that inhibition removed, and it started the release, synthesis and release of the melatonin, and it reaches in the peak during the midnight, and that introduced the sleep. So this is their biological function. So basically, the mechanism of action of melatonin is there are, melatonin receptor, MT1 and MT2, they are G protein coupled receptor. So when melatonin as a ligand bind to it, it causes the inhibition of the adenylate cyclase as well as the guanylate cyclase. And to reduce the cyclic AMP and cyclic GMP, on the contrary, they stimulate the phospholipase C to trigger the IP3 and diacylglycerol mediated pathway for the activation of the downstream signaling. And there is also receptor present in the retinoic orphan receptor in the nucleus, and also in the MT3 receptor available in the cytosol. They, receptor independently, it can execute its action. So when there is a inhibition or absence of melatonin, it causes the sleep disturbance, and it is also associated with several kind of diseases, life-threatening diseases. The major gateway is the inflammation, and that leads to these several life-threatening diseases like cancer, diabetes, cardiovascular, neurological. So on the contrary, melatonin having the well-known anti-inflammatory, antioxidant property that is executed mostly due to the inhibition of the NLRP3 inflammasome, the caspase activation, and the cytokine release and the pyroptosis. It has also the inhibitory action on the nuclear factor kappa B to translocate to the nucleus to prevent the COX and INOS mediated pathway inhibition. So melatonin is very well known as a supplement or as a over-the-counter medicine to use to correct the different kind of insomnia, and it is well known reported in the literature having these beneficial effects that like immunomodulation, regulation of the circadian rhythm, mitochondrial function, so anti-inflammatory, antioxidant property. But when we try to use this melatonin as a molecule exogenously, it faces certain challenges. So those limitations are they are having very short half-life and the poor solubility, low viability, extensive fast pass metabolism they are subjected to. So we hypothesized, if we use a appropriate nanodelivery system, we may overcome these certain challenges and we can potentiate its therapeutic efficacy. So with that aim, so we have prepared different kind of nanoformulation in our lab utilizing this melatonin. But today I'm going to talk with the, melatonin-loaded polydopamine nanoparticle or nanocomposite that we have prepared, and we have explored this against this Parkinson's, this Alzheimer, and the diabetic retinopathy. And these are all preclinical models. So first, I'll touch the three. So I'm going to, go very quickly. So the first project is the, exploration of the melatonin polydopamine nanoformulation for the Parkinson's disease therapy. So we have prepared and tested whether this, nanoformulation having some therapeutic efficacy against the Parkinson's disease. So I'm just skipping the, introduction because we are out of time. So basically, Parkinson is a dopaminergic neuron death, and there is a cytoplasmic, like Lewy body, deposition. So those are all characteristic. But we have particularly focused here synucleinopathy. Synucleinopathy, what is that? So there is a, like the alpha-synuclein get highly phosphorylated at its, serine 129, and that causes deposition of the as a Lewy body, and that is toxic and causes the neuronal cell death. So we targeted them. So, and why this combination? So as because there is dopaminergic cell loss, so we thought maybe if we can use this dopamine, that may replenish those kind of dopaminergic, neuronal needed. And melatonin is already known for the, cytoprotective. So this combination, that's why we have tried here. So this is the nanoformulation we have prepared. So utilizing the modified reverse emulsification technique, they are showing 70 to 90 nanometer with a negative zeta potential. So utilizing them, first we have developed our in vitro PD model. So we have used the rotenone, that is a very well known like the PD model introducer. So we have used two neuronal cell line, SHSY5Y and IMR32, and we have checked the cell viability, and we could find the IC50 value. And further, we have propagated all the work with the SHSY5Y for this project. So after determination of the IC50 value, we have challenged the cells with this IC50 value, and then we have treated them with this nanoformulation to see whether they have protection or not. So what we could find here, so they are rotenone causing about 500 nanomolar, causing like 61% of the cell death. So when we use the cytoprotection getting into the means like the cell viability is getting up to the 92%. So significantly prevent the rotenone-induced cell death. So reactive oxygen species generation is a rotenone-induced model. It's a common factor. So we could see the treatment, the nanoformulation treatment, significantly reducing the ROS level and preventing the apoptosis that induced by the rotenone here. Then we have checked the mitochondrial membrane potential. So you could see the utilizing the JC1 dye. So the monomer is the like the damaged population. You can see the green one here. After the rotenone, it is lightened up. So that means they are introducing the mitochondrial damage, which is prevented following the this nanotherapy. And the last column, if you can see. Also, there is a upregulation of the caspase-3. You can see after the rotenone treatment, that also inhibited following the therapy. So they are working through this pathway. Then with the RT-PCR, we have checked certain anti-inflammatory genes and the apoptotic genes. So we could see the rotenone-induced rotenone is causing like the interleukin 1B, NF-kappa B, AKT. So they are highly upregulating. So that is checked or inhibited following this therapy. And as because we said like that we are particularly targeting this alpha-synuclein, the synucleinopathy. So monomer to oligomer is a problem. So for the Lewy body formation, so you can see after the rotenone treatment, the last column of the Western blot, there is a oligomer formation, the trailing. So that is inhibited following the dose-dependent treatment. If you see the left column, and also there is inhibition of the caspase-3. Then organotopic brain culture model, we could see the rotenone is causing the alpha-phosphosynuclein here. Also the activation of the caspase-3. So that is diminished or inhibited following the nanotherapy. Interestingly, rotenone is causing the, we also check the tyrosine hydroxylase. So in the rotenone panel, you can see that is, completely abolished there. So that is retained in the basal level following the nanotherapy. So they are also protecting the dopaminergic neuron in the brain slice culture. Then we have checked the biodistribution. They are well biodistributed and excreted from the system time dependently. And we could see they are located in the brain for a longer period of time. And after the H&E staining, we could not see any kind of aberration in the histological profile. So they are highly biocompatible. Then we have developed the rotenone-induced PD model, and the gait analysis we could see there there is a significant behavioral changes and the sensory motor dysfunction that triggered by the rotenone is prevented by this nanotherapy. And this is the model after the establishment. You can see the dopaminergic neuron is completely lost in the PD model. So, which was retained, after the treatment here in the basal level, and also the alpha-synuclein that is, gone up through the rotenone treatment, also gone down in the treatment group. So we have represented graphically that one. They are significantly doing this. So we can conclude like this nanoformulation is causing the inflammation, anti-inflammatory as well as the anti-apoptotic effect, and reducing the serine phosphorylation at 129 to execute this therapeutic efficacy in this preclinical PD model. So next, we have used the same nanoformulation, but it is differently prepared. But the key goal is here to check whether they have the anti-aggregation property against the beta amyloid. So this is the pathway you probably know, like when the amyloid precursor protein produce this amyloid beta short peptide, so that form the oligomers, and then they get aggregated and form a, like the cause of dementia and the amyloid plaque in the cells. So we targeted and try to see which pathway or which steps this therapy can, disintegrate or prevent the aggregation in our model. So first we have checked the stability, and in a different pH. So we could see they are highly stable, but they are, degrade in the higher pH, means the alkaline condition. They are highly stable in the NIR, charge, but they are cannot be sustained into the H2O2, and after that, we have checked the dose dependently the photothermal ability of this nanoformulation, shining the NIR laser. So we could see the dose dependently, when we concentration dependently use them, so they generate the higher temperature. So, and from there, we have done this experiment where we have intermittently shined the NIR and turn it off. We could see the temperature is rising when we are turning on the NIR light, and it is going down also when we are turning off. So this is the NIR image of the particle. And also we could see, as because this is also the melatonin encapsulated formulation, so their releasing profile is they are releasing about like 52% when we are not shining the NIR light. But when we shine some point the NIR light, they are releasing the burst release of the melatonin in this system. So we have done, as because our goal is to check whether they have the anti-aggregation against the beta amyloid or not. So we have done two assays. One is the amyloid beta aggregation assay, and another one is the amyloid beta protein misfolding cyclic amplification assay, the lower panel, the first one. So and we have monitored their aggregation profile with the type 11T. So what we could see in the both cases, they are preventing when we introduce the nanoformulation to the reaction mixture, they are preventing the growth phase. And when we further, charge with or shining with the NIR light in the third panel, they are completely disintegrating those aggregation. So TMMH representation here to see that kind of disintegration property. The third one is the NIR. So they can, this nanoformulation can able to quench the ROS. We have done that one. And their axonal growth assay we have tested. So they are highly, biocompatible till 15 days we have observed. So there is no toxicity. But when we introduce the amyloid beta to this axonal growth assay, they immediately die within the 96 hours. So this is another in vitro assay where we have used the amyloid beta to generate the AD model. So you can see after the treatment with the amyloid beta, the plaques are available. You can see within the cytosol, the red fluorescence is representing. So the treatment with this nanoformulation can reduce that amyloid plaque. And when we shine the NIR to it, it is completely inhibited or disintegrate that plaque within the cell. We have also the mouse brain slice culture we have done, where we can see the amyloid beta treatment is depositing that plaque within the mouse brain culture, and there is also activation of the microglia. So that is reduced, and NIR shining completely abrogate them. So also we have done the ocadoic acid model induced AD model. So there we can see that when we treated with the ocadoic acid, amyloid beta is upregulated. And in the second, Western blot, you can see the similar condition when we treat them, that upregulation is downregulated and completely, inhibited following the nanoformulation with the NIR. So we can conclude like that they have the anti-aggregation property, this nanoformulation, and the photothermal property and cytoprotection property against this preclinical Alzheimer disease model. And this is the last project. So where we have used the dopamine melatonin nanoformulation for the diabetic retinopathy. So basically in the diabetic retinopathy is a diabetes mellitus associated complication where the neuronal apoptosis and the microvascular complication happen due to the highly oxidative stress, inflammation, apoptosis, angiogenic factors. So dopamine and melatonin both having, antioxidative, anti-inflammatory, anti-VEGF activity. So why not we try to develop a therapy? So this is our model. And this is our hypothesis. So based upon this property, we have tested against this diabetes retinopathy model. So this is the synthesis and characterization method. So they are spherical inside, and there is a 90 to 120 nanometer in range, and there the FTIR and the Raman spectroscopy showing they are non-covalently interacting with them. So they are becoming crystalline to the amorphous in the XRD study. They are encapsulated in the nanocomposite by the DST study, and they are control release profile we could see in the ex vivo model. So then the cell we have treated, the rhodamine treated, fortified this nano formulation. We could see the cells are nicely uptaken these nanoparticles. And after that, we have developed the diabetic retinopathy model using the retinal epithelial cells, and we have used as a high glucose, we have used as a, to introduce that model, and we have monitored the caspase-3 as a caspase-3 and the VEGF, because those are the apoptosis and the angiogenesis is the challenge in this model. So we have developed that one, and after developing this model, we could see that when we challenged first with the high glucose and then treated with this nanoformulation, they are preventing this level, VEGF and the caspase-3 here. They are also reducing the reactive oxygen species in the intercellular level, and also the high glucose induce the cell death or mitochondrial damage is prevented by this nanotherapy. They are also, this nanoformulation also reducing the apoptosis introduced by the high glucose. Then we have done the biodistribution. We could see they are located in the brain, and also they are accumulated into the liver and the kidney. They are mostly the highly metabolic organ and may be attributed due to the excretion of this nanoparticle. They are highly safe. There is no changes in the histology. So pharmacokinetic study we have done, and where we can see they are increasing the median resident time of this melatonin maybe 20 times. So from 1.6 hour, the bulk one, to 26 hours. So due to this nanoformulation. And then we have used the streptozotocin. This is a very standard model for the diabetic retinopathy. So the blood glucose level, that body weight alteration by the streptozotocin is returned to after the therapy. And we could see the open field behavioral study, the cognitive impairment, and they become normal, almost normal after the treatment. Then we have done the retinal section where we could see the streptozotocin group showing the swollen ganglion cell death in that particular STZ, which was prevented in the therapeutic group. So, and we also see the flattened that thickness of the streptozotocin is also become compacted following the therapy. So then we have done the NG2, which is a marker of the retinal it is a marker of the pericytes that is residing into the molar cell layers of the retina. So that is completely gone or inhibited following the STZ treatment, and they came back after the therapy. And also we monitored the CD31 as a blood vessel marker. So we could see they are highly enlightened, means stimulated following the STZ, which is also returned to the normal following the therapy. That is the next slide. Next panel is for INOS. That is a very well known, like the means like the anti-inflammatory marker. So that is lightened up following the STZ therapy and diminished following the nano formulation group. So finally, we have checked the involvement of the VEGF and caspase-3 here. So you can see the VGF is highly stimulated following the steroid therapy, which was become control or almost level of the control following the therapy, and that is supported by the Western blot. So you can see the caspase-3 activation and following the STZ, and also there is a VGF upregulation, pixie delta upregulation, which are inhibited following this therapy. The last panel of the Western blot. So we have, presented them in the graph, bar diagram here. So they are significantly controlling this pathway for executing this kind of efficacy. So we can conclude like this nanoformulation is showing the anti-diabetic, anti-retinopathic activity in the both in vitro and in vivo model. So through the inhibition of caspases, VGF-dependent pathway. So last slide is the acknowledgement. I'd like to thank my director, my collaborator, Dr. Roy Chaudhary and Professor Sharma here. So he helped us a lot. Thank you, Professor Sharma, and the funding agency, and lastly, my hardworking students. So these are the students mostly involved with this project. So thank you very much for your kind attention. I'd like to take some questions.
Thank you, Dr. Surajit. Actually, we can have one quick question because after that we have to start another session. Any question, please? Okay, there's no question. Thank you very much, Surajit. Now I invite all the speakers on the dais. Please come so that we can felicitate.
Thank you all the speakers for your wonderful presentations. Okay, let us continue with another session. It is one of the emerging area on the pharmacogenomics and precision medicines. So without wasting much time, I would like to invite Professor K.B. Tiku. He is HOD of the Department of Pharmacology and Toxicology, NIPER, S.A.S. Nagar, as a chairperson. And Dr. Abhay Sangamwar is a co-chairman of this session. Associate professor working in the Department of Pharmaceutics. Please come on the dais. and do the needful. Thank you.
Now we are heading towards the next session about the session 11, pharmacogenomics and precision medicine, with the first speaker, Dr. Veera Suvarna. Dr. Veera Suvarna holds MD in pharmacology from Grant Medical College and Sir J.J. Group of Hospitals, Mumbai. He also holds a master's in pharmaceutical medicine from Hibernia College. He was a medical advisor at Lethico Pharmaceuticals Limited, Indore, and head of operations at Pfizer India.
There is some delay in her flight, so we are going to start from the second speaker, Narahari Shastri. Dr. Narahari Shastri, I would like to invite Dr. Narahari Shastri. Just a small introduction by Dr. Sangamwal.
Sorry. Now, Dr. G. Narahari Shastri is currently the director of CSIR, Northeast Institute of Science and Technology, Jorat, Assam. Prior to joining the director, he headed the molecular modeling division at CSIR, Indian Institute of Chemical Technology in Hyderabad. He completed his first postdoc from Hebrew University with Professor Sassan Sheikh, and subsequently second postdoc from University of Freiburg, Switzerland with Professor Thomas Bailey. He is a recipient of several awards, which includes J.C. Bose National Fellowship of the Department of Science and Technology, New Delhi. He published more than 300 research papers in reputed journals. With these great accomplishments in his life, I invite Dr. Shastri for a talk.
Caller mic. Change the. Thank you very much. At the outset, I would like to thank
the organizers for giving me an opportunity to come here. I have been visiting NIPER from 1999 onwards, and it's a pleasure to be here. Can I have my slides? So today, my lecture will be slightly different from the lots of intense clinical data that you have been hearing from the morning. So I just wanted to look at the different perceptions that we have on our understanding of disease, health, and drugs. While the pharmaceutical chemistry, pharmacology, pharmacognosy, pharmaceutics have played a very important role, there is a separate vertical which separates it from medicine as well as the natural sciences, chemistry and biology. I just wanted to see from chemistry, it's just doing experiments, creating your own molecules. And if you look at biology, this is like observing. So they are quite different, and chemistry is sort of associated with diversity, whereas biology is associated with complexity. But how about biochemistry? Biochemistry is slightly different, but what is more different is chemical biology, doing biology with chemistry. While we have been getting divided into these perfusious slots that are there, we are very happy to see that all of them are coming together now and then talking to each other. So this is probably possible with the drug discovery or biology or mathematics. So 1950s, if you look at probably the drug discovery is associated with pharmacists, pharmacologists, and probably chemists have played an important role biologists have entered, and now computer scientists also have entered in the area of drug discovery. So earlier we have logic, representation, reasoning, these things have played a very important role. And now probabilistic modeling, learning, computations are also playing a very, very important role in the drug discovery. So when you look at the transition, we are going from the conventional mathematical models to probably artificial intelligence and machine learning. I will try to go through it. There are different scales. You have physiology, there are clinical parameters, there is biophysics, and also physical chemistry, and when you go to the bottom, you have quantum mechanics. So for example, there is a theory called there is a drug receptor interaction. There is a small molecule with bind with the known receptor or unknown receptor or somewhere and elicits a biological response. So the host allows only a specific guess into it, just like the lock and key model. So this molecular recognition and specificity appear to be very important, and this particular model was there close to a century. And we are also having a thought process that there is a disease which has one target and one drug. Now we know that this is probably not a very acceptable idea. So we also have receptors are usually related to proteins, and to predict the three-dimensional structure of a protein from the amino acid sequence, or how does a protein fold into the structure, is a very, very interesting question in its own right. So nature does this protein folding. Can we understand this act of Mother Nature, or how a three-dimensional structure comes from the primary sequence? So there are several questions that are there, which led to different fields like computational biology, bioinformatics, chemoinformatics, molecular modeling, so on and so forth. So when you look at a very quantitative way of understanding, this is more than 2 centuries old statement, that when you can measure what you are speaking about it and express it in numbers, you know something about it. But when you cannot measure it, when you cannot express it in numbers, your knowledge is of a meager and unsatisfactory kind. It may be the beginning of knowledge, but you have scarcely advanced to the stage of science. So this is something. Can we subject biology to mathematics, or medicine to mathematics, or what we are talking about, this thrombosis or ischemic stroke versus cardiovascular complications, and all of them, is it possible to subject to mathematics? That's the question. So as somebody like me who started with computational quantum chemistry, you can subject everything to mathematics, provided it has 2 particles. And if you have a third particle, you cannot solve it, and there are so many approximate ways of solving it. And we learned how to subject various problems in natural science to mathematics very effectively, and this particular area has emerged into something very, very strong in chemistry, material science, biomaterials, and so on and so forth. So if you look at drug discovery, there is a new therapeutic indication, and from there you can try to develop a drug, or there is a disease, then you can try to develop a drug for the disease, and what you have to do is whether you can measure the risk versus benefit ratio, because every drug will have a sort of a side effect and also might have some therapeutic potential. And you can try to look from the structure of a molecule, or several molecules will elicit a particular biological response. So there are different ways of discovering drugs based on a drug approach, drug-oriented approach, or a disease-oriented approach, or a structure-based approach, or a ligand-based approach. So we have come up with different protocols of looking at drugs, and essentially you have to look at the drug discovery and drug development, which are different ball games. And in the drug development, things are getting more and more complicated due to the several factors that are there. And drug discovery, also there are more and more advances that are there. So identification of new chemical entities is one, and drug repurposing is another area which has emerged very strongly. And we are also looking at the formulations. The phytopharmaceuticals or botanicals have played a very important role. And in India, we are also trying to look at the Ayush mode, where people are looking at the herbals and other drugs. So let us look at some simple things. I don't want to talk very complicated things today in the morning. We try to say that why are molecular structures important? Because people try to look at the structure determination. The reason is physical, chemical as well as the biological properties are determined by the molecular structure. This is a concept that we all know. So then we extrapolate it and then say that similar molecules are expected to have similar properties. Is that true? I like this paper which appeared 2 decades before, that you have 8 similar molecules and we are looking at some different properties like volumes and surface potentials. They have a particular pattern. Hydrophobic and polar regions, they have a particular another pattern. And you have hydrogen bonding donating potential, yet another pattern. Hydrogen bonding accepting potential, yet another pattern. Molecular electrostatic potential, we have a different pattern. So the type of similarities or differences that you look at depends on the question that you pose. So it becomes more and more complicated to subject it to mathematics. So if you look at the molecular structure representation, we know the geometric structure. But we also have to look at the electronic structure, electrostatic potential, connectivity. So you have a 1-dimensional structure, 2-dimensional, 3-dimensional, 4-dimensional, 5-dimensional, and higher dimensional structures. So there are different ways of looking at these molecules based on different parameters. Probably the computer will be very happy if you give it in a bit vector. So how do you transform the knowledge that you have into a form of a bit vector, which is very, very possible now, and lots of people are working on it. So chemoinformatics has become very important. So we all know that the SMILES format or INCHI key, for most of the students that they will be knowing it. So subjecting a complex biological information to mathematics has made a lot of inroads, and there is a problem with this information and simplicity. If you want to simplify it, the information will be lost, and if you want to get more information, you will have to go to more complex things. Even if at a fundamental level, you can see that reality can only be measured to the limit derived by Heisenberg. Heisenberg's uncertainty principle, all of you probably have heard at some point. It says that you cannot measure the position and the velocity or momentum of any particle with infinite precision. The very process of measuring will perturb the system when the system is very, very small. So we attempt to model the reality of an analog world with digital computers. Hence our models, however accurate, will only be depicting the epistemological reality but not the ontological reality. In other words, our models, however honest, will always be approximate. Having said that, the innate motivation of any models has been to mimic the objective reality as closely as it can be perceived. We all know that computational models and theoretical models have some approximations, but experimental models have higher levels of approximations. The error bar, every measurement that you have will have an error bar. There was a theoretical chemist who had taken 190 different examples where the experiments are not in agreement with the computed results. And they have challenged that these experiments are not correct because they are not in agreement. The reason is these experiments were done with a type of analytical instrumentation which is not accurate enough. So we will have to have a very good interplay between theory, experiment, computation to get the correct results. So nowadays in chemistry, when I did PhD about 3 decades before, all the chemists used to be in some isolated place and they never used to talk to experimentalists, and the experimentalists will never bother about the theoreticians. But now you will have a very good interplay between theoreticians and experiments. So is in biology. So about 3 decades back, people in pharmacy colleges, they were not doing high-level research. Most of them are having a different thing. But now, very, very high-quality research is being done in pharmacy colleges. So we have to somehow circumvent this mindset that this is a particular area and we cannot do it. We can do any area, and we have lots of data, and I'm so happy to see the morning lectures that we have a huge amount of data and analysis by the clinicians, whether it is a transcriptomics or protein structure comparison or binding energies or the binding scores or ligand similarity and data mining and network analysis. There is a lot of data that is there, and computer-aided drug discovery market appear to be very strong. I will go through it quickly because I just wanted to tell you that even in pandemic, the artificial intelligence and machine learning has played a very, very important role. Let me try to look at the field that artificial intelligence is indispensable in 21st century in any field. I will try to come back to this, but more importantly, I wanted to address a particular problem, especially in the context of India. Most of us who are doing computations, like we are looking at supercomputing, high performance computing, the capacity, capability, mathematical modeling, statistics, first principles calculations and simulations. And we also know about software, database, web portals, apps, and IoT-based device development. However, the computational models can be divided as people who develop theory or people who develop methods based on the theory, the methodologies to solve a particular problem. And then there are people who develop algorithms and programs and softwares. And these particular people can be for developing them from academic purpose or commercial purposes. And there are application scientists who try to effectively utilize these softwares, and there are users, and the users are very high in our country. And probably this theory about 30, 40 years before, we used to have a large number of people, but somehow the algorithms and program and software developers, that number is getting down. Very, very small number is there compared to the size of the country. When the users are more, if they have no idea about the methods, algorithms and theory, they are sort of becoming addicted to using them because they use it in a black box program. So I just wanted to say that some of these computational softwares are very expensive. And we try to overtly depend on the commercial software in academic training leads to some undesirable long-term challenges. The reason is lack of availability of this software for commercially deprived academic institutions. For example, a small college may not be able to afford it, but more importantly, are people who are reluctant or averse to use commercial software in principle. I may have money, but I don't want to use a commercial software because that will not allow my students to develop their knowledge. And it may be too expensive, especially in the third world countries. It may be most important, it's a black box, and often the methods and algorithms are not clear to the application scientists. This is not good. And from a very practical point of view, you may want to make an incremental change to the software, right? So when source code is not available, when you use it as a black box, it's difficult to incorporate even small incremental changes or new ideas with the existing software. So this is what we feel. When the source code and everything is available, you know what is the method and then you may be using it. But if you have a new idea, you can implement it by writing a small script. So the way forward is strengthen human resource across the country in the area of computational modeling. It is important to inculcate the culture of writing our own codes and share it across the academic community. This open source culture has to be developed. And many people, Debashish Mohanty also has developed several codes, and our group is also doing. There are Bharatam Prasad, his group has developed quite a few codes. And while there is a lot of scope in developing commercial programs, they may be restricted to profit-making institutes and not for pure academic setup. So we try to develop a code called Molecular Property Diagnostic Suite and developing this indigenous open source drug discovery software so as to de-addict the academic fraternity. So this is very easy to say but difficult to implement. And we started this idea I got in 2004, and we started working in 2011. And some of this, one paper was published after a long time, and here you can see Bharatam Prasad also is here. And this particular idea of developing a molecular property diagnostic suite, which is an indigenous software tool, which is disease specific, has been developed, and we are continuously working on it. It may take some more time because these things will take a lot of time. The molecular property diagnostic suite is being developed with the purpose of strengthening computational drug discovery in the country. We have different modules like data library, data processing, data analysis, and advanced modules. We have published that first with tuberculosis and diabetes in 2017, and the general theory was also published. This year we published a fragment library module, and these things are under preparation. I just wanted to tell you that this molecular property diagnostic suite will integrate all the data and tools that are there in the open source, and we also write several of our own codes. And with Galaxy, you can write the code in any particular language, and you can link it up. This is for the open source thing, and Galaxy is a platform which is just for the academicians. Everything is transparent, and we try to first look at the chemical space. I will go, if somebody is interested, I will be happy to discuss with them. We try to give every molecule a unique ID. We call it as an Aadhaar card for this, because we have the first time systematically classified the chemical space into different classes and clusters, and this Aadhaar card will have a class and a cluster and a number, and this particular page will be generated on the fly for a molecule, and every information about that molecule can be stored, which can be used for different purposes. To make this short, we have a series of these MPDS suites of programs for each disease, and the disease will have disease-dependent modules, where a particular disease will have its own way of tackling, or disease-independent modules, and these disease-dependent and disease-independent modules are segregated, and as you can see that 60 to 70% are disease-independent, and if you make a strong, robust MPDS, the 70% is common, and when you want to customize it to a particular disease, only 30-40% you will have to make it. So there are different challenges that are there in the drug discovery, and these challenges are different for different types of diseases, and the cost or the slow pace and new formulation of existing drugs, and there are problems with the partnerships, and probably the solution lies in crowdsourcing science, and the drug development is rapid development of novel, affordable, and effective therapeutics. I'm also trying to talk to you, so I will quickly pass through this. There is a lot of scope in AI in drug discovery and development. There is preclinical, biomarker, lead discovery. In all these areas, AI and machine learning is playing a very important role. And you can also look at the information available in multidimensional platforms and areas, mine data to quickly identify the direct target of the novel drug. There's a lot of information that is there, and then we will have to make some use of the available data. The text mining and other techniques will play an important role. Aggregating and synthesizing information, understanding disease mechanisms is a continuous process, and generating novel drug candidates will have to have a strong backing from these two things, and AI plays a very important role. And I don't have to tell you about the AlphaFold and the Rolls TTA fold. The AI accelerates the drug discovery, and multimodal AI, AI 2.0 with federal learning, gene writing, quantum computing, mapping the RNA therapeutics. It's extremely important that we will have to keep pace with these technologies that are there. And more importantly, I have seen that there are a record number of drug trials that are expected to utilize the decentralized elements. And this initiated worldwide drug trials identified as involving virtual or decentralized components where these clinical trials can be done. And these trials utilizing virtual decentralized companies projected to rise 28% from 2021. So there is a completely new paradigm that is going to come, and we will have to see that where we as a country stand in looking at it. So there is different softwares which are coming up, the discovery, preclinical, clinical, and post-approval. I just wanted to bring your notice to this particular thing. The number of drugs per billion. There used to be over close to 100 drugs we used to get per billion about 70 years before. So, 10 million per drug. So, as we come down, we can see that there is a problem. It's costing more than $1 billion. Now it is 1.5 billion, or some oncology drugs it may be over 2 billion. So, when FDA tightens regulations, post-thalidomide, you know the notorious story of thalidomide, and then the number of drugs have decreased. And FDA clears backlog following these PDUFA regulations, relaxes on HIV drug. So, in 90s, lots of HIV drugs have come. So, it all depends on now the regulation. And we also have seen that in the COVID times, we have many more number of drugs which were repurposed and also altogether coming as new. So, I will try to take another 5 minutes to see what is the understanding of health, disease, drugs, food, nutritional and nutraceutical relationship. I like this statement of Richard Feynman that the difficulty with science is often not with the new ideas, but in escaping the old ones. So, it's very important, learning is important and unlearning is also important. Can we shift the focus of research in drug discovery from disease point of view to the health point of view, and we control the function and property of proteins. So, this is very important. We all know that frailty is something that is the onset to say that you are not healthy. But these are the phenotypes of frailty: unintentional weight loss, weakness, exhaustion, slowed walking, low levels of physical activity. And if you go to the doctor, they say that they look at some parameters, they say that you are healthy. So, our understanding of health is a complete physical, mental and social well-being, and not merely the absence of disease or infirmity. So, now they are trying to change this definition by adding 2 more dimensions, that health is a physical, mental and social well-being. Social well-being we don't normally bother so much. But more than this, emotional and spiritual well-being. Spiritual means not religious and all. How enthusiastic you are, how good are your spirits. So, health is not only physical, mental and social, but emotional and spiritual well-being. And this has to be taken in an important way. So, you look at the pathogenesis of the frailty syndrome. So, we will have to consider various factors like aging, genetics, lifestyle, diseases and environment, and what are the markers which are there for, because we have been exposed in the last few lectures also, what is the frailty phenotype and what are the health outcomes. You know that these health outcomes are very important, and there are different parameters like age, physical health, mental health, environmental, nutrition and population, and we will be able to get parameters which can be subjected to mathematics. And our group is trying to work on this, and methods to assess the nutritional status is also very important, and we have to link the various diseases like lifestyle diseases, metabolic disorders, infectious diseases and rare diseases. They all seem to have some relationship, although they appear to be very, very different. So, many times unlearning appear to be more important to collate these things, and artificial intelligence and machine learning play a very, very important role in this. So, as this particular talk is on precision medicine, I dug out some old slides of mine, and this medicine, the lifestyle, genetics, and we all talk about gut microbiome, and our friend has said that the virome and many other things. So, it's very important to look at this data and lots of things. There is a lot of opportunity for people to think in different ways and in original ways. So, there are different biological processes. I am just going through these things, especially for younger people, to see that there are different opportunities, different ways of looking at it. And in our MPDS, this particular new data is being added, and we are very happy that this antiviral model which is published, and membrane permeability, toxicity prediction, we are publishing lots of small modules which are based on AI and machine learning, and we are incorporating with our MPDS. So, So we have started this advanced computing and data science thing in Nice. And this year we are working in database development or chemoinformatics and machine learning, so mathematical modeling, protein-protein interactions and polypharmacology and SARS-CoV-2. Our group was very active. And our group is, traditionally I was trained as a computational chemistry and material science guy, and we are also working in that area. So if somebody is interested, we will be very happy to look at it. Another 1 or 2 minutes, I say that Northeast is having a lot of biodiversity, geodiversity, and seismic diversity. AI and machine learning play an important role. And we conducted some summer research training program, and some of you may be knowing this. And I just wanted to say that the computation and data sciences can be used either in biotechnology or petroleum or material science, ecology and environment, floriculture and aroma mission, because these are all the areas that our institute is working, infectious diseases or software database development and modeling. I think I will try to stop. Some of you, if you are interested to look at the drug discovery hackathon lectures, all of them are available on our website, and you can look at that. We also have a genomics facility and SARS-CoV-2 facility, and this is CSIR Nice. If some of you are interested to work with us, you are welcome. And if you happen to be in Northeast or in Assam, I welcome you to my institute. Thank you.
Thank you, Dr. Shastri. It was an excellent presentation. We had an overview of, so I would like to invite students, any questions? 1 or 2 quick questions. You can discuss after the session also with Dr. Narahari.
Hello, sir. Sir, can you please shed some light on quantum computing and AI that we can use for drug discovery? As I did not clearly understand, like quantum chemistry I know, but how can we use quantum computing? Because that power I don't think so we have in our institutes.
Okay, very good question. So if you look at the quantum computing, that will increase our power to compute many-fold. So the types of chips that we have, the types of information, the density of the information depends on the type of material that you use. But quantum computing will cross that barrier and will allow you to be much more efficient. Yes. Artificial intelligence is an overall area where you know molecular modeling, yes, you have an input and you have a particular mathematical principle and you get an output. And in machine learning or artificial intelligence in a broad sense, you know the input, you know the output. You have a large number of input data and large number of output data. You know why this input is related to that output. So you give this information to a computer like a neural network or whatever it is, and you try to come up with your own theory. That is artificial intelligence. So you may not have a logic or a basis which you understand for a correlation to be established. So that's a fundamental difference between molecular modeling, which is mathematical modeling or whatever it is, that is third paradigm, and fourth paradigm is machine learning and artificial intelligence.
Sir, so can we use, is there any scope that we can use quantum computing power in foreign or something?
Yes, now there are a large number of areas that are coming, and in the future it will be better.
It will be, thank you, sir.
Any other questions? Yeah, just I had one query, Dr. Shastri, about the next decade is about RNA therapeutics. So do we have algorithms with RNA, just like small molecules with...
That's a very big area. When they got the Nobel Prize about 6-7 years before, now a lot of work is going on in RNA therapeutics, even post-COVID also. Yeah, people have to look at various approaches. and small molecules and biologics and RNA therapeutics.
Right.
And then people are now trying to look at how the biologics can be replaced by small molecules. So there are a number of opportunities to see that how we can make progress. And it's very difficult to predict in science. And definitely a lot of people are working and they are all wise people and they have a reason to work.
Okay, thank you, sir. So just, I would like to invite Dr. Murthy to come on stage, just a token of appreciation to Dr. Shastri. Dr. Murthy, please. So our next speaker.
Now the next speaker is Dr. Viras Suvarna. Viras Suvarna holds MD in Pharmacology from Grant Medical College and Sir JJ Group of Hospitals, Mumbai. He also holds a Master's in Pharmaceutical Medicine from Hibernia College. He was a medical advisor at Lethico Pharmaceuticals Limited, Indore, and head of operations at Pfizer India. Dr. Suvarna has been at various presidential positions at multinational pharma companies such as Dr. Reddy's Labs, Lupin India, Biocon Biologics. Currently, he holds medical chapter at Roche Pharma India. Okay, sir, please.
Hello. Hello. Yeah. So thank you very much, Professor Sharma and all the others here. Out here, I just wanted to specify that the last line is also important. So it's not just from a Roche perspective, but it's as a member of the OPPI, Medical and Regulatory Committee, because I believe that there was some interaction between OPPI and NIPER, and NIPER had requested OPPI to send some speakers, and I'm honored to be one of them. I'll try to do my level best in terms of justice to this topic on precision medicine and pharmacogenomics. This is the usual, disclaimer slide which is there, because these are, you'll find a lot of it as my own opinion, so it need not necessarily reflect those of the organization that I represent. Now this, I'm sure some of us who have read Tintin comics would be familiar with Thompson and Thompson, and the fact that they always talk about, to be precise. So therefore I thought I'll use this cartoon because I wanted to talk a little bit about precision medicine, but also about personalized medicine and try and differentiate the two. So precision medicine, of course, is very important, but at the same time, I'll show you one area where precision may not necessarily represent accuracy. And this is what I read in Harrison's Principles of Internal Medicine, where in measuring blood pressure, I know here I'm showing a mercury sphygmomanometer, and now actually we shouldn't be using such instruments. We should be using those clinically validated automated oscillometric instruments. But the point is that sometimes that instrument may measure your blood pressure every time, so repeatedly, or it is reproducible. Every time it comes as, say, 140/90 or whatever it is, that same number comes. So from that perspective, you might think that this instrument is precise because it's reproducible. But is it accurate? Because sometimes it may not necessarily measure your BP accurately, which means every time it is measuring your blood pressure, it is being precisely wrong. Therefore, that was one interesting aspect of this word precision, because it's more about reproducibility vis-a-vis accuracy. Now, this, what you're seeing here, is some definition of personalized medicine, and all that I wanted to say is that how I look at precision vis-a-vis personalized medicine is precision medicine is more about, for example, in cancer, it's more about the tumor. But it is the individual who has the cancer who is also important. So when you look at it from that perspective, that means every individual could be different in the way he or she experiences or expresses a particular cancer. And that same individual vis-a-vis some other individuals, both having the same type of cancer, may also respond differently to certain anti-cancer drugs. So what's important to know is not just precision from a tumor perspective. In a tumor also there is heterogeneity, but even we as individuals, we are so unique, the fingerprint, etc., that perhaps the way we experience a disease and the way we respond to treatment could also be different. And this is what our forefathers actually recognized long time ago. And that's how, if you look at the real Ayurveda, it's all about the person with the disease rather than just the disease. Whereas allopathy was more towards, the disease. So two people having, say, hypertension, both would probably get the same drug. Now, of course, we're getting into pharmacogenomics and precision, personalized medicine, etc., even in allopathy. Homeopathy also, where they basically study the personality of the individual, and then based on that, whether you're sulfur or phosphorus or whatever, then they decide the treatment. So clearly it is the individual with the disease which is also important. And specifically as far as tumor heterogeneity is concerned, this is also important, that sometimes you may take a biopsy from a particular portion of the tumor. That tissue, when analyzed, may actually be negative for some mutation, say epidermal growth factor receptor mutation, and you may label this patient as EGFR negative. But sometimes you find that the person actually responds to those receptor tyrosine kinase inhibitors, and you wonder why. And later on we realize that if you had taken a biopsy from another part of the tumor, that could actually have been EGFR positive, which means even tumors express heterogeneity. Now this all of us know, and I'm sure because of COVID-19, the pandemic, everyone seems to know everything about DNA, RNA, but all of us definitely understand that there's translation, transcription, then there's mRNA, transfer of soluble RNA, ribosomal RNA, so, and then of course proteins. So this is something which is now I think almost everyone would be knowing about it, but I thought if I were to understand what really is the genome, like from my understanding, genome basically is the genetic makeup of a cell, and it's not only in the nucleus, it's also in the mitochondrion where there is genetic material. But I came across this book and I thought this is an interesting way the author, Matt Ridley, has described what he calls the genome. So there are 23 chapters called chromosomes. Each chapter contains several thousand stories called genes. Each story is made up of paragraphs called exons, which are interrupted by advertisements called introns. Each paragraph is made up of words called codons, and each word is written in letters called bases. And when we look at how the genome is organized, so clearly it's organized at various levels, and aberrations in any of these levels can cause disease. So chromosome is of course the most condensed form of the genome. There's something called as a nucleosome, which is the DNA plus histone. And ultimately, of course, we know about the double helical structure, which is DNA. And cancer, this is just one example, is a disease of the genome, and excessive exposure to these factors increases the number of these genomic alterations. So whether it's radiation, carcinogens, viruses, and other infectious organisms, a vast majority of these damages are either repaired or do not cause cancer, but in rare instances, these alterations can lead to carcinogenesis. We know that in healthy individuals, unaltered or unmutated genes produce healthy proteins that function in signaling pathways vital for normal cellular functions. And this, of course, we know, we just discussed it. And there are all kinds of, I don't think this comes here, but all kinds of other mediators which are involved with proliferation, with survival, and all of these, of course, are important. mTOR, incidentally, there is some link to an Indian because it stands for mammalian target of rapamycin, and rapamycin, the name has come because it was isolated from an island called Rapa Nui by an Indian called Sehgal. So when genes are altered or mutated, the resulting proteins produced can have altered sequence, structure, and function. And sometimes no protein or incomplete proteins are made, resulting in dysfunction in the pathways that regulate cells in terms of growth, division, and death. And that, of course, causes cancer. And, you know, when I look at cancer, ultimately it is our own cell which typically is regulated by a system, you may call it programmed cell death, but it's when those cells escape from that regulation, they become autonomous, and then obviously they can keep proliferating and competing with our other normal cells, and thus they cause destruction and even death. But imagine if at some point, very early in the journey that cancer has made in that person's body, if we can actually identify it and try and arrest this process, you know, when our normal cell is becoming cancerous. And imagine if that cancer, you know, the cell which is now trying to become a tumor cell, this change has not yet become irreversible. If we can actually catch it at that point and then try and reverse the process, who knows, maybe a cure for cancer can actually come out, rather than, you know, typically now what we try and do is either with chemotherapy or all the different therapies, we just kill those cancer cells, because at that point there's nothing much we can do. But imagine if, you know, this can be detected much earlier, and whether that process is actually reversible. That can make a big difference to this uncontrolled proliferation. And then it's like almost that cancer cell wants to become immortal, but it obviously will die because it will result in the death of the individual which houses those cancer cells. Now, if you look at the human genome, a few hundred are known to be involved with cancer, so just 1.5%, which leads to 22 to 25,000 genes per that Cancer Genome Atlas, and it started with the Human Genome Project. And if you look at cancer, there are 4 major types of genetic alterations that drive tumor growth. So one is, of course, a base substitution. So as you can see below this, instead of ATAGC, so you have that adenosine, thymine, guanine, cytosine, and then you have an extra C coming in between. One example is something called as BRAF V600E, and there are drugs specific for that mutation, like, for example, vemurafenib, dabrafenib, and some others, cometinib. Then, of course, an insertion and deletion, and one example is this EGFR exon 19 deletion, where if you have that deletion, then if you give that patient a drug like, say, afatinib, the chances that this patient will have a long kind of period of both progression-free survival as well as overall survival are higher. So this is, you know, where you're not just diagnosing the cancer per se, you're also diagnosing a particular mutation in that cancer which will guide therapy. So sometimes these diagnostics are actually called theranostics because it guides therapy. It's like molecularly guided therapy options, or MGTO. So here, for example, afatinib, if you have a mutation, T790M, I think it's threonine, methionine, then osimertinib would be specific for that particular mutation. So it's like how you're being so precise in terms of giving the right drug to the right individual with that particular. Otherwise, it's like you're just thinking, okay, he has EGFR mutation, non-small cell lung cancer, just give him any of those receptor tyrosine kinases. But if you go deeper into it, you can select the best drug for that individual. Then, of course, copy number alteration. So HER2, for example, if there's overexpression or amplification, we all know about trastuzumab, but then pertuzumab and some others also have come out. And similarly, you can have rearrangements or gene fusion, so ALK or anaplastic lymphoma kinase. So there also, it started with crizotinib, now there's alectinib and many more. So this is the way cancer therapy, at least, definitely, when we talk of precision medicine, it's more common in oncology, but people are talking about it in diabetes as well, perhaps even in cardiology-related conditions. And in terms of the evolution of genomic sequencing, so as you know, immunohistochemistry, there's FISH, there's CISH, so fluorescence, or even chromogenic in situ hybridization, then PCR. Sanger was the first generation sequencing, and now you have next generation based hotspot testing. Then, of course, there's CGP, or comprehensive genomic profiling and whole genome sequencing. So clearly, there's a lot of advance being made in diagnostics and, as I said, theranostics. So now what's happening is you're literally able to individualize or customize treatment per, rather than say customer, we'll just say patient, because ultimately, it is the individual with the cancer who needs to be treated. And incidentally, the word patient comes from a Latin verb called PATI, which means to suffer. I know people might think in Hindi the word pati can stand for husband, so does it mean that husband suffered? No, I didn't mean that. The point I'm trying to make is, can we at least take out the suffering from that patient so that the patient can go back to being a person, an individual like he or she was before he or she was diagnosed as a case? Why can't we accept the fact that there is an individual with that? So even if you have cancer, fine, you're a person living with that cancer. It's not as if you label the person as, oh, cancer, which means that person is going to die. No. Let's treat individuals as individuals, and okay, they may be living with this or the other disorder. So ultimately, when we talk of comprehensive genome profiling, it is the tool to drive the revolution of cancer care, because it's becoming more personalized, as I said, moving towards an individualized treatment plan. So one would be where the drug fits all. Two is you identify patient groups, biomarkers, and drugs. And eventually, one patient, one tumor profile, and you have many options. So advances in cancer biology, because CGP also tells you about the biology of that cancer, and it's driving a future where cancer care truly can become personalized. So from precision to personalized medicine, one patient, one tumor profile, more options. I mean, on a broader scale, it's like, rather than all the time trying to say my drug is better than somebody else's drug, let's try and understand from a doctor's perspective, what he wants to know is my patient responds best to which drug or treatment regimen. That is more important. So let's try and help doctors identify, that patient substrate which responds best to a particular regimen, rather than all the time say my drug is better than someone else's drug. Because even if you do parallel group designs, let's understand that patients in one group have never been exposed to that other drug which was given to patients in the other group. How do you know that same patient may have responded better to that other drug? And therefore, people talk of N of 1 randomized controlled clinical trials, which are difficult to do in the real world of clinical practice, but essentially that's what doctors do in their practice. They try and identify to which treatment regimen does my patient respond the best, and accordingly they treat. So it's not easy. This is about hotspot approach. So here only sequences, select regions or hotspots of a gene are identified, while in CGP, sequences coding regions of selected genes in their entirety. So just to differentiate between the two, I will come to it in some detail in the subsequent slides. This is, of course, CGP, as we already know, comprehensive. FMI stands for Foundation Medicine Institute. It is basically a Roche company, but in Roche what happens is Roche allows these companies to function independent of Roche. So for example, in the US, there is no Roche, it's just Genentech, although Genentech is a Roche company. Similarly, FMI is a Roche company based in the US, but again, it can work for other companies as well. So this way, what happens is you have what I call competitive collaboration. You don't always have to compete with competitors, you can collaborate. You can help other companies also. They may have a certain drug. You might have the test the theranostic, which can help doctors identify if this patient has this mutation, I will give that drug. That drug need not be a Roche drug. So basically a comprehensive genomic profiling approach of testing all of the known clinically relevant cancer genes for all classes of alterations. Category 1 is the routine single marker molecular test. It's almost like your control F function, like IHC PCR FISH, and it will continue to play an important role in cancer diagnosis, but it could miss a few mutations. Category 2 is the hotspot, which we saw earlier, and that would identify, so all these are part of NGS, pre-specified mutations occurring in very limited areas of genes of interest, and they can fail to detect all classes of genomic alterations. This is the typical NGS technology, and to be very frank with you all, I'm not certainly a person who has done this, so I'm presenting it, but I wish the person who was supposed to take this lecture, Dr. Arun Kumar, if he had come, he's done a PhD in exactly this, so he would obviously be able to give you details, but I still thought rather than say that no one would present on this topic from OPPI, I thought let me at least come and be brave enough to face this audience and show you all what exactly is this NGS technology, right from the sampling, the genomic DNA, then the shotgun library from which you get fragments from the genomic DNA, you hybridize that, then wash it, pull down, capture DNA, DNA sequencing, and eventually mapping, alignment, and also looking for those copy number variants. And this is, or rather these are the steps, so right from the DNA sample preparation, you have a library, cluster, generation sequencing, reference mapping, variant calling, and a bioinformatics analysis, so that you can scientifically curate that particular report. Now, moving on from, say, oncology to another field, cardiovascular disease, actually a similar kind of thing exists even in other therapeutic or disease areas. Only thing is it ready for clinical practice? And clearly in research settings, it is already being done. It's just that can you now translate it into clinical practice? The problem, of course, here is we have so many patients. I don't know whether doctors have this kind of time, but all they really need to do is maybe send the patient sample, either a blood sample or in the case of cancer, it would even be tissue sample. And then you could express these genes and ultimately decide, okay, this person has whichever cardiovascular disorder, but can I now map from a metabolic pathway perspective or even a gene expression profile. And you can have personalized care even in cardiology. So we have all these omics, so genomics, transcriptomics, proteomics, and even metabolomics. But of course, the most important omic is also the economics, because if it's too expensive, then naturally access becomes an issue. But clearly, these are the ways you can pick out these alterations. And here, all that I'm trying to tell you again is the reason why we need to understand that it's important to treat that individual with the disease. And even Sir William Osler has said this. What you are seeing in terms of that gun, it's actually a quote from Dr. Inas Inas, an Indian who now has established this coronary artery disease in Indians in the US, where he says, while genes load the gun, environment pulls the trigger. So what he means is ultimately it's the interaction, the play, the interplay between your genes and the environment that decides who gets the disease and who may not. So that's why it's important to understand these phenotypes before treating everyone as, and that's where I keep saying that our forefathers actually knew this much before all of us. And therefore, this could be the path to personalized medicine. So one is this random selection, or second is omics-guided treatment. And ultimately, you follow them through, and the usual outcomes in terms of efficacy, safety, but one can also look at certain other outcomes in terms of economic outcomes, quality of life, etc. Now, a little bit on pharmacogenomics. We all know about warfarin. This is one example that I'm giving. And where you have these alterations in the gene that codes for certain enzymes that are involved in warfarin metabolism. So one is CYP2C9, and the second is vitamin K epoxide reductase subunit complex 1, or VKORC1. Now, because of these, I would say pharmacogenetic alterations, patients need not necessarily respond to warfarin equally. So there's inter-individual and to some extent intra-individual variability, and therefore with warfarin the problem is even if your INR, for example, if you're using warfarin to prevent stroke in patients who have atrial fibrillation, you know that the INR has to be kept in that range of 2 to 3. But in 2/3 of cases, even if that patient is in that range, in 66% of cases, the person can have a bleed. Why? Because the labs which measure INR, there also there's a lot of variability. They don't use that WHO reference standard of prothrombin. It can lead to variation. For example, you may ask a lab to give you the INR report. It may come below 2, say 1.8. So naturally, you'll want to increase the dose of warfarin based on that report. And then you realize the patient has had a bleed. Why? Because now when you check the INR at a different lab, which is probably better, it was actually 3 point something. Why? Because that 1.8 was not 1.8, it was some 2.6. So there's that coefficient of variation, which, because from lab to lab there is so much of variation, the clinician is actually, it's difficult for the clinician to decide. Similarly with clopidogrel, you also know CYP2C19, so the cytochrome P450 isoforms. Here again, this can lead to a difference in the way you would respond to clopidogrel, and whether this is now clinically significant, that is something that we need to, because sometimes clinicians tell us, I don't think this makes much of a difference. I've used clopidogrel over so many years and so many patients. I don't think all this is important. It's only in a research or academic setting. And similarly, with statin-induced myopathy, I mean, people have used Auditorium and other statins in so many patients. Not everybody gets that myopathy, the rhabdomyolysis and myoglobin precipitating out in the renal tubules. But is there a way of predicting it by doing these tests? But then if the tests only are going to be expensive, then again, doctors would hesitate before using tests and then deciding treatment. But this is my second last slide. So the last slide is, all that I'm saying is, just as you have 4 Ps in marketing, in medicine also you can think of these 4 Ps which are very important. So one is in terms of predictive, then of course preventive or preemptive. Some people also mention personalized or precision or pharmacogenomics. And there's one more P actually, which is not there on the slide, and that is pharmionics, which is the scientific study of drug concordance. Means for how long is the patient taking the drug? Is he compliant or adherent to what the doctor has recommended? Because that also makes a huge difference to the outcome for that patient. So that's all that I wanted to leave you with. Please feel free to ask questions, and I would actually learn from you all rather than the other way around. Thank you so much.
Thank you, Dr. Suvarna. Quick questions, if any one of you want to have. Dr. Sharma, yeah.
Quite common in many patients. So those kind of variations actually, like what kind of Indian data actually shows about variation, like few patients are getting, and some of the patient even 80 mg of atorvastatin is given, actually like they are not getting, but few people are like getting certainly this myopathy actually. So is kind of a data is available in Indian population actually?
Sir, the problem in our country is that even if that data is available, it would be in pockets, in small numbers, so I'm not sure it is generalizable. But clearly there is data to suggest that some individuals experience the myopathy. With some statins, this association is higher, especially if you combine it with, for example, gemfibrozil, because that inhibits the metabolism of the statin. Fenofibrate is considered to be less likely to cause that interaction. So that's why, in fact, that's the reason why doctors believe that it's okay, I've used this drug in so many patients and very few have experienced this myopathy. And that's how here the medicine is more in terms of reactive rather than being proactive. Like, there's a common understanding among doctors that you have, for example, you mentioned 80 mg. Of course, 80 mg of atorvastatin, for example, would be for those patients who have had an acute coronary event, and now, post PCI, naturally 80 mg initially, and then you can reduce. But in general, people believe that, when you test these drugs on the Westerners, the Caucasians, you require higher doses. When you come to Indians, you require lower doses. Now, that is more, I think, of a clinical judgment. But if you ask me from a genomic perspective, is there data? So from the Human Genome Project, people have actually seen that the differences in the way people respond to drugs between continents or between countries is only 7 to 8%. But within a country, the variation could be as high as 80%. But unless we have more data to prove that, and especially from India, clinicians will continue believing that Indians always will require, like 10 mg ramipril was used in, say, the HOPE study. Here, doctors will start with 1.25 mg, then go to 2.5, and then, if at all, to 10. Yes, sir.
Yeah, with permission of the chair, Professor.
Sure, go ahead.
So basically, I am trying to understand the personalized medicine, how organoid can extend that, the way for personalized medicine as a tool for the personalized medicine. This is one of the components.
What is the first word you used?
Organoid.
Organoid. Okay.
Spheroid, organoid, mainly I am thinking about organoid sample, how it would be a useful measure for personalized medicine.
I would not be... First, let me try and understand, when you said organoid, you meant?
Organoid with a specific chronic problem, the clinical sample, it is tested with, used from the pluripotent stem cell, from the blood sample, pluripotent stem cell, organoid can be developed. This organoid shall be used for several testing of the drug, sensitivity, resistance, and the kinetic component, all this together, it comes into that, the drug personalized, whether it works or it doesn't work.
No, it's a good thought, sir, and I think maybe there is already some research going on into whether at a cell or tissue culture perspective, whether you can test in vitro or ex vivo and then be able to, translate that into human beings, because obviously we can't, as you know, clone human beings only for testing. So maybe at an organoid level, it's a bit more ethical to do so.
Yeah. This is one of the aspects. Another question, again with the chair permission on this, how the pharmacy practice qualified people would be useful in that intermittent with the personalized medicine in between that the clinician and then other clinical service. Pharmacy practice nowadays it's emerging. How their service can be utilized, whether you are using that pharmacy practice qualified people service for this purpose?
So, sir, in the US, as you know, it's not called PharmD, it's called DPharm, and those individuals, they actually go through prescriptions of clinicians, and they have the power to actually tell the clinician that they are, maybe they've made a mistake here because they've not looked at, of course, that is at the level of drug-drug interactions, etc. But what you're suggesting, yes, if a pharmacy graduate gets into, precision medicine, pharmacogenomics, and if it is kind of mandated that we need to do that kind of, investigation and get into not just diagnosis but theranosis, then yes, that will feed back into, means when you do, for example, comprehensive genome profiling at the Foundation Medicine Institute, the FMI report will tell the clinician not only about the mutation, but also based on that mutation, which kind of drug is most likely. It's like, for example, sir, when we have infection, so we isolate certain bacteria from the patient's sample and test to various antibiotics. Now here it's almost like you take the tumor cell, maybe from a biopsy, and then test it to, first you detect the mutation, and then test it to different anti-cancer compounds, and select the one to which that tumor cell is most responsive. So something like that is actually happening here.
Thank you.
Yeah, Dr. Suvarna, just quick question from my side. You talked about economics. How is it becoming affordable, the Roche company, for the precision medicine? For example, we've been knowing that warfarin, statin, Selective. Are these metabolic chips available for the normal individuals to be, and what is the cost? Is this affordable medicine or not?
So, sir, with Roche, as you know, we are not really into cardio or even diabetes. It's mainly into cancer. So there, what I have experienced is that these medical oncologists, when they treat patients who have different kinds of cancers with all these mutations, now there's a big push towards making doctors understand that rather than just decide on a particular drug, please send for these tests. I don't know the exact cost of FMI. I have joined Roche very recently, but we are definitely, Roche is all about access, so which means we try our level best to make a particular test also as affordable as possible, because naturally if the test itself is going to be too expensive, then how will the patient afford when he can't even afford the drug? Because sometimes the drug also is extremely expensive, and therefore we have patient support programs. And similarly with FMI, and as I said, FMI is not only for the Roche product, it could be for any product, but at least you do the test, find out the mutation, decide the drug. It doesn't have to be a Roche product.
Okay, thank you very much. May I request Dr. Ravichandran to give a token of appreciation from NIPER? Please, yeah.
Sorry.
Moving on to the third speaker. Now, I would like to welcome the third speaker of this session, Dr. Debashish Mohanty, PhD from Indian Institute of Science, Bengaluru. He is a postdoctorate from Hebrew University, Jerusalem, Israel, and the Scripps Research Institute, La Jolla, USA. The main focus of his research is to understand the structural principles that govern the sequence to structure relationships in proteins and ligands. He is awarded with National Bioscience Career Development, Rajiv Goyal Youngest Scientist Prize Award in Life Sciences, and many more prestigious awards. Please, sir.
Good afternoon, and thank you for the kind words of introduction, and let me thank Professor Sharma and Professor Panda for giving me the opportunity to come and talk here. But I am aware that we are running late, and I am standing between you people and lunch. What I will try to do is I will give you a quick overview, and maybe those of you who are interested, we can discuss during lunch or afterwards. So what I am going to talk is some of the computational methods which we are developing to address few questions relating to microbiome and things like that. As you heard in the previous talk, for precision medicine, you require genome sequencing or things like that. But now, microbiome is attributed to almost many these things. So now it is not only our genes, it is the genes which make up the entire microbiome, so that goes to the millions. So now, since there are so many different things, microbiome has been implicated. So now you are getting the complete genome sequences from the microbiome, how we can use it to understand the things and design better experiments. So the way microbiome affects many different things, but the language through the language of small molecules are the chemistry, and these are the three ways it can synthesize certain molecule, biosynthesize de novo, it can modify a host-derived metabolite, and it can also do a xenobiotic derived, either a molecule, small molecule which comes from the diet or the drug, it can modify them. And there are experiments coming to address these questions, but with huge volume of data, what sort of computational tool we can develop to use this whole genome of the microbiome and address these questions better. So these are the tools which initially have been developed for the genome-guided natural product discovery. Like natural product discovery was initially a domain of chemists, microbiologists, but with genome sequences came, so there are bioinformatics developed, and same tools can be used to answer this question which I am proposing, and all this involved dealing with sequence, structure, function, on which Dr. Shastri gave a very good overview. And the type of molecules we are looking at is polyketides, non-ribosomal peptides, and a class of molecules which are called bacteriocins or antibiotics, but now their diversity is so much, they are being like, it is a molecule like nisin. It is a peptide which is made in the ribosome and post-translationally modified. And these are being called RIPs, ribosomally synthesized post-translationally modified peptides. So now, and coming to this, then this is the area which is also equally important, like this is the example from digoxin. So a microbial gene which is coming and modifying it so the drug becomes inactive. So as more and more examples coming from experiment, as computational biologists, if you analyze that data properly, we would be in a position to develop the tool which we can use better to predict these things, and that is the purpose of this talk. And this is just a quick overview how, I mean, it looks really challenging that how can you analyze genome, the genomic sequences and predict chemical structures of the small molecule. But if you take the analogy, if you get enough examples of protein sequences which go to certain 3D structures, you see the commonality or difference between them, you are able to predict structure, and machine learning is helping there. And same thing can happen here. Like what is shown here, these are different natural products. It may be coming from a bacterial genome or microbiome. If you look at their biosynthetic genes, there is a good degree of commonality, and only where there are differences which are shown in the red or the yellow color, those parts of the molecules are changing. So if you keep track, more and more experiments coming and this track, so you can derive pattern and you will be able to predict that. And this is where, like, the conventional ideas of bioinformatics, biophysics was helping earlier, and now machine learning is going to help further. And this is where, like, this is area we have worked for many different years. There are many programs here, and some of these programs are developed like SBSP kits to look at polyketides, non-ribosomal peptides had been developed in our group. A challenge was, if you analyze the genome sequence, predict the catalytic domain structure, what you will be able to predict is probably the linear structure of the metabolite, but it can cyclize in many different ways. So now a chemist will be able to look at it and tell this is the correct cyclization, others are wrong. But when you are dealing with big data, and this is where the machine learning is helping. If you give examples what is correct, what is incorrect, machine is able to learn the patterns. And now if you give a genome sequence here like this one, and instead of telling which genes, this is a completely automated prediction of metabolites which are encoded by the biosynthetic gene clusters. I will skip the details, like all the chemical fingerprints and things like that has gone into it. In the interest of time, I will skip all these, and these has to be validated in different trust validation and things like that. And this is we have published in earlier from our group. And there is another class of molecules which I gave you the examples. These are called RIPs. These are ribosomally synthesized post-translationally modified peptides. So what is happening here is these are small peptides. They do not come from the non-ribosomal pathway. They come from the ribosome. But there are modifying enzymes which are putting crosslinks, and many different crosslinks are coming. So to do that, I will skip the pathway. Like what is happening is like nisin. This is an example which is a food preservative which is used, and so this is the peptide. This has only many cysteine and serine, and using the enzymes, the serines or threonines are get converted to DHA or a DHB, and another cyclase joins the puts the crosslinks, and you get this complex chemical structure. So if you look at it, theoretically you can assume that any cysteine will join to any serine or threonine. So you can theoretically say these are the crosslink patterns, but there are, if you go to the examples which are characterized, you will get the correct crosslink pattern. Then it becomes easy to train a machine learning model which can train which are the crosslinks which are occurring in nature, because there are certain types of structures are preferred. So that is where you will be able to predict the chemical structure of these crosslink peptides. And now, if you do that, and this is what we have done in our group, and now these are the open source software which Dr. Shastri was referring to. So now, if you do that, then of course now we are in a position to go to the genome. I will skip this, and to the microbiome and try to answer questions. What sort of molecules which can predict in the microbiome. So this is what happened when we developed these tools. This is the paper which came in Nature. So this is a molecule which was effective against VRE, and this was coming from a RIP pathway. It was a nisin-like molecule. So obviously this was published, but we took our software, tested it on the genome, and we could see that we are almost able to predict the same compound which they had found through experiment. And this encouraged us to look into the whole microbiome, and there are different types of microbiome data. Some are isolates, these are complete genomes, or some will be MAGs, metagenome assembled genomes. So using these, you will be able to see what sort of natural product or the RIP repository it is making. So there are different types of RIPs are listed in this, the lanthipeptides, head-to-tail peptides, lysopeptides, glycosin, botryomycin, cyanobactin, and which types of bacteria are making which types of molecules, so you will be able to predict in an automated way. But the question is, just prediction itself is not enough. If I want somebody to do experiment, I have to tell what sort of function it has, at least even in a prediction way, then probably somebody can do experiments. And of course, as anybody will do when you have used data, first you do cluster them and see how many different clusters. Now you take all the known RIPs for which we know the function and cluster them. So what you find is the ones which cluster with known RIPs, at least you get a functional clue. And this is the type of analysis we have done, and for many groups you will be, but the percentage is not, probably about for 20% of them you will be able to predict some sort of a function because they are making some sort of molecules for which we already know and for which we know the function. And this is the reason for this. Then the question here is, like another thing, the ones which we do not cluster, how do you think these molecules might be really made or they are artifact? But if you look at the sequences, these are very small peptides, but if you see the conservation is very high. That is what is shown in this one. Whereas corresponding biosynthetic genes or the proteins are very diverse. So despite divergence of the biosynthetic enzymes, if this short peptides is having such a high degree of conservation, there must be some basis. Probably it is linked to, it has to maintain some sort of a cross-linked structure and some sort of a function. So with that idea, so our tool RIPMiner genome, it can run through the whole microbiome and it will be able to predict these types of molecules, and wherever it maps to a known molecule, it will be able to predict the function. And same thing we have done for lysopeptide or other classes where things have been done. So this is a summary of all the like microbiome. These are different bacteria on this axis. These are the different class of RIPs, lanthipeptide, lysopeptide, thiopeptide, and wherever you have a black color dot, that means we don't know the function, or if there is other color things, that means you can associate some function like pore formation, inhibits food spoilage or cytolysis, whatever function is in literature. So this gives a way to take the whole microbiome genomic data and run these steps of a computational analysis and assign some sort of a function to some of these. So this is next thing I would like to give just few quick slides on. Can we address this sort of a question, like where a microbiome derived enzyme is coming and modifying a drug? So obviously we have to be guided by experiment. Some of the experiments came from literature, like people took different bacterial strains, tested it on different drugs. They found that 95 are not metabolized, there are this many are metabolized. And there are many interesting experiments that took from a microbiome, this particular bacteria, gene fragment, cloned them into E. coli and tried testing which drugs are modified and which drugs are not modified. So with this data, today there is about 25 to 30 enzymes which are known, which are coming from different microbes, and they are introducing modification on different drugs. So that using this data, so now you are in a position to, these we call XME or genobiotic modification enzymes. So they can potentially modify it, and the type of modification, it could be a nitroreduction, hydrolysis, this is known, like even your cytochrome 3 metabolism, how drugs are modified, these are host enzymes are modifying. Only difference here is these are coming from a microbiome derived enzyme and these are the modification it is doing. So now using this database, so if you scan through like different bacteria, so what you can assign is which class of microbes are hosting these modification enzymes and what they can do to different types of drugs. And this is the static point, and as more and more drugs, like example will this, you can this, but at this point, it is a straight cut matching, which is standard bioinformatics tool, this one. But as we have more data, we will be in a position to use machine learning as I used for the example of RIP. So you do not have to go with just the assumption that the same drug is being modified, because the modification is happening on a functional group. So you will be able to predict what sort of modification, which types of enzymes are doing, and even if this a particular drug which is not covered in the experiment, you might be able to still predict if the volume of data is large, and with machine learning, what sort of modification a microbiome derived enzyme will be able to make. So this is the point I wanted to make, and as Dr. Shastri was telling that we all try to prefer have these as open source software. So all these are in the open source. This is not published, but this is microbiome MedDB. So we have a database of this, and somebody gives a genomic sequence, a microbiome derived sequence. It will be analyzed, and either the modification or the enzymes, the de novo biosynthetic, whatever it is making, will be predicted. So this is the summary of my talk. So what I tried to essentially cover is evolutionary analysis of known natural product biosynthetic pathways in combination with protein structure modeling can help predictive rules for correlating sequence of secondary metabolite gene cluster to chemical structure of their biosynthetic product. Combination of bioinformatics with computational chemistry and machine learning approaches can help in discovery of new pathways, and analysis of RIPs in microbiome using RIP minor genome, this is the tool we have made, helps highly conserved chemical scaffolds, and functions can be predicted for some of them. And using information about genobiotic metabolic enzyme, we have predicted genobiotic metabolic capacity of the human microbiome. So I would stop at this point and take few questions, and obviously those who are interested, we can have discussion over lunch.
Thank you, Dr. Mohanty. It's open for discussion. Any quick questions? Or we can have...
Sure.
In that way, the disulfide linkage it will form. It will depend on that.
No, that under the physiological conditions, it will maintain the disulfide bond. If we change it beyond that, the disulfide bond will be reduced. Yeah. No, these compounds are known to be stable. This sort of things, they don't get reduced by small change in the pH or things like that. And this is how they are doing their function. And these compounds have all originated from when a bacteria is surviving the natural niche. It is the chemical warfare agent of the bacteria. It uses it to kill other bacteria so that it can use the resources better. They are fairly stable compounds. As you know, what you said is right. Some cases, these things can change with environment, but there the design is different, or in a laboratory condition, we change it, go from a non-physiological range, then we can reduce the disulfide bond or make changes to the structure.
Okay, thank you, Dr. Mohanty. So I would request Dr. Shashibala to come on stage to give a token of appreciation to Dr. Mohanty, please.
Please come, please come. आइए, इधर आइए। Nowadays it is getting
I also request. Please stay. Thank you very much for all the speakers for their wonderful presentations. And it is a time to honor our chairpersons. I request Dr. Sasibala Singh to honor our chairpersons, Dr. Tikku and Dr. Abhi Sangambar. Okay, thank you very much for all. So we break for lunch. So we will reassemble by 1:30 sharp for starting the session, next session. So yes, we have valedictory functions afterwards, and we will be starting in time. Thank you.
The behavioral science of a consumer. Now, coming into our research centers all over the world, I mean, we have our headquarters in the US, Ada, Michigan. That's where most of the research happened on the cosmetics and home care products. Then our headquarters in the Yuna Park, which is basically a Neutralite's headquarters, and that's where we do our research on all of our formulations related to health and wellness. Here also in and around US, we have our own farm, in Quincy, in Washington, and outside US we have our the organic farms in China, and that's where the UC China is our agri research center. Our cosmetic research basically is in the Korea, South Korea, and of course headquarters in India is in the Gurgaon. We have our manufacturing plants down south in Madurai, Tamil Nadu, and that's where we have also last year we have opened 4 new research labs in India. So in Gurgaon we have a research lab for our analytical sciences, method developments, for the formulation development of solid oral dosage forms for the nutrition. And in Bangalore we have our botanical science and egg research laboratory. In Chennai it's a food research lab, and in Madurai where is our manufacturing plant is there, where we have a pilot plant. So if you look at that, we have more than 75 scientific research laboratories worldwide in a different stream. So with that, I'll come back to the thing which I wanted to share with the the students and is that if you look at the wellness business or the size of the market, it's like about vitamin and wet management segment is about $130 billion, which is growing with the 3% CAGR. If you look at the wellness and nutrition specifically, it's like even huge. So if you look at the size of the wellness economy itself, which certainly have a lot of other segments and into it, it's a big kind of a market and opportunities. Now we all had pandemic situations, and all over the world. It wasn't like something which probably the first time which came as a which does not have a geographical barrier, like all over the world everyone has gone through that pandemic. And what we kind of identified that there are the emerging accelerators which were identified during the COVID phase, that how does the consumer become more aware and what are the basic needs which has arrived. So if you look at that, the clean label comes up one of the first newer trend, like the consumer looking for the more clean label. And when we talk about the clean label, it is more of like the product should be free from any preservatives, natural identical colors, or any of these synthetic ingredients into it. So all of a sudden the awareness of the consumers has increased during the COVID phase. Of course, transparency and traceability, so that the consumer wanted to know whatever they're consuming is coming from where, and what kind of the transparency the companies are keeping up or the product have, and the traceability which the product comes from. And that's where the what we are practicing from the years long before these needs started coming up comes in picture, where we say that we control the quality from the seed to supplement. Another thing which has come up is the kind of sugar movement. All of a sudden, like the sugar become like less or no sugar products requirements has increased in the market. Food level literacy, of course, as I said that the consumers have now kind of very cautious about before what they are taking as a health supplements. They wanted to know about the product and about the ingredients which they are taking up. And sustainability. So environmental sustainability is another thing which is consumer is wanted to contribute and ready to pay. That something, the brand or product which comes from more sustainable source is appreciated. Plant-based movement, I think the vegan is something which is a global phenomena now. We as a country is a kind of the milk and milk product consuming country, but even in India we have seen that this kind of movement is increased where people are moving away from any source of ingredient which is coming from an animal source. And you would be surprised that Amway practiced this plant-based movement for a long time. One of our biggest brand on the protein is called APP, all plant protein. So which means we don't use any of the ingredients which are from milk or milk-related products. Fortifications and increasing the role of science is the thing. So if we look at one of the quote when I was kind of thinking that what we should start with also is the Greek physicians, Hippocrates, who says that let the food be your medicine. So while here from last 2-3 days, and of course I myself come from a pharma background, and we do a lot of research and development and to make people life better and consumers patients life efficacious. What the philosophy is coming from long back is that can we have a food which we are taking and which can help us to have a better life. Now the philosophy is behind this focuses to around the preventions. So one of the conference where I was talking about bridging the gap between disease management, and my view was that the nutraceutical supplements and these healthcare wellness companies or the product formulations comes even before you get into a disease stage. So bridging the gap is start once you get into a disease, and then you start managing it and maintaining it. But we operate at the level where how do you actually can get prevented and not get into that stage. Of course, the nutraceutical terms which was coined long back in 1899, which was combination of nutrition and pharmaceuticals, because nevertheless, while the nutrition and the food are kind of prime focus, nobody can ignore the technological advances which has come into a pharmaceuticals. And it is a kind of I would say the need and of a nutrition and wellness companies like us also, which we follow a lot of, that how do we can combine these technologies which we have evolved over a period of time from the pharmaceuticals and comprise them into a nutrition to make it more efficacious and better and kind of more compliant. So yeah, it's like nutrition is around the health, the pharmaceuticals is around curing and making a remedy solutions. The pharmaceuticals is typically more around the prevention approach. Now, while it's not that easy, kind of a subject in terms of regulations, because even if you try to find out the regulatory definitions of nutraceuticals, it would be hard to find. And what I'm trying to show here is also that if you look at the world around, the regulations, it's very different. Different controlled by the different regulations and the organizations. While there are a lot of standardizations happened in the pharmaceuticals, all over the globe, in the nutraceutical and food, this is still not there. So we are continuously kind of working with the different stakeholders in the governments, in the regulatory framework, where we want to harmonize the method and processes and the regulations. So as the ring bell is on, I'll just see that. So what are the key drivers in nutraceuticals? One of the key drivers in nutraceuticals are like awareness. All of us heard and all of us become more aware about, and I think, I think COVID has one of the key, the motivator, or where people are now more consciously aware, and you can talk about immunity to anyone in the country. I mean, you talk to any layman and talk about immunity and kind of things, he will have a lot of to contribute and talk about it. So all of a sudden the awareness has increased. Like people wanted to more aware about their health and prevention kind of things. Increasing in the lifestyle diseases like diabetes, hypertension. We are talking about like from last 2 days. They are still two of the major the disease which is there. Food quality, I mean, there is certainly a level of, the deteriorations, I should say, or the kind of shift in the quality of food which we used to eat, like, the lot of Western diet which we have adopted, and whether that is suited for our environmental or the body type is still a question. So that's there. The affordability has increased, certainly that people are being more affordable. There are, the disposable income has increased, so they can afford more kind of health supplements kind of stuff. And of course the natural trends. So these are the key kind of growth drivers for this. So what are the different opportunities if you look at that, from that? So certainly innovating a delivery formats, and as I was talking about that in pharmaceuticals, we have made a long way on the developing technology platform like, say self-emulsifying drug delivery systems, right? The sustained release formulations, delayed release, combination of the targeted release formulations. Now, can some of this technology be used into an health supplement to make the product more efficacious for the consumer? And we do have a kind of a, some of the research going on especially on the SEDs to work on the fish oil, to improve the absorption of fish oil, DHA and EPA, to improve the cardiovascular health, or in general the cholesterol health. So that kind of technology's adoption is kind of an opportunity. The opportunity is on the sustained release, like one of the products which we launched during COVID was on sustained release vitamin C. Like while we have seen that a lot of vitamin C high dose formulations popped up into a market and people were consuming them left, right and center, but the research suggests that beyond 400 mg if you consume a vitamin C, it necessarily doesn't get absorbed into a body. While it's a water-soluble vitamin, but still the receptors get saturated. So it is a need that if anything which is beyond 400 mg is going, it has to slow release and should be released over a period of time. So that kind of technology is an opportunity and things which we are adopting and building up into our nutraceuticals. So I mean, I will quickly go through like some of technological innovations, synthetic biology, Ayurvedic legacy. Of course, I mean, we are a country of huge, science-based Ayurveda, which was there, and we are adopting a lot of Ayurvedic concepts to bring them to a science, and things like when we develop a product around the tulsi, we ensure that we are able to identify the marker compound uracilic acid up to an end product in the formulations. So not only into the extract, but also up to an end product. Similarly, like when we worked on the ashwagandha, then methaloids, content of methaloids into it was one of the key research which we worked internally in India and developed. So product development strategies, investment in verifying the health claims, and investment in the research to do more of the behavioral and also clinical stuff. So we are doing a lot of initiated now a lot of clinical studies onto some of our formulations, where we're going to be doing double blind the clinical studies to evaluate and measure the effect of the formulations on the overall health. So what continue to remain a challenge for health supplements and nutraceuticals is, of course, the changing regulatory climate. I mean, it's good, it's for good, but as we are seeing that it's evolving. It's not that, as I was talking about, it's not that standardized, or, all over the world, but also within India also, we are seeing that, we are slowly and steadily evolving and bringing new regulations. So these all changes are kind of become challenging because you need to redo a lot of, formulation and understand them. Of course, cultural customization is another need, and you see if we see it in India is like you can relate it like in Europe. I mean, because the culturally we are so different. Every 100 kilometers, our food habits, our cultures, traditions, everything change. So for us, when we fall into the formulations which are fall in India, which fall under the food, we need to be very sensitive of cultural customization, and we need to customize a lot of formulations related to the acceptability. Of course, cost and claimifications. Sensory organoleptic, as we are moving more now from the, as you must have seen outside, we have our stall where we have put up our innovative products. You will see we are now also moving from our tablet capsules formats to a gummy and more of a food format like bar and drinks and kind of things. And that required a lot of sensorial and organoleptics, to be adopted for end consumer. And of course, the pharmaceutical versus nutraceutical is always going to be a kind of a gray area and continue to be, evolving as we move around the regulatory. The key trends, you see Ayurveda for immunity is one of the key trends which is now coming up. The demand for clean label nutrition, holistic nutrition for a better health and wellness, clean nutrition for mental wellness, rising consumer drive for convenience, and the rise of the plant-based meat and milk. So these are the rising trends in the nutrition and wellness industry right now. I mean, I won't go into detail of it, but just to mention the mental wellness. Mental wellness is all of a sudden has gotten very attention post specifically the COVID, and you will see that there are a lot of products and demands and rises. Even in the pharmaceuticals, there are a lot of developments happening to improve the mental well-being.
I think the time.
So this is the last slide, and at Amway when we work on the research and develop a formulations, we keep that human-centric design thinking research framework, which is more of understanding the consumer base, exploring and editing the prototypes, and then implementing the product and materializing it. So with that, thank you so much, and thanks for the opportunity.
Thank you so much, and I think we are running out of time. We don't have much time for question, but I'll just, one comment or question which I can ask is, you mentioned that there are several manufacturing hubs in India. So those manufacturing hubs are meant for distribution of product in India or abroad also?
So manufacturing hub means about Amway, you mean? Yeah, so Amway,
yes,
we have a manufacturing facility in Madurai, and where we are manufacturing almost our 99% of product which we distribute in India is manufactured in India at that site, and from there also we are exporting. So we are exporting to Indonesia, Thailand, Philippines, and some of the European markets are under consideration.
Yeah, so how much of the percentage revenue export or distribution within India can?
So in India we don't account that revenue because that's only a manufacturing which we does. So the revenue actually falls under that. But if you talk about the manufacturing capacity, I think we use about 20% of 15 to 20% of capacity to do that.
Yeah. This is just for the benefit of students, that
sure.
In case they. And second is for technical, there is a lot of purification steps, analytical steps involved in nutraceuticals. So most of the analytical groundwork is done in United States and technology brought here, or from India the technology goes outside.
So thank you for asking this question. I think this is So, you know, I mean, if you ask in India, we were like, you know, when we are like 25-year-old in India. So most of technology we were adopting from the US and tailor-making it to meet the Indian regulations. But as I said, that we have now the research labs investment done in here. Now we are actually developing a product which are going actually for an outside India. So recently we have developed a product which is going to US for manufacturing. So we developed a product here, they will manufacture and actually the product is developed for a Brazil market. And similarly we are developing a product for Indonesia market here, and then the Indonesia will produce that.
Congratulations. Thank you so much.
Thank you so much.
Thank you very much, Mr. Arjun, for that interesting perspective on nutraceuticals. So we are delighted to now welcome our next speaker, Dr. Kamla Rai. Dr. Kamla Rai is currently Vice President, Senior Global Program Head, Global Health Development Unit at Novartis in Hyderabad. She did her MD from All India Institute of Medical Sciences and had previously worked with Merck as Research Director. She has been awarded with numerous awards like DST FICCI Award for Excellence, Amsonian Ambassador Award, and Global Diversity and Inclusion Award by Global HR Board of Novartis. So with this introduction, I invite Dr. Kamla Rai to deliver her talk on drug development, emerging trends and perspectives. Over to you, madam.
Thank you, chairpersons. Am I audible at the back? Awesome. First, I would like to thank the organizers, but above all, my special friend, Shyam, for inviting me, because ideally, this year end, and I think Arjun knows it, we from the corporate world, it's closing for us, and we have a lot of activities lined up at the end. But I think it was for Shyam's insistence, and I thank you so much for giving me the opportunity, because I wouldn't have it any other way. This Saturday, I would rather spend with all of you. So quickly, in fact, I think, Arjun, you made my life very simple, because the context which you said has really, you stole some of my thoughts. In fact, some of your key takeaways from your slides, especially when it comes to the growth of nutraceuticals. I am a consumer of Amway products, and I think one of the best products is the home product which takes away stains. It's the very kind of the robust stains which don't go away. Amway has some nutraceuticals. I consume those as well. So I think, thank you for that. Thank you for also highlighting the trends and above all the design thinking. Maybe by the time I can, it's not in my presentation. Ideally, I've stopped preparing slides, sir, I've begun to believe there's something called death by PowerPoint meetings, right? So I've stopped making slides, and I kind of wanted to stretch the envelope, and I said, Shyam, can I just do a talk without slides? And Shyam said, nahi, kuch to kar lo, 3-4 slides hi bana dena. So yesterday night, I came back because we had a global visit from, so I was a little busy, and I had no slides. I said, okay, I had my thoughts. I said, okay, I'll just put together in my slides. So 10:30 is what I put together, and I had a pleasure because I think we have so many perspectives, right? I spent 22 years in the industry, and I know Viraj as well. We've been in this journey together, and we have all these perspectives, so I think it was easy for me to just but I think the important thing that I want to kind of highlight is, I'm not here to do a data dump or I've not gathered information for the information's sake. I think the important thing is I just want to unleash curiosity. If I have done that, I think my job is done over here. It's not an information overload. I hope it is not. I really want just to stress on 2 or 3 key takeaways, and I kind of take a cue from you because I've taken 2 or 3 key takeaways from your presentation. So I don't know where my slides disappear, baby. Sorry? To give away my password. Okay, so while we are warming up to this, so I think just to take a cue from, we'll do a little bit. Are you game for a interaction? Yeah. Okay, I'm going to ask a question, and the chairperson will give you an award. Okay. So the question is, Arjun talked about the growth of nutraceuticals, wellness. So please, anyone who's been paying attention, what was the size of nutraceutical in the wellness space? You raise your hand, fastest finger first. You're not allowed, you're the front. You're the most like, no. Okay, sorry, I didn't mean to be rude. Sorry, how much was it, sir? No, you're the only one. So big round of applause to him. He gets it right, it's 4.5 trillion. And I think it's amazing, isn't it? Because the healthcare industry that we all represent, you know how big our market is? Anybody? If wellness is 4.5 trillion, you said? Trillion? And you know how much Indian economy is? 2.5, 2.6 trillion. So the wellness industry is bigger than the Indian economy. So how much do you think is healthcare? Students of NIPER, you're the smartest from India. You need to get this right. You're working for this industry. Anybody? It's 1.3 trillion. 1.3 trillion, 1.4. It's going to be, by 2027, it's going to be 1.7 trillion. So I think it's huge. And out of which, you know how much is the pharmaceutical industry? I'm so sorry, apologies for this. It's the controls in my company. I'm not allowed to give a USB, and apologies for that. That's why I couldn't download it. Yeah, so I think the industry is huge, and 10% of the healthcare industry is pharmaceuticals typically. So I think I'm very happy that all of us continue to grow this industry, because after oil and gas and food, I think healthcare is the largest employer. And according to WHO, about 65 million is the current workforce of all of us put together. We are about 65 million that contribute to 1.7 trillion, and we are 15 million short. So that's a huge demand, and I think Arjun pointed out about some of the automation, and maybe that would really help address the gap that we have in workforce. So, okay, can we go back to one slide, please? Go back. Oh, sorry, I should be doing it? Okay, so disclaimer, these are my views, not my company views. Yeah, I think you heard Arjun say about mental health and how nutraceuticals is playing away. What I wanted to do over here is, there are two things that are impacting healthcare. While it is a large industry, it's about 1.7 trillion, but if you look at the statistics, it's quite alarming when it comes to the global burden of disease. And the issues that we have, the current model is the patient goes to the hospital, right? But I think that's where the problem is. The problem is it's a problem of time and space. And mental health and well-being, health equity, transformative medicine. You heard how Arjun described the transformative, the synthetic biology and all of that. Maybe if you have the time, we'll go through that. We need to reimagine global health in its entirety, the preventive aspects of health, not just test and treat, but prevent and cure. Digital transformation. Of course, we'll explore some of these perspectives. And I loved when Arjun stressed sustainability. It's a big thing nowadays, right? So environmental, social, and governance. I'm just reading how, unfortunately, while doctors want you to avoid pollution and all that, but you know, healthcare is the biggest contributor to greenhouse emissions. I think it's so unfortunate. Healthcare industry is 4%. It's responsible for adding to the carbon footprint. And that's unacceptable. In fact, China just published a paper that about the 70% increase in birth defects because of environmental pollution. And this is an absolutely and burning need today. Climate change is upon us. And while we contribute to the issues of, we use a lot of plastic in our pharmaceutical value chain, we use a lot of, so carbon neutrality is something that we should be consciously aware about. Social, our previous speaker already highlighted sustainability and how health is not just about treating, but also considering the social determinants. And some of these perspectives, given the time, I'm happy to take you through. So interestingly, what's the scorecard of the industry? Until yesterday, I just looked up quickly. We have about 84 novel substances that are launched in 2021. Launched, I don't mean approved. Approved is 50. And the total launches in 20 years, about 883 novel compounds have been launched. And interestingly, the orphan drug registration is about 40, out of which first-in-class is about 60%, and others about 40%. And out of the FDA approvals, about 50 FDA approvals last year, although it was dominated by small molecules, but slowly if you look at various reports, be it Evaluate Pharma or Deloitte or any other report, you will see that the biologicals are slowly and slowly getting to be at the center of approvals. So they're gaining ground. So biologicals are in fact perhaps a big competitor to the small molecules when it comes to the therapeutic modalities. And these are all approved. So even oligonucleotides are approved by the FDA. I love this quote by Marshall Goldsmith. What got the industry here won't get us there. And some of the points Arjun already pointed, we need a renaissance. We need a change in the way we work. We need a reboot. We need to reconsider how we're approaching. The traditional models are getting disrupted, and we have new entrants. So data scientists are now as valuable in the pharma industry as is, let's say, a big clinician. So the rock stars of the industry now are data scientists. And I think that's an interesting trend that we are witnessing. Quick reflections, again, I just got this yesterday from the WHO. So what's happening is the use of, again, our speaker alluded to that, the use of AI and machine learning will continue to expand. AI has, and I don't know how many of you are tuned into the 5G and the 6G by 2030, they're going to be smart hospitals. Internet of Things is going to be replaced by Internet of Everything, and that's going to be an interesting space. It's not just Internet of Medical Things, but the new word now is Intelligent Internet of Medical Things. So what's happening is, while the smartphone was dominating and trying to get digital components in, but that's not enough. So smart healthcare will be replaced by intelligent healthcare. So I think those are the buzzwords, you as students, I think you need to get a lot many familiar, because the power of computing has become so incredible. So edge computing, for example, right? So I think the power of computing, I don't know how many of you tuned into the Moore's Law and Clayton Christensen's famous work, a Harvard business professor who kind of said that it will multiply every 2 years, but we've seen that it's happening even faster. So 6G is upon us right now. And the way healthcare will be dispensed is going to be a completely different way. And the pundits are predicting smart hospitals and the interconnectedness of everything. So it's going to be Internet of Everything that's going to be dawning upon us. And therefore, AI has the potential to drastically reduce the amount of time required to develop a new drug. Of course, innovations will occur. We have now virtual arms, we have synthetic arms, that will replace the traditional randomized arm, right, a placebo arm, if you will. So these are quickly what I just thought. I will say some of the things already covered by my previous speaker. So I think the things that matter to all the industry players remains the same. So AI and data analytics. The interesting thing is, when I started many years back in clinical development, we used to go to the power of whatever the gravity, let's say, resided in the hospital. So we designed our protocol to operationalize it around the hospital. Now that is being replaced, we're going to where the patient is. So it's being designed around the patient. SA development, advanced manufacturing, you heard us previous speaker talk, synthetic biology. I just quickly alluded, I want you to really be curious about virtual trials, because that's the next big thing and an opportunity for you to impact there. High performance computing, I already talked about that. Cybersecurity, if we have the time, we'll talk about that. So I think, interesting, this is 2018 data, but I think this has moved on. I think this is 4 years old, but still, nevertheless, the point is we generate lots of data every day. So what, the world generates 2.5 quintillion bytes. This is in 2018. And the point is, when we look at data, the point, it should be, there are FAIR principles. So just remember this, it should be findable, it should be accessible, it should be interoperable, and of course it should be reusable. So the FAIR principles guide data, and of course a very quick snippet on, because industry, we are very privileged to be sitting on big data. So the company I work for, Novartis, does 500 trials a year. So we do generate a lot of data. I think we have the maximum data in all of industry. And big data. So I think these are, again, for the benefit, the 7 Vs, as they call it, about big data. Volume, velocity, variety, veracity, validity, value, volatility. We don't have the time, but I think, so I think I will quickly take you to some quick other trends. I won't take more than a couple of minutes. Again, the point is, look at the fabulous growth of the clinical trial industry. This is only from 2011. I joined in 2000. So 2022, we had 450,000 trials registered. And this was only up till March 2022. So I think it's incredible how the industry is kind of exploding. And clinical trials are generating triple the amount of data than previous decade. Therefore, I have now got some certificates in data science. I had no clue what data science is all about, but I think it was imperative, and I think it's important for all of you to start getting, if you have not already begun the journey into data science. And I think, as I alluded before, patients' experience is the center of that. Again, I won't go into the details, but the trials also are changing. We had traditional randomized controlled trials, right? But now we're looking at umbrella trials. We're looking at a single disease with multiple interventions, umbrella trial. A basket trial is multiple disease with a single intervention. Adaptive, platform, dose ranging, so, and Bayesian. And so we're moving, as I mentioned earlier, from smart healthcare now to intelligent healthcare. And we always measure quality of life, but as you enter the industry and you start seeing that, you start have to looking at quality of services, quality of experiences, and all of those in addition to quality of life. So forces facing, I won't go into this, but there are incredible cost, complexity, risk, and more important, trust issues, right? So to conclude, we need a new kind of leadership. It's not just pharma leader. We need an ecosystem leader. So please challenge yourself to say, how can I be the ecosystem leader of the future? And somebody who's very resilient, and COVID has taught us that, and who can demonstrate great agility, and also invest in new capabilities. We have something on antibody drug conjugates, radioligand therapy. So we need to have newer skills, newer competencies, adopting end-to-end partner ecosystems, and planning for strategic workforce. I already mentioned we are sitting on a huge shortage of talent in the industry. And I'll conclude by saying that we need to really be at the forefront of course, embracing digital. So on that note, I'll stop here, and I really want to thank all of you and the chairpersons for giving me the additional minutes. Thank you so much.
Thank you very much, Dr. Rai. In spite of the fact slide a little bit messed up, you finished within time, so thanks a lot. Any questions from the audience? Maybe I'll start, and then, in the initial phases, you mentioned about the number of drugs approval, particularly coming from US FDA. And recently the trend has been a little bit higher compared to, so it keeps on going up and down, but overall the trend is becoming, and the number of approval really depends upon the regulatory tightness. One of the example is Alzheimer's disease, where US FDA relaxed a little bit, yes, and the drug was approved, and there was so much of feedback. Your opinions and your how your reaction is upon that FDA should relax a little bit, and promote a little bit of more drug approvals, yes, or it depends upon the area to area. So because
yes, so I think to your question on how much appetite there is for risk within the regulatory authorities, in the past 20, 22 years since I'm in the industry, I'm seeing a bit of a dynamic thing. While at times it was, I moved in straight, you might be aware 1996 was the epic year for regulatory when it was a maximum number of approvals. But after that the industry went into kind of a funk, I guess, because of the recalls, and we all know that. Then it went and had a wake-up call and kind of rebooted itself. Today I see a very different kind of a regulatory makeup. Scott Catlett, he's I think incredible in the FDA commissioners. I think the quality of interactions we are having before at the IND stage or pre-IND discussions, I think the regulators are partnering with us because we are also doing, we've moved into a very bold area, we're doing bold science, we are really doing incredible, and I think of course the failure rate is high because the science has gone so much deeper and so much bigger and bolder. But the interesting thing is regulators are willing to even accept real world evidence data. And so I'm thinking the regulators are not seeing it as putting in enough safety measures around the approvals and giving us time to keep developing the asset. So while the science has become bolder, the attrition has gone higher. Of course, that's a fact. That's why drug development costs have gone more. But the FDA is partnering. I mean, the health authorities, even I was at a statistics regulatory workshop recently, and interestingly, that was run by regulators of Europe. So I think there is so much appetite for doing it together and collaborating. And Transliterate, I don't know how many of you tuned into the latest thing. I really encourage students to study Transliterate. I think that's a great thing because they're also looking at ICH and saying, can we revisit it? So they are introspecting as well, and I think the industry is working quite well with the regulators now. There's a sweet spot and the trust is building back again slowly.
Yeah, thank you. That exactly, I can elaborate a little bit on the US FDA. You can have so many pre-IND meetings without spending a single penny. And that is all to help. Any other question from the audience? Yes.
Okay, please come in.
Thank you. It's for the incredible information, and I saw a lot of pharmaceuticals also where you mentioned of artificial intelligence, machine learning and data science, which has become going to be important. And one of the things which you mentioned on the patient-centric approach, and also we are seeing in the wellness industry, like a lot of personalization is need of personalization is coming up. Like one shoe doesn't fit all kind of approach, right? So is there any also you have insight on the personalization in the drug discovery or the?
Yes. So the problem is I have the mic. I can keep talking because this is my favorite topic. But yes, the answer is a quick yes, because that is what drives us. Because while we have 10,000 diseases that we know, the treatments exist for only 500 and they're all mostly symptomatic. So we are moving into prevent and cure, and we are understanding biology. We still don't know a lot. We know only perhaps maybe 5% of what we need to know. So I think as we're moving into that, precision medicine is at the heart of what innovative pharma is all about. So yeah, we can have we can take it offline, but it is at the heart of what we do. So, sorry for that very abrupt.
Thank you. Thank you.
Thank you. Thank you very much. Any other question? If not, let's give a big hand to Dr. Rai. Oh, there was.
Thank you, everybody.
Dr. Rai, 1 second.
Hello.
So we would like to present this memento to Dr. Kamla Rai for her.
Ma'am, my question is there.
Yeah, I think you have a question.
Yeah, ma'am.
There is, I can't see. Okay.
Okay, sorry. मैं दूर बैठी हूँ. Good afternoon, ma'am. And thank you to give this a beautiful knowledge. Artificial intelligence is my favorite topic because I'm working on it. So I want to ask that one word you are using again and again, that is a smart hospital somewhere else. So what it means that robots, they're smart hospital, or the dosage we are giving, that is in an automated way. What it means, I'm confused in that word.
It means that also, but I think smart hospital means that it's all connected. It's connected. I mean, the patient experience is connected to the hospital. It's not disconnected. Right now, the traditional model is all disconnected, right? Between two visits, the physician doesn't know what's happening with the patient. So I think when we move to the smart hospitals, again, I can keep talking, but I'll take it offline. I'll tell you what I mean. There's a very nice white paper on smart hospitals. It's all about connected health. How 6G, right now metaverse is changing the dynamic of healthcare. And although metaverse is not, it's 1992 concept, but augmented reality, and when they start building edge, because right now, you know, what's happening is the systems are disconnected. Although they may be digitized, they're disconnected. And it doesn't mean robots will take over, but I think what I see happening is the connectedness of health. When one smartphone and the physician's smartphone and all that will be an intelligent phone, not just a smartphone, and then exchange data much more seamlessly. And so decisions will be real time, and quality of interventions will be so much more better.
Okay, thank you so much, ma'am.
Thank you so much, sir. What a pleasure. Thank you so much. Thank you so much.
We would also like to invite Mr. Arjun on stage. We missed you presenting the memento. Sorry for that. Please. We now move over to the next speaker in today's session, Dr. Sumit Madan. Dr. Sumit Madan is currently working as Associate Vice President, Product Development Research at Sun Pharma's R&D Center at Gurgaon. He had completed his Master's in Pharmaceutical Sciences from Delhi Institute of Pharmaceutical Science and Research, and he did his PhD in the field of biomedical engineering from Indian Institute of Technology, Delhi. He specializes in formulation development of oral solid dosage forms, 505(b)(2) products, and contributing to CMC section of regulatory filings. So with this brief introduction, over to you, Dr. Sumit Madan.
Okay, hi, good afternoon, everyone. First of all, I would like to thank the chairpersons for such kind words to introduce myself, and thanks to NIPER and the organizers, specifically Professor Bansal, for inviting me to deliver a talk at this grand event at NIPER. So, for today, the topic which I have selected to talk about is an area which is bothering for the pharma industry since last 3 to 4 years now. And I strongly feel that today it's the time that academia needs to step up, step in, and come to collaborate with the industry to address this bothering concern. So the topic is achieving nitrosamine controlled drug formulations, an opportunity for academia industry collaborations. This presentation reflects my views, and these are not the views of Sun Pharma. I will start with an introduction why nitrosamines are a concern as in date, the risk assessment and control for nitrosamines, what is the approach which is being followed both for the APIs as well as for the drug product, a perspective on the acceptable intake limits, safety and toxicity assessment, and where can academia step in to support the industry. I hope most of the people sitting here would know what is a nitrosamine. I just refresh everyone and give a brief introduction. Nitrosamine means a class of compounds having the chemical structure of a nitroso group bonded to an amine. They are also referred to as NOCs or N-nitroso compounds. When is the nitrosamine formed? Nitrosamine is formed when there are secondary, tertiary or quaternary amines which interact with nitrite salts under acidic conditions. Because under these acidic conditions, the nitrite salts form nitrous acid, which reacts with an amine to form a nitrosamine. As such, nitrosamines are not, which is something new. They have always been present in the environment. Nitrosamines are common in food, water, tobacco. The chemistry is not new, the potency is not new, the toxicity is not new. Everyone, all of us have been exposed to some level of nitrosamines. And publications as old as 1968, they do report that when nitrites and secondary amines were concurrently fed to the animals, tumors were produced. Nitrosamines are genotoxic, they are carcinogenic, they are part of ICH M7 cohort of concern. The pharma industry got sensitized in 2019 specifically, and nitrosamines popped up as a concern when FDA and other international agencies started finding traces of nitrosamines in the sartans. Specifically, it started with valsartan in June 2018, when FDA was informed of the presence of an impurity, NDMA, in valsartan. Subsequent to that, other sartans like losartan and irbesartan formulations from different manufacturers across the world, they also reported unacceptable levels of nitrosamines. And after sartans, then there were reports in ranitidine, nizatidine, and metformin. All this was early 2018, and then the end of 2018, early 2019, when these nitrosamines got reported, all this made clear that there was a need for a risk assessment strategy for potential nitrosamines in any pharmaceutical product where there was a risk for their presence. And as we have moved from 2018 to 19 to current 2022, we see that there has been an evolution of the understanding. Evolution of the entire industry has taken place with regard to understanding the nitrosamines. Before 2021, all the recalls which took in US specifically, they involved small and more common nitrosamines like NDMA and NDA. Focus was majorly on the 8 nitrosamines, which all the regulatory agencies have also listed. And most of these smaller nitrosamines were present in the pharmaceuticals due to the contribution from the reagents or the solvents used in the API. Post 2021, what we see is now additional nitroso impurities from the API itself, the raw materials, the reagents are gaining tremendous importance. And what we call these other NDSRIs, which are the nitrosamine drug substance related impurities. Means these are those impurities which are formed when the API itself is a secondary amine or a tertiary amine. And it is capable of nitrosating under suitable conditions. NDSRIs are formed when the API, the intermediate used in the route of synthesis or the impurities of the API are secondary or tertiary amines. Example of the recalls due to NDSRIs include varenicline, which was due to N-nitrosovarenicline, and in case of quinapril HCTZ, which was a recent one in March 2022, when Pfizer recalled due to N-nitrosoquinapril. I'll quickly skip this. Step 1, Step 2, Step 3 is something which has been published by all the regulatory agencies in terms of risk assessment for the nitrosamines. As a Step 1, you need to do a risk evaluation to identify whether the API or the finished product is at risk of nitrosamine. Step 2, if the risk is identified, proceed with the confirmatory testing. And if the presence of nitrosamine is confirmed, that's a Step 3. We need to implement effective risk mitigating measures through submission of variation. These are the limits which are proposed by various agencies now for all the known nitrosamines. FDA, EMA, Health Canada, and Visa, they have published their acceptable intake limits, which are almost similar, but these are only for the published known nitrosamines till now. When we talk about the risk factors associated with the nitrosamines, we start with the API. When we talk about the API, it could be use of nitrite salts and esters. It could be the nitrite formation by the oxidation of hydroxylamine or the nitrate release from nitroaromatic precursors. It could be use of disinfected water in the presence of secondary or tertiary amines. It could be oxidation of hydrazine, hydroxides and hydrozones by hypochlorite, air or oxygen. So that means even oxidation is also a trigger sometimes for oxidation of these hydrazines and hydroxides, which leads to nitrosamines. Use of contaminated raw materials or starting materials or intermediates and carryover of nitrosamines deliberately generated. So these are the 6 risk factors which we need to identify when we are doing a risk assessment for the API. And with regard to the manufacturing aspects, with regard to the manufacturing area and the facility, the GMP facility, we need to consider the cross-contamination due to different processes being run successively in the same manufacturing line, carryover of the impurities, or use of contaminated, recovered or recycled materials where the recovery is outsourced to third parties. So, because recovery processes carried out in non-dedicated equipment also needs to be considered, and there could be a contribution from these aspects as well. When we come to the risk identification, when we start the process of risk identification for the API, we need to firstly, the first basic test is we need to look at the structure of the API, whether there is a risk of NDSRI or not. Because there could be the APIs like varenicline, propranolol, brinapril, where itself the structure itself indicates that there is a secondary amine or a tertiary amine. And going forward, currently what we are focusing on is secondary, tertiary, quaternary amines. Going forward, we are anticipating risks could even be due to N-alkyl amide, amide, carbamate, or any other functional group which can be nitrosated in the API. There is a risk for nitrosamine formation. Then there are APIs which do not have the structural alert for nitrosamines, but the reagents or the solvents which are used in the manufacturing process may involve nitrite or nitrate, which could nitrosate. For example, in case of the valsartan recall, which happened when the FDA recalled valsartan, NDMA was identified as a contaminated, and the contribution for NDMA was due to the API process, which used DMF as a solvent and sodium nitrite as a quenching agent. Similarly, in case of ranitidine, when nitrosamines were observed, it was due to nitromethane, which was used as the nitrosating agent, which could have served as a nitrosating agent. Quality of water, which is used in the manufacturing facilities, both at API and formulation, is again a critical factor which needs to be considered. Potable or the deionized water may have nitrite, and NOCs, which needs to be seriously evaluated. Even the shared equipment should be free of nitrite, nitrates, and amines. So, quickly I'll go through the questions which need to be answered for evaluation of the API. Whether the starting material have possibility of presence of nitrosamines, whether the manufacturing process involves recycled solvents, any of the recycled solvents have amines, NOCs, or nitrosating agents. If nitrosamines are being monitored in the API, then how are the acceptance criteria determined? Are there sensitive analytical methods in place for these nitrosamines? Is the facility that makes the API dedicated to make only this API? Are sartans or any other API with known risks related to nitrosamines being made in the same facility? And does the cleaning validation program take into account the formation of presence of nitrosamines? So once the risk assessment for API is done, next we need to consider the controls. When we talk about the controls, we need to optimize, so that is more at the API end, but even we as formulation scientists are ultimately responsible for making the finished product in dosage form. We need to be sensitive enough that is the control of nitrosamines being done in the API or not. So the approaches which are followed for control of nitrosamines in the API include avoiding the reaction conditions that may produce nitrosamines. Like in pioglitazone, it was found that the NDMA formation was avoidable by eliminating DMF as a nitrosable solvent. And specifically, acidic conditions promote the formation of nitrosamines. Generally, when we move to neutral or alkaline conditions, the tendency to form nitrosamines gets reduced. Then we have to be cautious when using the solvents like DMF. The process needs to be designed so that which can facilitate the purge of nitrosamine impurities. Removing the quenching steps from main reaction mixture, avoiding cross-contamination. Based on the risk analysis, we need to establish the safety levels of the API, provide a control strategy, and if needed, control the relevant NOCs in the intermediates or the API. Once the risk assessment for the API is done, now we come to the risk assessment for the drug product, where the root cause analysis would involve the study of nitrosating capability of the formulation. So this can be done by using a common amine, mixing it with a mixture of different nitrites and see whether there is a nitrosamine capability of the formulation or not. Major source of nitrosamine impurities in the drug product are the APIs. Because API may comprise amine, alkylated amides, any amine, if it is there, a secondary, a quaternary, a tertiary amine, we are at risk. But in formulation, what we can do is we need to cut down the source of nitrites and nitrates. And the major source of nitrites in DP are the excipients. So when the drug substance, I'll move to, so there are some excipients like prolamine and FDNC colors which have nitrosamine as impurities. Specifically, much common excipients which we use as microcrystalline cellulose, pregel starch, sodium starch glycolate, povidone, all these have residual nitrites and nitrates, and they are the excipients which we need to critically evaluate as formulation scientists to ensure that we do not have nitrosating agents in our formulation. So controlling nitrites and nitrates in excipients is not easy. It's a challenge. In rare instances, rather, excipients may have some amine residues which could react with the nitrite residues in other excipients, and then they can form nitrosamines. So a recent report indicated that in the production of water for injection, where residual chloramine was used to disinfect the incoming water, it reacted with dimethylamine leaching from the anion exchange resin used in the demineralization step, and it formed NDNA. So water is critical, excipients are critical, even plastic and rubber are potential source of NDBA. So even when these samples are being tested, or analytical colleagues, analytical scientists, when they are testing, if they are using plastic syringes or nylon filters, they may increase nitroso compounds, and you may have a wrong analysis and a wrong indication that nitrosamines are there in your product. Packaging materials for the drug product, which are composed of rubber or other elastomers, also contribute to nitrosamines. So finished products which are packed in blister packs, which have nitrocellulose, they also contribute to formation of nitrosamines in the product. And as I said, pH of the formulation is critical, both in API and formulation is critical. So when we talk about controlling the nitrosamine in the drug product, we need to, as a formulation person, a formulation scientist would always give a preference that the API should be free of amines, but that is not possible. So what we need to do is we need to cut down the source of nitrites and nitrates. Since in the view of time, I'll move to important part. Acceptable intake limits, I think what we need to understand is that all the nitrosamines are not carcinogenic also. The size of the NDSRI, the nature of the substitution can play a role in determining whether that NDSRI is carcinogenic or not. And in most of the cases, the surrogate approach is used. The Tanimoto score is used, wherein what we do is, if there is any new nitrosamine or a novel nitrosamine, we try to find out a molecule with a structural similarity to that nitrosamine for whom there is some toxicity assessment available. And based on that, the acceptable intake is proposed. And in terms of safety and toxicity assessment, in silico study with direct nexus and SARA-based and database kind of softwares are available which provide some toxicity assessment. But Ames test is something which is not recommended by the FDA, and in vivo assays, transgenic assays, bacterial mutagenicity assays is something which FDA expects to prove that the product or the API is free of nitrosamine toxicity. So the industry is dependent on the surrogate methodology and matching up novel nitrosamines with structurally similar nitrosamine that have already been tested in rodent carcinogenicity assays. So I'll quickly go through the main points which I want to speak about today. Where can academia step in and support the industry, and where do we need an academia-industry collaboration? First, to start with at the API level. As every API is different, so the methods which are related to each NDSRI is distinctive, and it takes time and resource to develop. Academia can focus research on development of further improved analytical methods for NDSRIs, methods that are more rapid, with improved screening, methods that are not overly cost prohibitive and which can be adopted easily. Currently, industry, we are heavily relied upon LCMS and GCMS techniques for analysis of nitrosamine impurities. These techniques are important during the confirmation of the presence of a particular nitrosamine, but these are again costly, these are not easily available, and they can also give false positives due to the presence of artifacts arising from the formulation. Academia can work to identify alternative analytical methods which are fast, which are reasonably priced and common, and that can be easily used on a regular basis for NDSRIs, which need to be checked on release and stability. It's not an easy task, but this is one area where I strongly feel if academia can put in more resources, more time, because industry is busy handling the current workload which is there. The current burden which has come or whatever due to the alarm which has come due to the nitrosamines, industry is busy handling the routine products, the already commercialized products. So if academia can focus on developing good analytical methods, here's an opportunity for us to collaborate. Secondly, at the drug product level, fast, affordable, and precise determination of nitrite and nitrate in reagents is critical. And this may be considered as the first step towards understanding the risk of nitrosamines. So, academia can support by identifying the root cause of nitrites in excipients or developing commercial process for manufacturing nitrite-free excipients. Currently, some suppliers for microcrystalline cellulose, for example, have changed their processes to come up with nitrite-free, or it's not nitrite-free, it's low amount of nitrite. But if we can work out and spend some more time into basic research as to why these excipients have such high level of nitrates and can we cut down the source of nitrates, this will help resolving the nitrosamine issues in the formulations. If we can develop analytical methods for testing nitrites and nitrates in any such excipient where there is a possibility of presence of nitrite and nitrate, that could be another contribution to it. And thirdly, one of the most important help which can be done by academia in the formulation is if we can evaluate new nitrite scavengers which are acceptable as per IID. There are nitrite scavengers reported in academia, it's many, but only ascorbic acid and alpha tocopherol are currently acceptable within the IID levels. If academia can do some research, come up with more nitrite scavengers which are acceptable as per IID and we do not interfere with the bioavailability of formulations, there is a great scope to contribute to numerous products and resolve the nitrosamine concern in the formulations. Then, in terms of setting the acceptable intake limits, if academia can support through the in silico screening for checking the mutagenic potential of impurities, or support research on alternate in vivo assays that can be considered adequate to qualify an NDSRI. The target is definitely speed within decent cost. Academia can also assist to justify the approach strengthened by the scientific basis of molecular weight correction, which currently EMA and Health Canada have not accepted. FDA is also silent on that. But if we could work out some scientific rationale on this or come up with an alternate approach to predict the acceptable limit of NDSRIs based on a surrogate method, that would be really helpful. And one of the challenges for the industry currently is to obtain suitable reference standards for nitrosamines being tested. So academia could explore this area also to proactively support with the availability of nitrosamine impurities or reference standards for the APIs which are secondary tertiary amines. This could be helpful, and even if it is not at a GLP level, it's just a preliminary work at exploratory level or non-GLP levels, that can also be a part of collaboration to get an early indication and a quick screening. So this is my last slide. I'll just conclude. In terms of the risk assessment, I have talked about the API needs to be thoroughly screened to understand the role of all the raw starting materials, intermediate solvents and everything. Next important step in terms of finished product is to cut down the source of nitrites and nitrates. Excipients containing nitrites and nitrates like MCC, previous starch, SSG, povidone needs to be screened diligently. Alkalizers have been helpful to achieve nitrosamine control. Antioxidants like ascorbic acid are acting like nitrate scavengers. We need much more than ascorbic acid. For NDSRIs, strategy should be to find out the appropriate surrogate to read through study and justify the ADI in wider limits. So I will conclude by the statement that nitrosamine controlled drug formulations are possible to achieve with very low and acceptable quantities. If we do a timely risk assessment, if we have a better understanding of the mechanism of formation, if we have more powerful analytical methods, if we timely execute the control strategies, and all of the above can be facilitated by an active academia industry collaboration. Thank you.
Thank you very much, Dr. Madan. The problem is huge in the industry, so questions.
What you mentioned is, I think, really a very alarming and specifically if it's coming from a very common excipient which we are using in formulation, right? So is that these excipients also have some standards now on nitrate contents, like because most of them are like pharmacopoeial standards, right? So is there any controls coming from there also on their standardizations, or we have to do all the, I mean,
in USP, nitrosamines appeared for the first time just 3 years back in 2019, and then it got implemented in September 2020. In European Pharmacopoeia, again, it got published for the first time in late 2019 and got implemented in 2021. In IP also, general chapter, it has been introduced just now in 2 months back, although there is no date of implementation. But there is a guidance in general chapters of all the pharmacopoeias now, just 2 years back these have been introduced, which talk about all these risk assessment and everything. And there is a basic guidance as to how we need to proceed with the risk identification, the risk assessment at API stage, at the formulation stage, and all the excipient stages. Now, when we come to excipient by excipient, currently there are no pharmacopoeial limits. But the industry is sensitized, the excipient manufacturers are sensitized. If you talk to any excipient manufacturer or MCC manufacturer, he is aware that what is the level of nitrite or nitrate which is expected. Currently, they don't have the controls. A lot of work is needed for that.
So even they don't have a like kind of current control limits for them, right?
They have the limits, but we need to go much below. For example, I'll just share with you. For example, Ruket provides MCC, DuPont provides MCC. The limits are around 4.5 ppm. I'm talking about ppm, but 4.5 ppm is also high. We need to talk about ppb limits now, because otherwise controlling, if any traces of nitrates and nitrates are there in the excipients, they react with the amines. And you look at the APIs which are already approved worldwide till now. Many have secondary amines, many have tertiary amines, many have quaternary amines. So API you can't stop now. These are the APIs we have. In terms of excipients, we have to cut down the nitrates and the nitrates.
Thanks, Sumit. I mean, it was really very important topic, and you touched base on. Thank you for that insight.
Yeah, there's one quick question and quick answer.
So very good presentations, Dr. Madan. So we are also working, being the academia or Institute of National Importance in NIPER Ahmedabad. We are working from last couple of years, and we have almost covered whatever you were talking about on sensitive methods. In fact, we will be having ISO as well as NABL accredited lab for that. So quick question I have for your presentations. You are talking about nitrates and nitrate both, but why nitrate? Because generally nitrate cannot convert to a nitrite to do a nitroso.
I think here's my presentation. Because nitrate can convert. So and it can, under the acidic conditions, it may form nitrous oxide, and then it may react.
Okay, means I will try to catch up later, but because nitrate converts to nitrite, it generally are reductions, and which can happen only if there is a metallic impurities and other things are present, but otherwise they are not there.
Right, but so, nitrate can convert into nitrous oxide, and then it may...
Okay. And another thing what we are doing currently is in generating an in situ NDSRI. So we have a protocol developed for any of the molecule because the standards are not available. So we also get a lot of queries from different companies like Sendos and a lot of other companies. So they are also not getting NDSRI standards.
They are also not getting
standards. So what we have generated is we are generating an in-house, in situ generation of NDSRI and trying to do a weight percent or a RRF based on the inverse corona CAD and advanced analytical technique. But is it acceptable where the standards having a RRF means whatever we are generating is not 100% pure?
As I said, it will help us in the early screening.
Makes
it will
sense.
give us a quick indication. Currently what we are doing is whenever there's an alarm or there's a new nitrosamine or a novel nitrosamine with which we become aware, we need to first prepare those standards and everything. If we can get support, so that will help us in an early screening and quick indication whether there is some risk or we are...
Yeah, means we are supporting some of the industries.
I think we can discuss it later, and Dr. Sumit Madan has given a very extensive overview, and we would like, on behalf of the organizing committee, we would like to recognize him.
We move over to the next speaker now, Mr. Ashish Gogia. Mr. Ashish Gogia is Executive Vice President, R&D at Medrich Meiji, Bengaluru. He has worked in several leading pharmaceutical organizations in the product development and research department, like Ranbaxy Research Labs, Aurobindo Pharma, MacLeod's Pharma, Cadila Pharmaceuticals. He is a master's in pharmacy from Delhi University, and today would be talking on a perspective from the CDMO side on creating portfolio of complex enabling technologies in CDMO environment. Over to you, Mr. Gogia.
Thank you, Doctor. Very good afternoon to all of you. And first of all, I would like to extend my gratitude and thanks to NIPER Organizing Committee for inviting me and representing our views. So on behalf of Medrich and Meiji, I would like to first take a few minutes to introduce my group. And as the previous speaker mentioned, I may be going for dumping of many slides over here, so please don't get scared about it. It is just to showcase you the emblem, the kind of technological words we feel, and we are going to show the delivery of the drugs which are going to happen in the future. Currently, our span is for generic drugs, what we do here, but we are aspiring to become one of the delivery house in India, Bangalore, by not only conceptualizing the product at our R&D, converting those thought process from R&D, making batches, to our clinical batch manufacturing site in Bengaluru, and then post-approval, those products should reach out to the market which are based in Japan as our primary market, followed by Europe, Canada, Australia, New Zealand. At the moment, we are not much into the US, so I will be restricting my discussion towards the group which I represent, and I'll be little more on the fast-forward manner, keeping these time constraints about it, so please forgive me for that part. So Medrich is a part of a major pharma giant in Japan called Meiji Group, and we represent in India their CMO and CDMO arm. We do the business with the major clients, and this association happened in the year 2015. Now in 2022, we are 47-year-old existence in India, but now we are known as the group of Meiji. This is the group we are talking. It is known for more chocolates, confectionery, dairy products, and we are the part of ethical pharmaceutical group. Meiji Seika Pharma Company pharmaceutical segment. In this segment, they have two arms. One, they call it as their innovation pipeline. Another is called as the generic segment. So the generic segment is now the Medrich, which takes care of this operation from India. This is the global map, what we operate. We are based here in Bangalore, but we do the business globally through our subsidiaries here. And I'm particularly talking about the space, how the CDMO companies operate. We don't have our own products to sell to the customer. However, we provide services to many pharma clients for producing the cutting-edge technology drug deliveries options from these products. In total, we have the regulatory approvals from UK, Australia, Canada, Romania, and also we have included for PMDA Japan. We have close to 220 scientists engaged at this center in Bangalore. We have 30 officials in regulatory affairs, and we primarily focus on the oral solid dosage forms. This slide is more like showing the engagement that year on year, or I should say month on month, every month, how many studies go through our clinical bioequivalence study programs, and literally it is every week one product is going. So which makes around 36 to 48 products, first time developed in our R&D center, going through the clinical batch manufacturing, and then putting it across for the registration market. So this is just a track record to showcase that engagement. These are the photographs of our center. I am based here at Bengaluru in R&D, whereas all our GMP operations happen through these sites. And this site is particularly dedicated for our Japan portfolio of projects, which gets manufactured, then supplied to Japan market as our unique capabilities toward it. Now, coming a little bit out of Medreich and explaining about the CDMO scenario, so I thought it is better to give me a context here that India is a hub which does a lot of exports in the pharmaceuticals, but in terms of the revenues or the valuation, we hardly get in the ratio what we expect from the volume. So the volume speaks the language, but the values doesn't speak the language. Why? Probably this is the reason which we got it from it, and this was our broad understanding. We mostly operate from India on intermediate and bulk, commodity generics, conventional dosage form, and maybe this decade of 2020 onwards, we are seeing a trends happening of newer and complex formulations going as a value-added and branded generics. The future definitely lies if India can have the capability to be associated with NIPER and in fact brainstorm towards it, how we can escalate this kind of a capabilities in India and produce more and more into the higher margins part. Imagine if with the same volumes, keeping this kind of a margin, the effect on economy, the effect of products which we'll be making will be an outstanding performance from the group here. So my talks will be mostly covering on the topic which we make is how many complex formulations we have done so far historically. So if I see from the last 6-7 years and what we are going to do in the next 5 to 10 years, this is the emblem through a case studies model and presenting the actual live products, what we are developing, what we have developed, what we are filing or commercialized. I thought it is best to explain to my fellow scientists who are in this field and would be prospering to come and do in the similar environment of developing more and more high-tech enabled products. So the first case study which I would like to put across is the multi-unit particulate system. In India, my colleague Sumit and the earlier fellows have explained, these are the big companies who do a lot of research, commercialization. They have their own capabilities to do multi-unit particulate system dosage form, particularly for export to the regulated market like Japan, US, and Europe. But what about others? They look for such kind of an opportunities. Can we make an affordable dosage form by putting across a GMP or a R&D site and put these ideas into the action, even if they don't have that kind of a capabilities of their own? So CDMOs become a definitive answer in such situation. This is the product I'm naming here is Europidil, which was developed, started somewhere in 2010. And now in 2022, it has seen the light of a full-blown commercial at our site in Bengaluru. And it has gone through a lot of stages of complexities and all, which you will be able to see here. Initially, it was originated in 3 years, we should be able to commercialize it, but it took 8 to 10 years to reach to this level. So the brand name is Abrantel, 3 strains. It is available in Europe. Not much literature is available. We were asked to do the reverse engineering. These are the major excipients which control the drug product. Pellets are a population. These are all white colored in a capsule, but we don't know which percentage of pellets are of what release, how many immediate, how many sustained, how many modified release. So we did this study. These are the 5 major performances. Initial 2 attempts failed. Next 3 we passed, and that's how this product got commercialized, and we are now making this product. But the complexity never resides only on solving the product in development or in the beginning. It starts when you are in the commercial mode. The batches were taken in the development, maximum 58 kgs. Dossier got approved from the Europe. Now the company is making today a batch of 1.3 tons, and we have been asked to produce such batches, minimum 45 to 50 in a year. Any equipment available which is commercially viable facility, they cannot produce more than 12 batches a year depending on the complex cycle of this product. So this is the kind of complexity we deal in oral solid, and now we are making efforts to further make more and more smarter way of producing these kind of products in a pellets.
Another angle of complexity here is proving to the agency when you have done the bioequivalence studies, the 5 studies plan which I have shown you, the moment you change the manufacturing site, the moment you change your scaling up from 58 kgs to 300 kg and then 300 kgs to 1.4 ton, they ask for repeat bio studies. How many studies you will do? What are the possibility of getting those approvals? So we took the approvals through IVIVC model here, and the registration was granted through this successful complexity model, and now we are in the happy space of commercialization. Some cases we resolve, it took time, but some cases we have just started the journey where we are still not able to resolve, and we'll be looking forward to the NIPER and Professor Bansal and his lab and many more experts in the academia to tell us how to solve this kind of a problem. So this is the second product, barnidipine. It is well there in the market, no competitor. People are asking us to develop the product. We could not see the light of the day of this formulation due to this reason. 99% of the barnidipine goes out of the system without even absorbing. The absorbed drug is only 1% of barnidipine, and within that 1% drug, it is so highly variable, 55% of the variability built in the compound. Sometimes it shows bioavailable, sometimes it doesn't show the bioavailable. How to get the approval of such kind of a product? So the study in fasting shows the drug product we developed is faster. In the fed, it shows we are drastically poorly bioavailable into it. So converting such opportunities, further brainstorming, putting the investments, and resolving is the future, something which will be depending upon taking the expertise from NIPER and trying to convert them into the commercial space here. Third important MUPS case is the MUPS ODT dosage form. This, according to our assessment, is a very, very niche area. Even though these many 11 MUPS ODT products are available in the market, no Indian player other than the top 3 or top 5 players are able to export such kind of formulations to Japan, to Europe, to US. They are restricting themselves to make such kind of a products in a capsule dosage form and exporting it, but not in a tablet form. Obviously, there must be some barrier. Obviously, there is a complexity. So this is the complexity. When you have to produce a tablet which contains 2 distinct particles, one which is a very, very heavy particle like a pellet, the other one is a powder, mix them into the tablet and prove to the regulatory agencies through all the yardstick, is going to be challenging. So this is the typical diagram how we have been conceiving this product. It starts with the small pellet. The red one is the drug which gets loaded, then start intermediate, enteric, different enteric, blend them, compress them, and which should give a right kind of a performance. And if you look at it at the typical composition, in our terminology, we call it as Gola technology. Gola means it is so many goal rounds here and the prefaces that you have to keep optimizing what it contains at each Gola layer to get the desired output of the MUPS tablet. But the problem doesn't happen if you have successfully designed a good formulation of MUPS tablet. The problem doesn't finish if you have a good impurity control. The problem is if you have to produce 2 tons of a tablet of a MUPS, how are you going to commercialize it? This product has a tremendous problem in the segregations. So this is what we have observed. And we came up with the answer like this. We were going for $1 million investment in putting a plant which will produce day in and day out, putting the international machine with a single hopper to produce it through gravity feeding. And we said, no, this cannot be the answer. The answer lies in these kind of machines which are still to take place in Indian pharma industry. Very few players, but Medric is the one which has invested now into this kind of capabilities. The second broad category of the complex technologies which we are imbibing at our place is we call it in a drug delivery system, and which is a disruptive technology. It is more than 30-year-old technology, but it's still no replicas, no generic players, no CDMO services. How to get an Oros product into any of these technologies called mono, bi, or tri-layer. This is Adalat product, very famous nifedipine. We are now the biggest manufacturer of nifedipine, laser drilled at our site, developed at R&D in a non-GMP environment, meaning having a laser drilling capability on the product at R&D site, followed by a mid-segment in a GMP, in a clinical trial segment, and post-approval on a commercial scale. So these kind of investments on drug delivery is only to facilitate that we enter into providing affordable but high-tech healthcare product for the industry. So this is the actual machine which is lying in our GMP location, and these are the typical programs. You have a bilayer tablet. The machine will segregate the drug layer containing the coated one, the colored one, and the below is the placebo layer. So it will flip, and only the laser drilling will happen on the drug portion. It will not happen on the placebo portion. And this tablet will come out and will produce a 24-hour drug release. This is the benchmarking we have done through the studies on the size of the laser drilling hole. And these are the bioequivalence studies. Initial failure, but we went on believing the capabilities of the group that yes, we will be able to achieve it if we are on a good scientific path, and now we are there in the bioequivalence meeting the criteria, and dossier is filed. We have another interesting product coming in the same laser drilling system, but it's a tri-layer tablet. You not only have to produce bi, but this time it is going to be a tri-layer and still a laser drilling system. We have an opportunity in a mono-layer tablet, which is a laser drilling, where the patent is only on the side of the tablet. It is not on the center. So they have created a release pattern of this product by drilling on the sides of it, and we have been asked to produce a formulation which is bioequivalent, but not following the patent because the patent is valid till another 7 to 10 years. Third option, very quickly, I wanted to put it, which is the industry we are doing, and a lot of work happens here also. The crystalline to amorphous strategies work using many equipments. But the one biggie failures which has come as an industry failure is from the patent point of view. Many of the industry launched the product Apixaban in the US thinking they are making a good non-infringing formulation. They have converted successfully a crystalline form to the amorphous form using spray drying or using other technologies. All of them has been blocked. None of them is allowed to commercialize. In fact, the whole process has gone back now to the drawing board, and we have been asked to redevelop as an industry, and people are afraid because the innovator company has proven that in the formulation, XRD is not the good technique. It is an inferior technique. So you have to look for a better technique to prove that the amorphous form is intact, and any contamination of crystalline in the dosage form is considered patent infringing. So these are the trials which are happening in our lab, whereby we are trying to first maneuverate with more sophisticated equipments to prove that we are not having any crystalline moiety. The option is we are moving towards liquid-filled hard gelatin capsule. Option is we are moving towards trial in hot melt extrusion and some other designing ideas here. Abuse deterrent, we are working on it. We have not yet started. We have a facility which deals in only these opioids controlled substance in Bangalore. We are the largest importer of these drugs, and we wanted to convert this portfolio from a conventional dosage forms of morphine, codeine, and such products into an abuse deterrent categories. A lot of opportunities here. I will not go details about it, but idea is just to give you this, that it has happened in the last 7 to 8 years. US products, major opioid categories are there, and we, as a CDMO, got the project signed off to develop them and launch into the market in the next 3 to 4 years of time. That's all from my side. This is the team which is responsible for doing it, and thank you very much for paying attention to it.
Thank you very much, Mr. Ashish Gogia. And in interest of time, because this stage has to be prepared for the next event, so we will not invite any questions. And we would like to recognize Mr. Ashok Gogia for his contribution. On behalf of NIPER, we want to present this to you.
Thank you, sir. Thank you very much, sir. Thank you.
The next speaker is Dr. Anil Kukreja. who is Vice President, Medical Affairs and Regulatory Affairs in AstraZeneca, Bengaluru. He had completed his Bachelor of Medicine from Government College, Nagpur, and his Doctor of Medicine from Grant Medical College. He had previously worked with companies like Wockhardt Limited and Roche. He also worked as consultant for various startup companies. So with this brief introduction, we invite Dr. Anil Kukareja for his talk, please.
Thank you, respected chairpersons, for this opportunity. And I really want to start by thanking the organizers of NIPER PharmaCon, as well as OPPI, for providing this opportunity to be on this dais and speak to this august audience. My topic, of course, is very broad, and I kept, as I think I heard Dr. Kamala Rai also, I am also in the same stage where I have stopped preparing the slides. But as we know, all the professors, usually we give it to our students as well, so I gave it to one of my budding medical affairs colleague, and he put across the slides for me, and I will be acknowledging him at the end so that we really contribute and acknowledge particularly the students who are contributing for the same. I want to start this topic, I'm sure each one of us, and we have been enabled and being here today without mask is thanks to all the researchers, companies, doctors, nurses, and all the manufacturers who have contributed and brought the various vaccines to this world, and we have been able to control and come out of pandemic in a reasonably, I would say, a huge contribution and applause, and I would say kudos and salute to all the people who have really worked in this direction. Topic is related because we are talking about biologics, and vaccines is one of the biologics, and therefore I wanted to start with saluting all the people who really contributed for bringing the vaccines. I'm sure this august audience is aware and keeping track of all the headlines which have made in the recent past. And we are talking about the future of medicine. And if you see the future of medicine, it is moving from, as we also saw in the FDA approvals in the talk from Dr. Kamala Rai, that we are gradually going from chemical compounds to the biologics. And if you look at the future of medicine, we are talking about the immunotherapy, we are talking about the immune checkpoint inhibitors, we are talking about CAR T cell therapy, we are talking about the gene therapy revolution. And of course, we are talking about, can we reverse the aging by having a youthful stem cells incorporated into the patient population. So these are making the headlines, and I'm sure these are the future trends we need to be very, very watchful for. Again, with the help of the same slide, the same message, small molecules to plasma and recombinant proteins, polyclonal and monoclonal antibodies. And the journey doesn't stop here. The journey is taking, how do we make the antibody a long-acting antibody? And there are examples where long-acting antibodies have been approved not only for the management of COVID, preventing the COVID, but also for other viral diseases like RSV. Recently, in fact. Last week there was a drug which was approved. One injection instead of every monthly injection to be given to the preterm infant. And that's the revolution we are making by changing the antibody and making it as a long-acting antibody. There's also the journey which is taking antibody to be combined with the toxic products, so that what we used to hear during our college days, I actually studied when I used to hear one line about magic drug. So you take the chemotherapy and deliver smartly only to the tumor cells, rather than delivering the chemotherapy to across the body, therefore preventing all the adverse events which are related to the chemotherapy. And that's what is called as the smart way of delivering chemotherapy, and we are talking about the antibody drug conjugates. We are also talking about the product which will convert. Why we target only one target? why not have a bispecific monoclonal antibodies? Target both the targets which are responsible for disease pathology, so bispecific monoclonal antibodies. And that's we are talking about the journey which continues, particularly in this biologic therapies. Again, going back to the basics, what are biologics? These are the medicines which are grown and purified from bacteria, yeast, plant or animal cells. And of course, there are diverse group of medicines which includes vaccine also, growth factors, immune modulators, monoclonal antibodies, and of course products which are derived from human blood and plasma. And these are different from the chemical molecules. We heard about the chemical molecules, but these are very, very different. And the high level differences, of course, again for the benefit of students, I think it's very important that we know what are the differences between chemical and biologic products. The biologic products are high molecular weight. These are very complex. These are from live organisms, and therefore there is a chance that there could be some protein element which may be immunogenic. So therefore, there is a possibility of immunogenicity. Therefore, for every biologic, immunogenicity studies have to be conducted. And that's very, very paramount and very important for all the biotherapeutic products. There are many critical steps in the manufacturing. There are less easily characterized because of the nature of the proteins. There are so many tertiary and folds which happen, which may differ. The basic message is the manufacturing process makes the product. And it's a heterogeneous mixture, and it's very sensitive to storage and handling conditions. So these are the large differences between, I would say, biologic. Just a diagrammatic depiction of what we are talking about. And again, I have not shown the bispecific. It will be far more further complex. I have not shown the ADC, antibody drug conjugate. It will be far more complex than what here I have shown the basic picture. These are just few examples, and I'm sure some of you would be following some of the monoclonal antibodies in different therapy areas. But these are the molecules which are approved. Started the journey with rheumatoid arthritis, some of the monoclonal antibodies. I am sure everyone knows about Humira, which is one of the largest selling monoclonal antibody. Then it went into inflammatory bowel diseases, psoriatic arthritis, then of course bronchial asthma, and of course more importantly in cancer. And there are so many biologics which are approved for the treatment of cancer. And we are talking about they are targeting different structures, different targets, and more importantly, these targets have not only shown to be beneficial, but they have transformed the care and the outcomes for the patients. And we are talking about almost curing some of the cancers which were incurable earlier with only help of chemotherapy. And that's the potential these biologics they bring. And some of the examples I have shown, PDL1, CTLA4, I will talk about because that's also another topic which has been assigned to me. What about immunotherapy? So these are the immunotherapeutic agents which I will be talking about in the subsequent slides. Now, before we go to the immunotherapy, I think it's important for us to understand the cancer and immunity cycle. And these are the various steps. Simply, if you look at the tumor cell, the cancer cell releases the antigens. They are presented by the antigen-presenting cells, which are recognized by the T cells. And these effector T cells then infiltrate the tumor, and then they go, recognize the tumor cells, kills the tumor cells, and then again this killed tumor cells releases the antigens. So this cancer immunity cycle works on if there is a good homeostasis. If the immune system is powerful, there is an elimination of the precancerous cells. If the immune system is not that powerful, or there is a dysregulation of immune system because of various reasons, there is a possibility of equilibrium, or there is a possibility of escape where cancer progresses and then eventually the disease happens and progresses from early stage to the late stage, and it actually is harmful and disastrous for the patients. Now, coming to what are the different types of immunotherapy? There are broadly 4. Of course, there are a lot of research is happening, so therefore there are various other targets which are under investigation. But cytokines, therapeutic vaccines, adaptive T-cell transfer, and checkpoint inhibitors. I'll come to each, but very high level without going into too much of details. Cytokines are, as we all know, immune cell release cytokines. So these cytokines are nothing but are used and administered from external sources which activates the immune cells and therefore they help. It is just like antibiotics. We give when the immune system is not able to take the bacteria, you are giving antibiotics to complement that. So here the cytokines are there to complement our immune system so that you are able to tackle the cancer cells. Interleukin-2, again in renal cell cancer, is a very good example. Coming to the therapeutic vaccines, again here I will talk about Cytolucel T example, but there could be many examples of such kind of vaccines. The antigen presenting cells along with the antigen, which is of key interest, is combined and they are given again to the patient, and there are examples where they have shown to improve the survival also. Again, not very commonly used, lot of more research has to be done to see whether how transformative such kind of therapies are for the patients. Again, coming to the adaptive T-cell transfer, very interesting concept, the novel concept, and this is going to change. And we have seen a huge benefit of particularly one type of adaptive T-cell transfer, which is CAR-T. It is nothing but you are genetically modifying your T cells and reinfusing back to the patient, and you are able to activate the immunity against the cancer. And that something is, I would say, revolutionary. Some of the hematological malignancies, we have seen that the CAR-T cell has shown kind of a complete cure of the disease, which were incurable. But again, technologically very, very complex. We all need to understand and see how best, as a scientist, the research community and all pharma companies come together. And there are examples in the market. We have seen the phase 1 readout from Indian CAR-T cell through ImmunoAct. We see the IIT and the TMH coming together. Very good example of academia collaboration with the IITs. So engineering and medical is working together to come out with the CAR-T cell, and I'm sure, knowing the competencies and capabilities we have, we are definitely surely going to have the CAR-T cell products in the market. Currently it is generation 1, generation 2 and generation 3. We are currently in generation 2. Generation 3 is being worked upon particularly for the solid tumors. So there is this space, please watch out for this space. I'm sure the students who are interested in cell-based therapy, I think it's important that focus more on this CAR-T cell and the practical applications of the same. A little bit about immune checkpoint inhibitors. This is the stimulatory pathway, whereas CTLA4 and PD1, PDL1 is the inhibitory pathway. And these pathways act in conjunction. If we look at further in the diagram, now this is the stimulatory pathway and these are the inhibitory pathways. Now this inhibits the activation of stimulatory pathway and therefore your immune system is inhibited. Some of the cancers, they have the overexpression of PDL1. So if you have an overexpression of inhibitory pathway, or if inhibitory pathway is more active, your immune system is inhibited to attack against the cancer cells. And therefore, some of these monoclonal antibodies inhibit either PDL1 or PD1 or in earlier slides CTLA4. So these inhibitory pathways, if you inhibit, then you are activating the immunity against the cancer cells, and that's what the checkpoint inhibitors and the examples I have shown, they do it. They basically inhibit the inhibitory pathways so that you are activating the immune system, so that immune system is able to care of the cancer cells. And these are approved in various cancers. And I will just share one or two examples of how in the clinic, how for the patients. As we know, the most lethal cancer is the lung cancer. Highest mortality is lung cancer. In NSCLC, the 5-year survival rate was considered as only 5 to 10% if the patient is diagnosed in advanced disease. If the patient is diagnosed in stage 3 or earlier disease, it used to be 15 to 25%. Now, if you give immuno-oncology products, again immune therapy, your survival is increasing to almost 42%. You can see the 5-year survival rate. In post-IO, it has definitely increased to almost 42%. And that's what the nature, you are almost doubling the survival, doubling the number of patients survive 5 years. And that's the impact of immuno-oncology products. Let me take example of metastatic, late stage, because majority of our patients in India are diagnosed at a late stage. And even in the late stage, if you see, the survival has increased from 10% to almost 31%, again because of immuno-oncology products. So you are significantly able to enhance the survival outcomes for the patients. Now, what are the challenges? Immunotherapy works in not all patients. So there is efficacy is unpredictable. There is a difficulty to identify the marker. There is a need for more predictive markers. And that's the area of research where the entire community is working to see how best we identify the predictive markers so that we select the right patients for immuno-oncology products. Because definitely the disease is heterogeneous, there are immune-mediated adverse events, and more importantly, these are expensive products. So we need to really identify the patients very, very carefully. Now, as I said, cancer immunotherapy drugs are expensive, and most of the biologics are expensive. Why? Because these are complex. It involves high-tech manufacturing. So therefore, what is other emerging trend? The other emerging trend for biologic is the biosimilars. And if you see the biosimilars, there are 39 approved biosimilars, 22 are already launched. More importantly, if you see, there are so many authorities, worldwide authorities, like European authorities, as well as the US FDA, they have come out with a very good guidelines for approving the biosimilars. And if you see, it not only has a lot of saving for the healthcare system, but also look at the market. Huge opportunity for biosimilar market to come in the world. Even in India, there are so many such products approved. And I have, if you notice on this slide, I have not used the word biosimilar. Why I am emphasizing on that? Because although these are approved in India, at least as a researcher or as a person who knows this field, biologics and biotherapeutics, I do not have a published evidence that these are truly a biosimilar products as per the international guidelines. I do not see the PKPD studies being published, neither I see a comparative phase 3 trials being published, which are of non-inferiority or equivalence designs. And therefore, although these are approved, and that is something which I would say a message to the colleagues who are sitting here, I think this is one area. It's a huge market, but still we have to significantly elevate the levels or at least the evidence of our biosimilar products in India. Just last two slides maybe. These are the trends to watch for, and I'm sure many, many elegant speakers have spoken about these trends. I am not going to go in the details, but these are the trends which all of us need to watch for. There is some research which has already happened. We have seen the success of mRNA vaccines. We are seeing the success of mRNA-based therapeutics in the clinical trials. I am sure in the coming decade, there are so many mRNA products which are going to come. And similarly, transchromatics, then synthetic biology. I think we all spoke about it. I'm sure the August speakers would have spoken about it. But these are the top 15 biopharma trends which are going to be the trends for future. Just to conclude my talk before I actually invite for collaboration with AstraZeneca, more targeted treatments are required. We need to identify better biomarkers. We need to see which are the combination therapy which will improve the outcomes for the patients. And more importantly, we have to also look at the immunopreventive strategies to prevent cancer and its recurrence. And that's the area for all of us to come together. Now, this is just an example of our biotherapeutic portfolio. Some of them are already launched in India. Some of them are going to be filed for regulatory approvals, and they will be launched in the subsequent 1 or 2 years. But these are all available globally. They are already approved, and they are transformative and changing the outcomes for patients across the world. Now, this is one of the slides which is, again, particularly for researchers who are sitting here. More from AstraZeneca as an organization, we have 2 opportunities for collaboration. One is a Catalyst Network, and second is our open innovation. And here, these are the challenges which we solve together. One is, how do we improve the patient experiences and outcomes for early diagnosis of CKD? This is the area of interest. Severe asthma care and treatment, that's the area of interest. Early detection of lung cancer, that's the area of interest. And of course, the MDTs, the oncology patient care platforms, reducing time to diagnosis, particularly for rare diseases, and particularly for the pharmaceutics and the biologic researchers who are sitting in the audience, we have a concept called as open innovation. And I would strongly urge all of you to go through the open innovation. In open innovation, there are 4 areas where we really invite all the collaborations, because we believe AstraZeneca alone cannot change the outcome. We need to work with the researchers, and we need to advance the research together. And these are the areas where we have, so not only in preclinical research, clinical research, most importantly, idea incubators and co-solve. So currently, as we speak, we have a co-solve challenge, and we come out with the challenges at a very frequent interval. We have a co-solve challenge from a sustainability point of view. I think I heard Dr. Kamala Rai speak about how sustainability is important. So this co-solve challenge is that how can we make our R&D systems more climate friendly? So there are challenge statements. Researchers are invited to come and contribute and solve the co-solve challenges together. With that, thank you so much, and I want to really appreciate Dr. Manu for putting the slides together. Thank you so much.
Thank you, Dr. Anil Kukreja. And we would like, on behalf of the organizers, present a memento.
Now I invite Dr. Ajit Dua, who is the Chief Executive Officer of Punjab Biotechnology Incubator in Mohali. She is an expert in food quality, safety and authenticity. She holds a MSc and PhD in microbiology and food technology from Punjab Agriculture University. She is also a technical assessor for NABL, and prior to joining PBTI, she worked with Markfed Canneries as Quality Control Manager and HACCP Chief Coordinator. With this brief introduction, I invite Dr. Ajit Dua for a talk.
Thank you, Dr. Bansal. Before starting, I would like to thank the organizers for inviting me to this gathering. In fact, I was finding myself out of place because I am with food background, but Dr. Bansal said there is a category natural products which overlaps food and pharma. So this slide, I would like to explain, reason being you all are from different parts of India and you are in Punjab, Mohali, and I am from Government of Punjab side. So I would like to highlight that what Government of Punjab initiative has taken in the form of Knowledge City, which is diagonally at the back side of NIPER. National level institutes, ISER, ISB, INST, NABI, SIAB, and PBTI, they are a part of this knowledge cluster, which is full one sector has been dedicated for this, so it is around 400 acres of land. So this is a very beautiful ecosystem cluster. Why I have touched this? Because Dr. Rai was talking about ecosystem leaders requirements, so we have built up a very good ecosystem and NIPER is very much a part of this cluster. I have been given, we are short of time, so I'll be skipping few of the slides. These are the major areas we are serving: analytical services, contextual research, advisory, upskilling, and incubation services. So these are the upcoming projects in Knowledge City by PBTI: Global Innovation Hub on 4 acres of land, Knowledge and Upskilling Center, Environment Resource Center. So we are basically working in the area of food, agriculture, water, environment, and little bit in health sector. And of course in the natural products to a little extent. So I'll skip this slide. We are accredited by different agencies, so total 15 recognitions are there. This I would highlight that we have collaborated with International Atomic Energy Agency, Austria, for working on food authenticity. So last week I was there, and in fact when Dr. Bansal called me, this idea clicked me there only because we are working on food authenticity using isotopic fingerprints. So I found my project partners from all over the world. There were 22 countries. So many are working on different natural products. So I thought, why not to give this idea that, it may be new or may not be new, I do not know. But isotopic fingerprints, they are finding quite a good place in food authenticity. And I think they are finding place in authenticity of natural products also. So coming to natural products and the ingredients, we see that this market has grown a lot since last 3-4 years. During COVID time, it has really jumped because people have become more health conscious and more towards the natural products. But at the same time, there are two types of such nutraceuticals, I would say, or the health supplements. They are natural as well as synthetic. For example, we see vitamin C from citrus foods also. We see vitamin C which is chemically produced also. But these natural products, they are in high demand. But how to distinguish that whether these ingredients, they are from natural sources or they are from synthetic sources? Because we know that naturally produced things, they are a bit expensive as compared to the chemical production houses, because in chemically produced, we can produce in bulk. So unfortunately, because of demand as well as because of, the global market which is growing, the high value labeling claims, they are getting, now I would not say it is adulteration. When we replace the whole product, it is not called as adulteration. It is actually we can say fraud, or it is the authenticity issue. So now how to distinguish the natural products and the synthetic products? I know there are other techniques also, but this is one of the niche techniques which is finding application nowadays. So isotopic fingerprinting, because based on the photosynthetic pathway, based on the origin, based on the geographical location, different products, they have different isotopic signatures. So this basis can be used for distinguishing between natural and synthetic and for distinguishing the geographical origin. So major stable carbon isotopes which are being used for this purpose, they are carbon, hydrogen, oxygen, nitrogen. For example, carbon is being used for testing the authenticity fingerprinting. Hydrogen and oxygen, because they come from water and from carbon dioxide, so they tell us about the origin, geographical origin. For example, we produce here in Darjeeling tea is being produced in India. and it is being exported also, but how to confirm whether it is actually from Darjeeling or not. So that can be checked using isotopic fingerprints. Similarly, nitrogen. Nowadays, a lot of organic food, I mean, there is a great hype about the organic food, but apart from testing the pesticide residues, do we have other methods to check whether it is organically produced or not? So there is a method which uses nitrogen isotopic signatures, which tells that the fertilizers which have been used, they were from organic or they are chemically produced. So this way we can distinguish between organic food and the organically produced and the normal food. Then these are few examples which I have listed where isotopic signatures, they are finding application. Like natural versus synthetic product, then product authenticity. I have taken in next few slides only two examples. One is honey and the other is a flavor compound. Then geographical origin, as I have already told. For example, we have basmati rice from India and from Pakistan, so how to distinguish it. So high value items to distinguish the source. For example, my project partners at international level, they are working on coffee. Many people are working on coffee. Costa Rica coffee is very famous. It is a high value. Jamaican coffee is very famous. So how to distinguish? So that is based on these isotopic signatures. Then organic authenticity, source confirmation, chaptalization in wine. Wine is from fruit juices, but people are adding sugars for fermentation, which get converted into ethanol. So how to distinguish that? And in wine, this method is being used since 1990s. It is AOAC method, and now it has come for food products also. So I take the example of honey, which is finding application as a very good nutraceutical, I would say, because it has very good antibiotic properties as well as the nutrient properties. So honey adulterants, how we can test? Honey is being adulterated with foreign sugar syrups of different types. So what happens? Honey bee goes to the floral plants for collecting the nectar, for converting it into honey. And almost all the floral plants, they belong to C3 photosynthetic pathway. There are 3 photosynthetic pathways which are being followed by plants, C3, C4 and CAM pathway. So most of the floral plants, they are C3 origin, whereas all others, they are C4 origin. So honey will have the isotopic signatures of C3 plants. Here you will find that C3 plants, they have isotopic signature of -22 to -30 per mil. This is the unit of isotopic value, this is in per mil. And if corn sugar syrup, high fructose corn syrup, which is being used in pharmaceutical industry as well as in bakery industry, so that is also being used for adulteration, apart from the normal sucrose, which was very easy to detect by using the chemical methods. So if C4 sugar adulteration is there and we check the isotopic signatures of honey, the isotopic signatures will change from -22 to -30 range. It will be raised to -8 to -13. So which will indicate that it is adulterated with C4 sugar. But as we know that fraudsters, they are more smart as compared to the scientists who are working towards the regulatory side. So they came up with rice syrup. Rice is a C3 plant, so when you add rice syrup, will not be able to detect the adulteration. But again, the regulators are working towards that side. So another technique, EA-LC-IRMS came, in which we, first using the chromatography, we separate out all sugars, then isotopic signatures of individual sugars are analyzed. So likewise, I always say that there is a race between adulterer and the regulator, and which continues, and I think always the adulterer is little ahead of the regulator. So India is the first country, I would say, which has come up with the regulatory standards for honey for authenticity, because food quality, food authenticity, similarly natural products, quality and authenticity we are hearing since long, but this authenticity is a new concept which has come. So foreign oligosaccharides, C4 sugar content, delta C13 max, fructose minus glucose, protein minus honey, all these are isotopic signature values which FSSAI has come up with a regulatory framework for honey. So these are the equipment. IRMS is isotopic ratio mass spectrometer. We connect it with the front end elemental analyzer and liquid chromatography. So these are the techniques which are being used. For other purposes, GC-IRMS is being used. So another example I have taken vanillin, which is the flavoring compound. So vanillin is produced from the vanilla flowers as well as from the synthetic sources. Now how to distinguish it? In fact, for vanillin, there are two synthetic methods. One is chemically synthesized, other is biotechnologically synthesized. So when we check the isotopic ratios of these three different types of vanillin, so there was a difference in the values. So these values, like -22 per mil for natural and -37 to -36 for biotechnologically synthesized. Similarly, for chemically synthesized, it is more negative than -25. So these isotopic signatures, they tell us whether it is natural or it is synthetically produced. Some other applications are like turmeric, curcumin which is isolated from turmeric, that is finding lot of application as antibiotic. So curcumin is also synthetically synthesized as well as from natural sources. We all know that from natural sources there are other components. We get a concoction which is always better as compared to a single compound which is prepared in the laboratory. Then saw palmetto, berries are with the medicinal properties. So these are also we can distinguish using isotopic signatures. Then swelling from shark liver oil and plant sources. So we find that vitamin D is from animal sources as well as from plant sources. So there is one concern about vegan diet also. Some people they don't want from non-vegetarian sources. So this is how we can distinguish whether that oil is from veg or non-veg. Similarly, research is going on the capsule shells which are gelatin. So gelatin has come from the plant sources or from animal sources that can be distinguished using this technology. So this technology has some challenges also. So those challenges can act as the opportunities for the researchers who are present here because this is based on database. All the analysis, we have to compare it with the database of the authentic samples. So database generation is very, very important. This is an area where researchers need to work. Then technical expertise in this area is still, I don't find much technical expertise is there. In India also in food for honey, now there are 8 laboratories, but when we started, we were the first one to start. In government sector, there are still 3 laboratories only. Then regulatory framework, because of lack of database, regulatory framework is not able to come up. So FSSAI also, we have, based on the published standard, we have given the limits, but still database generation is in process. Then authentic and traceable samples. We are working on honey authenticity, so we are finding it very challenging to get the authentic samples of honey to generate the database. So I hope I could complete in time. Thank you so much. Thank you.
Thank you so much, Dr. Dua, for helping us out for a few minutes.
No, I learned that Secretary Madam has come, so I try to finish.
Thanks a lot. And let us recognize Dr. Dua's contribution.
Thanks a lot for all the speakers for their more informative talk. Now it's time to honor our chairpersons. I would like to invite Professor Sharma and Professor Dulal Panda, Director. Please come on. Give the moment, please.
Good evening, everyone. All are requested to kindly remain seated for the valedictory function.
आगे बुके रख रखे हैं अच्छे लग रहे हैं।
ठीक लग रहे हैं।
फ्रंट पे बुके रखे हैं अच्छे लग रहे हैं।
अब सामान कौन अरेंज?
अभी कर रहे हैं।
Good evening, everyone. On behalf of NIPER S.A.S. Nagar, I am pleased to welcome you all to the valedictory function of NIPER PHARMACON-2022. With this, we are marking completion of the 3-day international conference on recent trends and future opportunities in pharmaceuticals held from 10th to 12th of November 2022. NIPER is privileged to have Ms. S. Aparna, Honorable Secretary, Department of Pharmaceuticals, as the chief guest for today's function. We extend our heartiest thanks to Ms. Aparna for accepting the invitation for this ceremony and for her gracious presence. We are also pleased to have with us Dr. Manish Diwan, Head SPED BIRAC, Mr. Deepak Berry, Founder and CEO, Centis Pharma Gurugram, Professor Y.K. Gupta, President AIMS, Bhopal and Jammu. Once again, I extend a warm welcome to all the directors of NIPERS, Dean, Registrar, heads of various departments, guests from the ministry, faculties of all NIPERS and other institutes, staff members, students, all the delegates, sponsors, members of the press to this valedictory function of NIPER PHARMACON 2022. Now may I introduce Dr. Manish Diwan to you. He is the Head of Strategy Partnership and Entrepreneurship Development at Biotechnology Industry Research Assistance Council, BIRAC, Department of Biotechnology Development, Government of India. BIRAC is an interface agency which aims at strengthening and empowering the developing biotech enterprises to take on strategic research and translate innovative ideas into globally competitive commercialized products and technologies. He has worked with organizations such as Dabur Research Foundation, Ranbaxy Laboratories Limited, Daiichi Sankyo. So with this introduction, may I now invite Dr. Manish Diwan to the dais, please, to present his keynote address on biotech startup ecosystem catalyzed by BIRAC. So.
Good evening, everyone. Ma'am Secretary, pleased to be here in your presence. I see all the directors supporting the NIPER Mohali, the faculty, some of my friends, contemporaries, colleagues, students, and some startups and industry representatives. It's a very pleasant moment to be here, being a pharmacist myself, and be able to share some of the developments that are happening across the country in order for the students, faculty, and the next generation of researchers to be able to cognizant of the fact that what is there, which is happening, and how you can integrate and kind of contribute back to the development of the entire biotech sector in the country. So I'll do that with a few sets of slides. I represent BIRAC, which is an enabling institution which was specially carved out of Department of Biotechnology to support the translational research in order for the knowledge which is generated in the institutions that can move forward for product development to address the unmet need for the societal gain. So BIRAC got conceptualized in 10 years ago. From 2012, we've been supporting entrepreneurs, startups, incubation centers, industry, including small and medium scale industry as well as large industry, to help build the ecosystem. So there are few components that come into being when you move the research out of lab from ideation to, let's say, development side. So the kind of laboratories that you would find in the institutes or the industry, large industry, would have separate verticals for each of these disciplines. When you conceive an idea as an individual or a small team, you do not have all that access of that entire environment. So in order to facilitate that, there's a large capacity building which is done in terms of common access infrastructure, something you can relate to as incubation centers, which is put in place. Then funding individuals, creating such capacity building environment, and then bringing all the network of stakeholders, including the industry expertise, exposure to national and global challenges, networking, awareness, what would be the regulatory compliances requirement, so that you can actually navigate your journey from ideation to all the way to commercialization. So in the 10 years, if you look at, there's a sea change that has happened, not just because of one single agency, but it's an ensemble of activity and support from all ministries, departments, stakeholders that have come together as a nation, where we see a large number of footprint, especially in the biotech sector, that has increased in terms of number of startups, number of incubation centers, ideas moving forward, yielding products which have reached to commercialization stage, as well as, I think more importantly, collectively, the bioeconomy of the country has also seen a dramatic change. We have been able to clock more than double-digit growth year on year, and that reflects that there is a potential in this sector. And just to re-emphasize, this sector includes healthcare, which is therapeutics, as well as wellness. We saw wellness figures touching 2.3 trillion. That's the largest in terms of the volume of the industry moving. Industrial biotechnology, which can offer greener solutions, replacements of synthetic chemistry, providing lower carbon footprint, taking us to the net zero, replacement of non-renewable biofuels with, let's say, blending of ethanol, bioplastics, biofertilizers, waste to value, circular economy, and clean energy. So there's an ensemble of activities which can be integrated through the biotech ecosystem. I took longer on this slide just to give the base so that we can catch up, because all the pharmacy being an interdisciplinary sciences, and I see students from all different individual subdivisions within the pharmacy, and they can relate to many of these aspects that we just spoke about. So in the past 10 years, through the combined stakeholder efforts, we've been fortunate to see large number of movements in this direction. I'm happy to share that just like NIPER Mohali, which is hosting this 3 days event, and you saw a large number of people coming from different disciplines, different stakeholders. We had the previous session, ma'am, through the industry experts, indicating diverse opportunities, what needs to be done, where the pharmacy institute can kind of contribute. So likewise, the 3-day of this PHARMACON session, very rich, intense scientific program. I think these are the elements which make and create the outcome which is required for the enrichment of the ecosystem. Three of the NIPERs have a bioincubation center support from BIRAC. Many of the audience who are sitting here, they got benefited from some facilitation or some funding from either from DBT or BIRAC. So collectively, I think these numbers have a contribution, direct or indirect, from the audience who are sitting here as well. In the COVID times, I think as we all understand, India could stand out tall and produce, develop, as well as immunize large number of population, just because we could have 4 of us are indigenous developed or manufactured COVID vaccines which were made available to all of us. So BIREC takes pride in supporting all of them. Not only the manufacturing of these vaccines, but even the development side, like clinical trial site network that were prepared, immunogenicity testing facilities that were made available to all individual entrepreneurs to large industry, as well as the enhancement of such capacities, including the immunoassays, where these common facilities could be utilized, including the BSL-3 labs. So BIREC supports the journey of entrepreneurs from all the way from ideation to commercialization, and that is done through stage-specific facilitation. From ideation to POC level, this is largely granting aid and enabling environment. As you move forward, there are different funding schemes and different provisions that you can propel forward towards commercialization, integrating the regulators, the private equity, private investors, as well as the large industries. As I mentioned about incubation centers, so now we have a footprint of 74 incubation centers in 21 states and catering to different subspecialization within the biotech domain, including the pharma, life sciences, medical devices, diagnostics, fermentation waste, or the agri-tech space. This has actually been able to harness the talent which is from across different regions and different states. Even we see the aspirants who are willing to tread difficult path of biotech entrepreneurship. That is evident from the graph in the middle. We get to receive applications from all across the country, and you would be pleased to see that these are not limited to, let's say, only tier 1 or metro cities. More than 50% of such applications are received from tier 2, tier 3 cities, as well as we have kind of mapped about 38 aspirational districts also are part of this, and this is increasing as we move along. This reflects in the number of new biotech startups that get seeded, and we can see there is a year-on-year healthy growth in such new startups getting seeded. These are various funding provisions that we provide. Somebody was asking me in the morning, so from an enabling agency, public funding agency, there is a provision that individual entrepreneur or individual startup can get up to $1 million through the granting aid or different mechanisms. The later stages are involving more return on investments, involving provisions, very specialized provisions like fund of fund, where we integrate with the venture funds who are SEBI registered, and they are nudged to invest about 2 times what we invest in them into the biotech ecosystem, especially the startup network. Likewise, we have also created a network of high net worth individuals, angels, and VCs through the Bio Angels platform, who are specially poised to invest in the biotech startups. It's easier to say that it needs to be done, but once we get to see that people who are trying their efforts in these biotech startups are able to actually gain from their endeavors, and we get to see that in our own experience, those particular startups who got funded, they've been able to raise close to 5 to 10 times through private investments from angels and venture capitalists. So that's a very healthy sign. And this is a very small cohort, more like a pilot initiative, which was just initiated from 2017 onwards. This is what we have seen within our direct beneficiaries. We also get to see, I'll show you that slide later, maybe in the eyes of others, like once the third person evaluates whether or not that the private investor finds a value in those propositions or not, that is also evident. This particular slide emphasizes that being a Government of India enterprise, we have the ability to integrate with all ministries, departments across the public sector, as well as private sector, which includes industry, investors, regulators, philanthropic agencies, both national as well as international. So all that power or all that network is made available to individual entrepreneur or individual startup who gets integrated into the ecosystem. So to enable and empower them to compete and become globally relevant. The competence or the comparison of the solution that we've been trying to generate as an individual startup or an entrepreneur or a PI in the lab, we must keep an eye that this endeavor is relevant not only for today, but for 5 years, 10 years down the line as well, because the competitive scenario would evolve and would change. So it is important that we need to know not only what's happening in next door, but as well as outside the country too. So a conscious effort is made for a global exposure to the startups and the propositions that are getting developed, so that they become internationally relevant. We take people out, startups, for that exposure outside the country, but we also create provisions that all startup, all the ecosystem can come together and learn from each other's experience, peer-to-peer learning, knowing of the best practices, networking, collaborations. This is Biotech Startup Expo that was organized in June this year. Honorable Prime Minister Ji inaugurated it, and we are also looking forward to something similar which is forthcoming by Department of Pharmaceuticals for the medtech startups and the medtech industry, I think in December or early January. So this is a biotech e-portal showcase that has been created, which is available in public domain. It lists up to at present 750 such innovations by startups and entrepreneurs. Maybe worth a while to browse through and see what kind of innovations people are carrying forward or developing through this biotech ecosystem and their endeavors. So maybe people who are wanting to move this side of the translational research, thinking of integrating with the startup ecosystem, might want to have a look. Yeah, this slide I was wanting to emphasize that not only in the eyes of, let's say, BIRAC being a direct supportive of some of the technologies, the startups have also been able to raise follow-on funding, which is outside public money, public grants. These are from venture capitalists, both from India as well as outside India. I think we've been hearing about unicorns. Probably that's a buzzword. Maybe all of us would have heard that. There are about 107 unicorns in the country as of today. In fact, today there is also an article that the government has called Invest India as Prime Minister's own first unicorn. So I think that would make it 108 though, but 107 are there. Why I mentioned about unicorns is that among these 107, we don't have much presence from the pharma side or the healthcare side. We only have just about 4, which are more towards services sector, SaaS based or e-services basis. We need to see more number of startups reaching that scale, and with deep tech and technology-led scalable startups which can beat the current environment or current what you call as competitive landscape, because the previous presentation from the industry speakers all pointed out that there are several evolving fields, and sooner or later within next 5-10 years you would see a paradigm shift in the competitive landscape. So I think it's time we need to look forward and see what is in store, how we can create an edge and move away from the conventional comfort zones. One example, I don't know why I put it here, but this is one example which is to recognize the potential of digital health tech space and integration of AI, ML, mobile health, interlinking with the medical devices or diagnostic or the other healthcare solutions that we envisage. The relevance of this slide is that here is that let's not be complacent with achieving an understanding of one particular discipline or one narrow subject area. We also need to be able to understand and crosstalk with the other disciplines. Pharmacy is one discipline which is inherently interdisciplinary, but within the pharmacy, there is high time that we need to see how the big data, artificial intelligence, machine learning is evolving. And there are examples you can consider from, let's say, automobile industry. The conventional leaders of the industry are finding newer competitors which have evolved suddenly overnight without having the competence of or proficiency of making those diesel engines or petrol engines, but they have proficiency in designing cars which are driverless. And that is a new field which has evolved. So there's a dynamically change of the industry per se. So we also saw Nokia got wiped out because it didn't move forward as the trends moved with the market and the changing environment. When we were beginning, at the beginning of, let's say, Birect conception, we did not have such rich or present ecosystem across the country. So we wanted that at that stage we were in the kind of a nurturing stage to build that ecosystem. I think 10 years have passed and we are now seeing that we are ready to scale and grow the ecosystem to a much larger extent. Intermittently, we also we had to fast forward our target achievement of bioeconomy. The bioeconomy target for 2025 was 100 billion a few years ago. That got upsized to 150 billion bioeconomy target by 2025. So the system, you would notice that in 5-10 years down the line, there would be a paradigm shift in what we see, what kind of solutions we are using, and how the industry is evolving. Looking at the growth of the bioeconomy around the world and India's bioeconomy, which is intended to grow up, we think that both the innovation arm as well as the capacity to manufacture in the biotech side, that needs to be enhanced. From the innovation arm, I think academia, the knowledge generation, talent pool, access, which is a large representation of, which can be seen here, that is the power that India has, and that is a dependable asset. And not only our country, but across the ecosystem, across different countries, they are looking India as global innovation hub, as well as expecting to see a manufacturing capacity so that some businesses can start moving to this side. As I mentioned, we saw the movement of bioeconomy substantial increase even in the COVID days when we saw either stunted growth or negative growth in various other sectors. But biotech sector showed that there is a large potential that can contribute to discover, innovate, as well as manufacture and replace many of the things that we currently import. Particularly, for example, 70 to 80% of medical devices are being imported in India. So the healthcare delivery or the healthcare side itself is largely dependent on innovations that can converge and convert the dependence on the switch the biasness from exports to imports. I have, that was regarding the background or the know-how that I wanted to share from the BIREC side and from the ecosystem side. I'll take this opportunity to share my personal suggestions to this August gathering. Like most of us would have noticed that in the individual endeavors as a PI or as an individual in laboratories, we pursue certain projects and we try to take it forward. But those for the entire length of product conception to product development, you actually need several players. Just like within the pharmacy, you have pharmacology, you have pharmacokinetics groups, you have medicinal chemistry, you have regulatory, you have farm management, and few more, right? Now, in the individual laboratory, one person might have an excellence in the, let's say, chemistry side. So one can identify the new scaffolds. Now, the same knowledge, if gets carried forward in, let's say, in the pharmacology labs to characterize and identify the lead compounds, for instance, and the DMPK side kind of characterizes and further filters and further does the preclinical, the toxicology, safety assessment laboratory can complement that piece. So one molecule or one project has seen all the labs, and that becomes the entire institutional effort. It's easier said than done, but I would suggest that that's my personal request. If we can consider maybe 10% or 25% of this complementary combined effort to take these things forward in a more kind of continuum manner, and rest 75% of the endeavors can stay like the conventional one, the individual excellence in a project or in an individual science. The reason I am saying so, that would actually bring a lot of strength to an individual project, and you would be able to filter off quickly some which have value to move and propel forward, just like an industry does. So the attrition, there is a selection and deselection process, and those which see merit, they move forward and they get prioritized. This is something similar that you would also see when you are applying for grants. So multi-institutional collaborative projects get identified and supported much better on priority than individual endeavors. For the being a pharmacy student in past myself, so I see that pharmacy students need to study from 8:30 in the morning to 4:30, continuous back-to-back theory classes or practical classes, and we get too engrossed to see anything beyond what we are doing. The curriculums and the landscape of science is evolving. My suggestion is that if we can also keep an eye on the more recent sciences, integration of those sciences. Few we mentioned about AI, ML. The one, like most of us would not know coding, for example. I was talking to Ambassador of Sweden once, and she mentioned that every single person in Sweden knows coding. Coding is integrated right from the school curriculum. So some of those elements, if we start imbibing by choice, that would be helpful. I think some of those metabolomics, immunology, molecular biology, these techniques are quintessential, indispensable now. We can start looking at those maybe in a more conscious manner. The multidisciplinary aspect from, let's say, specialization for in vitro, in vivo, understanding of in vivo, as well as understanding of ex vivo techniques would actually complement and help you, make you more industry ready, as well as able to envisage much more than the respective individual syllabi. I also want to encourage you to interact with more number of startups, and if possible, consider doing internship and visiting some incubators. I think I overstepped my task here, but thank you so much for listening. That's all from my side. Thank you.
We have with us today Mr. Deepak Behri as the next speaker. Mr. Behri founded Centis Pharma in 1990 and is the CEO. He is also the Managing Director of Centis Research Center. May I now invite Mr. Deepak Behri to the dais for his address.
Stop it. Hold it, please. I'll tell you when to start, then you can start. Okay? Can you remove this? Yeah. Rewind it, please, so that we can start from the beginning. Yeah?
So, good afternoon, good evening, everybody, and thank you so much for this invitation from NIPER, Professor Panda, Professor Bansal, Madam Aparna, pleasure meeting you today. I want to rewind to the year 2004. That was 18 years ago. Why 2004? Because in that year I was standing on this stage, I'm not sure if it was the same auditorium or the other one, and giving the same speech, lecture, whatever you may say, to the students of NIPER. And it was very interesting because I came here to give a presentation, and I was requested by then Professor Botani, who was also the Dean of the MBA program and later became the Director of NIPER. And he told me that We have a great bunch of MBA students in NIPER. It's the first batch. Can you please meet them? And I said, look, I'm sorry, but I have a very tight program. I didn't plan for that. But he said, just maybe half an hour, 45 minutes, it will be very inspiring for them. I said, okay, fine. So I remember there were about 25 students or so who we wanted to meet. And he actually wanted me to do some kind of interviews because all of them had applied to our company to be hired, to be selected. And our HR had already done some preliminary rounds. So I said, fine, let's meet them. We met 25 students in, I think it was in 3 groups. And I was so impressed. And I called up our HR and asked them that, how many students are you planning to hire from here? And they said, well, maybe 2. I said, okay, clear. So I told Professor Bhutani, we have to hire only 2, but I will interview all of them in any case. So I interviewed them and I was so impressed that we actually hired 8. And out of those 8, 6 joined. And I'm just trying to remember, out of those 6, one lady who joined us, she worked with us for about 14 years. And the other group is all of them are doing very well, those who are in touch with us. One is a professor in US, one is a vice president in Dr. Reddy's, and they all done very well. So that is what I really like about NIPER, and the quality of the students, the quality of the professors, the quality of the education, the quality of the experiences that the students, I think now at all NIPERs, are very lucky to get. So I have been invited today to inspire the students. Many of you will be moving on to careers soon, maybe in the next year. You will be graduating, many in a year. And as you transition from the academic world to the corporate or research world or even an entrepreneurial world, life is going to be very different for you. So what is that life going to be like, and how do organizations work, how do organizations think, and how do we build careers for you? It's very important. And those of you who are graduating, and as the saying goes, that if you think that your professor or your dad was very tough, then wait till you get a boss. So life is not going to be so simple. It's going to be tough. You really will have to work very hard, continue to work very hard. You will have to continue to put in your best. And it's not easy, as they say. Somebody says, yeah, he had good luck, he got a great job. Well, luck has a part to play. But as I say, it's 99% perspiration and 1% inspiration. So hard work is required. Good luck doesn't get you very far. So please keep up this rigor that you have of submissions, of deep analysis, and this learning that you have of going deep into every matter, doing the research. The research teaches you a very good thing, which is how to be detail-oriented, how to go in depth into everything. And that is required everywhere in life. There is no going away from the attention to detail. So what now I will do is we will show you this short video. It's very short, just to give you a glimpse of our organization. And then I will try to share with you our journey of our organization and how the corporate world is different and how you can adapt to it and what will be interesting for you. So, video please. Thank you very much. So that was a glimpse into life at Centis, and many other companies would have their own panorama which they would like to show you. But what are the important things that I'd like to share with you? That as you transition, can we just have the next slide show, please? Yeah. Okay. This will work? Is this connected? I need to point that side. Okay, so like this? Okay, very good. Thank you. This works like this. Okay, clear. Yeah, thank you. So, you saw, that we were not talking so much about our sales. We were not talking, we spoke about some very important things. We spoke about patents, which many of you understand. We spoke about values and culture. We spoke about giving back to society. So why am I saying this to you? Why am I sharing this with you? Because it's very important to understand that in today's world, what is very important is values, ethics, culture, transparency. These are very important qualities. In today's world, compliance is a very critical thing. Because in the pharma industry, in biotech, in whatever field you are, in healthcare, you know that all pharmaceutical companies, all biotech companies are trying to get on the international stage. And to be on the international stage, it's very important to be compliant with international regulatory requirements, with international statutory requirements, with international financial requirements. So, compliance also comes from quality, and quality of products and quality of everything you do. So, companies are built on pillars, on foundation. Just like a building has its own pillars, its foundation, companies also has its pillars. This is the pillars of Centis. We call it our 3 pillars: our quality, policy, our values, and our cultural anchors. Many of them will resonate with you also. And it's very important for the company to be built on this solid foundation. Remember, as you move ahead in life, what is going to hold you, what is going to be good for you is to have your own image. And your image comes from how you project yourself, how you build your values, how you project those values, and how much you live by those values. So, as you can see, we have discipline and compliance shown here, which I was talking about compliance. Compliance comes with discipline, and which is why quality-centric organizations like ours prefer quality-driven institutes like yours, right? We believe in quality. Quality is very necessary, and that's what you are all taught, and that's what the excellent education which you receive brings you to. And one important thing to learn is that quality should have one standard. You see, in the olden days, we had that, okay, this is a quality which was for the Indian market, which was of a lower standard, and this is export quality, which is for a higher standard. So when we set up our company, when we set up our own plant, we said one quality, one standard throughout the world. Now you all saw this very nice presentation before mine by Dr. Manish, which was showing some fantastic funding for startups and biotechs. Yes, amazing. I must really admire the government of India for taking such initiatives. Really amazing. I saw the number, $150 billion. Wow, that's really super. I'm going to rewind now to the year 2006, when a young man, me, went to the bank to ask for money to set up an R&D center and the manufacturing plant. in those days, there were no incubators, there was no startup funding, there was nothing. There was a bank, there were no private banks. Yes, it was PSUs. And it was very difficult to convince the banks to give money. Now, when I went to the bank and I gave our project report, the bank manager told me, read through the report, and he told me, you have some, either you are either crazy or you have some fantastic strategy which I don't understand. Because you are at I remember we had sales of about 50 crores and we were asking for a loan of 50 crores. So he says, you have 50 crores of sales and you want me to fund you 50 crores? I don't understand why. So he said, are you crazy or you have some special strategy? I said, both. I said, a little bit of both. And it's very important to be crazily passionate about what you're doing. So that's the message I want to give to all of you also, that be crazily passionate about what you have in life, what you want to do in life, whatever it may be. Be crazily passionate about your studies, about your education, about your work, about your hobbies, whatever you'd like. This is a very important mantra in life, and I'm probably going to stop here because we're running out of time. This is a very critical mantra which we follow, and I'd like to share with you, that you must plan, then do, then check, then act. And it's a cycle. I call it the circle of life. This is not my formula. This is a very standard quality formula. But I just want to share with all of you that if you are able to keep to this and plan, then execute, then check and monitor and track, then act and correct whatever you have to do, and then follow the same circle of life again. So, due to paucity of time, I will stop now, and I will leave you with some messages, very quick message. This is our philosophy. Since you are all brimming with ideas, that no idea is a bad idea. That's our philosophy in our company, that no idea is a bad idea. Come up with any idea. And no excuse is a good excuse, right? So don't give excuses for anything that you have to do. Okay, so this is my message which I said earlier to you also. Be crazily passionate about whatever you want to do, and I'm confident you will get success. Thank you very much.
Thank you, sir. May I now request Professor Dulal Panda, Director NIPER S.A.S. Nagar, to escort today's chief guest, Ms. S. Aparna, Mr. Deepak Behri, Professor Y.K. Gupta to the dais, please. I now invite Ms. Anandita to bring the bouquet to be presented to Ms. S. Aparna by Professor Dulal Panda. I now invite Ms. Astha to bring the bouquet to be presented to Mr. Deepak Behri by Professor Dulal Panda. I now invite Ms. Abhyanta to bring the bouquet to be presented to Mr. Deepak Behri by Professor Dulal Panda. I now invite Ms. Devanjana to present May I now request Professor Dulal Panda to address the gathering.
Good evening, Madam S. Aparna, Secretary, Department of Pharmaceuticals, Professor Y.K. Gupta, President, AIMS, Bhopal and Jammu, Dr. Manish Dian, Head, Speed Birac, Mr. Deepak Bahari, Founder, CEO, Centis Pharma, my fellow directors of all NIPERs, eminent speakers, my colleagues in faculty and staff of NIPER Mohali and other 6 NIPERs, ladies and gentlemen, it is my great pleasure to welcome you all in the valedictory function of the International Conference on Recent Trends and Future Opportunities in Pharmaceutical, NIPER Pharmacon 2022. I am extremely happy with the quality of lectures delivered during the 12 technical sessions spread over the last 3 days. Not only the lecture by the senior faculty and scientists from industry and academia, but I was pleasantly surprised and indeed quite excited by the level of scientific competence visible in oral presentation delivered by young researchers, including assistant professors, postdoctoral fellows, and PhD scholars. Approximately 400 posters were presented during the conference and 42 oral presentations were made. Several of them showed a high level of scientific expertise. It gives me hope and confidence that our younger generation has the innate capability required to carry out scientific research, and conferences like NIPER Pharmacon 2022 will provide them with the platform to convert this genotype into phenotype. Today's science is tomorrow's technology. Like I had noted in my remarks during the inauguration of the conference, we have been inspired by the words of Honorable Union Minister of Chemical and Fertilizer, Dr. Mansukh Mandaviyaji, during the NIPER week, organized as a part of the Azadi Ka Amrit Mahotsav celebration at the Institute last October, where he had asked the NIPER community and the pharma industry to draw up an action plan for the pharmaceutical sector for India 2047. We are grateful to Madam Aparna for her guidance in drafting the program of the conference. Her keen interest and the support of the Department of Pharmaceuticals have been instrumental in the conference reaching its goal successfully. During these 3 days, we have heard from speakers from academia and industry on their research experience. The presentations were interactive in the true sense of the word. I am sure that constructive feedback received from audience will provide several thinking points when the speakers go back to the bench. I am thankful to the speaker who participated in the several panel discussions. This informal setting allowed them to articulate their viewpoints on the necessity of basic and applied research going hand to hand, and provided a platform for the members of the audience to interact with them on some important issues in developing bench-to-bedside technology. Madam Aparna had specially emphasized on this academia-industry platform to strengthen during this conference, and this has paid rich dividend in terms of the need for standard operation procedure to be designed while carrying out research in institutes like NIPERs. The participation of startups and the presentation have highlighted this space has a lot of potential to deliver the goods once the initial teething troubles are taken care of. With the cooperation of academia, industry and startup, steps towards the establishment of Atmanirbhar Bharat in the pharmaceutical sector and the development of affordable health care will be a reality. I am thankful to the National Advisory Committee, particularly Dr. Sashibala Singh, Dr. U.S.N. Murthy, and Dr. V. Ravichandran for their continuous help and supports, and each and every member of the local organization committee who has played their part in ensuring the success of the meeting. The generous support and active participation of our students and volunteers has made the job of the organizing committee a bit easier. I hope that all the delegates have found the scientific deliberation fruitful and wish you all an enriching scientific journey and a safe trip back. Jai Hind.
Thank you, sir. May I now introduce Professor Y.K. Gupta to this august gathering. Professor Gupta is the president of AIIMS, Bhopal and Jammu. He is a distinguished and renowned expert in neuropharmacology, clinical pharmacology, and drug development, toxicology, and regulatory pharmacology. He is a fellow of National Academy of Medical Sciences, fellow of National Academy of Science, fellow of Indian Pharmacological Society, fellow of Indian Academy of Neurosciences, and fellow of Society of Toxicology. So he was a former director of Indian Institute of Toxicological Research, Lucknow. With this introduction, may I now invite Professor Y.K. Gupta to address the gathering, please.
Adarniya Madam Aparna ji, Secretary DOP, Dr. Dolal Pandey ji, Panda Sahab, Director NIPER Mohali, and I can see my friends, Dr. Murthy, and I can see Dr. Ravi Chandran, Dr. Shashi, and this is the galaxy of leaders of NIPERs. And when I see ये जो गुलाब का और different फूलों का गुलदस्ता है, this represents the unity of different NIPERs here, and this also represents BIREC, Dr. Manish. This represents the international faculty, Dr. Gulati. And I am proud of my students here, Dr. Shyam and Dr. Kamla and a couple of others, ladies and gentlemen here. Why I say that this guldashta, importance of this is a collaborative approach. A collaborative approach which results into not only giving flowers a shape, but is also productive. It smells good. The fragrance goes very far, and that is what the impact of joint effort of NIPER is. And we are proud of that Indian pharma industry today has outreached, I think, the maximum number of the countries in the world which any other country has not able to reach. It has shown that not because of the Hathi Committee was the beginning, when you talk of just post-independence Hathi Committee, the primary objective of Hathi Committee was to have a self-reliance on pharmaceutical sector. And today, we can say with pride that not only on generic medicine, but also in producing the human resource in pharma, we are number one now. And we have to reduce this lag period of clapping on our success. This should come instantly. And you should realize, you should feel proud of your success. I think that is what we are lacking here. Now I will give you some challenges to you. It is always good to pat on the back because that always motivates you to go forward. But it's always good to walk with care so that you do not meet an accident, which happens. And the challenges today are as follows. If you look for human resource today, I will agree with my previous speaker that he came with the expectation that he will select 2, and they ended up in select 8 out of 20 or maybe out of 13 or 14, but I will be more worried why rest were not selected. So was this the lack of job opportunity? He should work on it. Or was this lack of competence? You should work on it. Or the faculty should work on it. Was this that the 18 were looking for job? Why not all of them were looking for providing opportunity for job? Why not? They all thought that I will provide opportunity for job to you. So I will take over your company sometime. That should be the motivation you have. That should be the motivation of all NIPER graduates, at least the valedictory function, those who are attending the valedictory function should have. There are a few people who attend conferences. Always say, I have been attending conferences since myself, Professor Gulati, have been attending for 1970s. क्या होता है वैलिडिटी फंक्शन में आधे से भी कम रह जाती है, इसमें तो बहुत है. And maybe this is the charm of our secretary here. कुछ तो सेक्रेटरी की प्रेजेंस के लिए डायरेक्टर ने जोर दिया कि आ जाओ भाई. लेकिन ऐसा नहीं है. यहाँ पे मोटिवेटेड लोग हैं. So what I'm trying to say, valedictory function में you look for what happened in last 3 days. And I would say, Professor Dalal Panda mentioned कि हमारे इतने lectures हुए, इतने oral presentation हुए. I would say, what is the success rate of lectures? And there are different parameters of success. बड़ा अच्छा था lecture. जोश में बोले, हमने होश में सुना. लेकिन असली सक्सेस लेक्चर का वो है जिसने लेक्चर लिया उसको खाना मत खाने दो, घेर लो उसको सवालों से. तो कितने लेक्चर्स ऐसे थे जिनके लेक्चर के बाद में लोगों ने उनसे सवाल करने के लिए घेर लिया, उनको खाना नहीं खाने दिया, डिनर नहीं करने दिया, और डिनर करके चले गए तो मेल की भरमार कर दी कि अब जवाब दो इस सवाल का, मुझे गाइड करो. आई थिंक दैट टाइप ऑफ कल्चर हैज टू कम अप. We have to look for not only listening lectures, but interacting with them. You will never get an opportunity to interact with so many luminaries at one stage, so many directors, so many industry people. And I would say, take this opportunity because Madam is here. I was listening to a couple of lectures just now, few. हमारी इंडस्ट्री में इतना टैलेंट भरा हुआ है, लेकिन वो टैलेंट क्यों छिपा रहता है? Why not these industry experts are offered a visiting professor, adjunct faculty to these NIPERs? I've been talking this number of times. Maybe it may not require financial requirement. Maybe all those will try to repay what they have earned it. And I'm not so comfortable when we say कि यहाँ से जाने के बाद इंस्टीट्यूट से निकलने के बाद जब आप असली ज़िंदगी में आते हैं इंडस्ट्री में बड़ी मेहनत करनी पड़ती है, आपको बिल्कुल डिफरेंट लाइफ मिल जाती है. नो वे. You have to be taught in a manner that you transit when you were transition from academia to industry to regulator to a professional job. It should be seamless transition. It should not be झटका लगे आपको gear में. It should be automatic gear. And that can only happen when your curriculum, when your training is mixed with your future requirement. And that's what, as a chair of Industry Academia Partnership, when we did last year, seamless, right from the beginning हो कि आपका जो आप प्रोजेक्ट ले रहे हैं, जो आप थीसिस दे रहे हैं, वो नहीं हो जो आपका गाइड पीडीएफ बन के करके आया यहाँ पे. क्या होता है? हमारा पीडीएफ करके आया, गाइड हमने सेलेक्ट किया, उसने कहा भैया मैं तो यहाँ 10 साल तक पीडीएफ में ये सीख के आया, अब तुम भी इसी में लगे रहो. क्यों लगे रहो भाई? It's good if you are trying to develop a technique, infrastructure which is contemporary need of the country. But not just because मैं सीख के आया तो तुम भी यही करो. मैं कभी सास थी तो तुम भी वही करो जो सास ने किया. नहीं, जमाना बदलता है. Now therefore you have to look for what is the need of the country, what is the need of the industry, what is the need of the today's disease process. And I will just again say one thing to Madam here. What we lack here, one flow, one stream is lacking in this, is the medical stream. The pharma is working for whom? Pharma sector is ultimately working for industry, is working for patients. Is an industry working for maybe drugs for livestock or maybe. लेकिन जब हम पेशेंट्स की बात करते हैं तो डॉक्टर से हमको पूछना चाहिए तो तुमको तकलीफ क्या है, तुम मरीज की तकलीफ तो देख रहे हो, तुमको तकलीफ क्या है दवाइयों में. And that should be the filtering process of your research projects, of your guidance. ये है, but I think that need to be increased. I have not seen in any NIPER, any medical faculty teaching, coming and talking about. And this can be increased perhaps, the interaction can go up. When we say, madam, हमारे यहाँ farm D पैदा हो रहे हैं, हमारे farm पैदा हो रहे हैं, and then we need in medical institutions the people to work for drug analysis, quality control, pharmacovigilance, material vigilance, and so many other things. Why not? Right from the beginning, there is an interaction between medical institutions. There are 20 AIMS now. There are so many NIPERs now. Why they cannot be more interactive things right from the beginning to identify the diseases which we are looking for new drugs, maybe rare disease, maybe other disease conditions. So that can also be the to inflow from industry. What industry is looking for, that should be the project here. What medical science is looking for, that should be project here. What you think that you can give it to them, new idea, that should be the project to them. So that is how it should work. Startup, I greatly appreciate Dr. Dulal Pandaji and Bairak here, Manish here, that startup, कोई start करके छोड़ मत दो. Ignite करने के बाद में उसको थोड़े दिन तक fuel देते रहो. कि ऐसा नहीं हो कि आपने स्टार्ट करके धक्का मारा, वो जाके वहाँ पे खड़े हो गए. कितने स्टार्टअप जंक यार्ड में खड़े हो गए. The important thing is follow up these startups, because the enthusiasm, energy which you have now will have to be sustained till it becomes in a self mode. I think that is what we need to do it. Finally, I would say few things. That this journey which you are looking in this temple of, say, learning, of science, and I would say this is not learning of science, this is learning for humanity. Because ultimately you are producing some products which will be useful. And there should not be delay or the lag period when you produce a product and it will go to the public. And that has to cross what you say, a compulsory, mandatory regulatory system. And I would be very happy if the product of this institute or these people who are sitting here, they create an ecosystem where each and everybody contributes to development of the regulatory science in the country. Regulatory science has to be very dynamic. ये नहीं है कि हमारा regulatory science 1942 में बना और उसके बाद हम उसी को कोट करते चले आ रहे हैं. There have to be amendments. Every day there has to be amendment, and you have to think why and why not. And then you should make a critical idea. You have to make a critical logic and evidence, give it and make a regulatory science as a very important thing. Because otherwise product हमने बनाया और उसके बाद में वो वहीं पे अटक गया. 6 साल लग गए प्रोडक्ट बनाने में, दुनिया उसको आगे लेके चली गई. हम सोच रहे हैं अब तो now this is now time to make it withdraw, time to make it obsolete, and now you are putting it into the market. You have lost that critical time. So that has to be very important thing. Many times you might have seen that we are caught in a very odd situation. I'm not giving the recent examples or maybe where just because of some impurity, some toxicant or something, or some adulterant, whether accidental or whether it is intentional, whether punishable or whatever it is, you are painted differently. The hard-earned reputation of the country gets spots. Never allow these spots to be there. Have a precautionary thing. Why not? Can we say that nitrosamine का भी lecture लिया या maybe any XYZ things हैं, can we say that what should be the palatable, what should be the acceptable limit of detection? What should be the capacity in country for these 10 critical issues? And what should be the acceptable limit, not depending on what US or what US FDA says, but what is our database, what is our science says. And success is Pharmacovigilance Program of India. And I must say that there was a time when for looking for the safety of drugs, for deciding drug in national list of essential medicine, which I chaired, we used to look for the data from abroad. But today we look for the data first for the safety from Pharmacovigilance Program of India. That becomes the first and foremost criteria of deciding our safety criteria for our country. And that's why the regulatory system should also be for our country. And that's what can come only for critical thinking, out-of-box thinking. So my last message is develop some out-of-box thinking, develop some critical thinking, chase your teachers till he says, I give up, and I will come back tomorrow. So I will come back tomorrow to you. Thank you very much.
Thank you, sir. May I now introduce our chief guest, Ms. S. Aparna. Ms. Aparna is currently Secretary to the Government of India in the Department of Pharmaceuticals. She is a member of the Indian Administrative Service for the past 3 decades. She has a wide range of experience in the national, state, and local governments. After her initial years in the district administration, her principal areas of experience have been public finance, development policy and planning, and urban governance. Ms. Aparna has served on the board of directors of over 15 public sector companies operating in energy, industrial infrastructure, and financial services. She served as the executive director on the board of the World Bank Group, representing Bangladesh, Bhutan, India, and Sri Lanka for 3 years from 2017 to 2020. Madam holds a postgraduate degree in zoology and MPhil in energy and environment from India and a master's in business administration from Melbourne, Australia. Her priorities for the pharma medical device sector is to empower the Indian industry to meet its potential for global leadership through innovation. She focuses on global competitiveness, assured quality, affordability, increased drug security, resilience, and contribution to SDG 3 through a patient-centric approach. With this introduction, may I now invite Madam to address this August gathering, please.
Good evening, namaste, to all the distinguished colleagues here, as well as all our students and researchers who are participating in this conference. I'm very happy to be here, at least in the valedictory session. I had planned to be here on the first day, but there were other engagements, but I'm happy that I'm here today. And I would like to start with my very sincere appreciation for the efforts that have gone into organizing the conference, the first of its scale and kind by NIPERs, and particularly would like to thank the entire organizing committee, the moral and actual support of all the directors, to Director Dr. Panda in S.A.S. Nagar, and the efforts of all members, not only of the organizing committee, but also those who have participated in this conference. It is extremely heartening to note that we had both Indian academia, Indian industry, international academia, as well as presentations by students, faculty, researchers of more than 400 papers that had been offered up, and the proceedings that have been brought out is a testimony to the effort that must have gone into collecting all this work into one platform. So my sincere appreciation for the activities and the efforts that have seen culmination in the last 3 days. So yes, I think we should clap everybody. So very, very sincere appreciation to all of you here. As has been mentioned by many of the speakers and particularly Professor Gupta ji, government has been providing policy framework and programmatic intervention for the pharma sector because as all of you do appreciate, the pharma sector is of course a very important manufacturing sector. We have a size of close to $50 billion now. There is an export of about $24 billion, of which there is a net foreign exchange earning of about $17 to $18 billion from the pharmaceutical sector itself. So yes, it has a very important economic role to play in the development of the country. But the government's policy and programmatic support, including the support to NIPERs and to events such as this, also stems from the fact that this is one of those manufacturing sectors or industry which also has a societal impact very directly. And that societal impact is our ability as a sector to provide access to affordable medicines of good quality to our citizens throughout the country, of course, but also across the globe. All of you would, of course, be aware, I am sure, that India is now recognized as the pharmacy of the world. It is the third largest producer and market in terms of volume and possibly the largest in terms of generic drugs, where about 20 to 22% of the entire global generic market comes from India. And of course, I think you must have heard our Honorable Minister also say at various forums that 1 out of 4 generic medicines or pills in the US, which is a large market of over $430 billion. So 1 in 4 pills, generic pills, are from India. In the UK, I believe that is 1 in 3. And this has been achieved through a long process of building up the industry and ensuring that affordability and access and quality go hand in hand. So this is a sector which you should be proud to be a part of, that it has not only an economic value for the country, but also a societal value. And I think that in order to maintain that kind of a level, both at the national and at the global level, everybody here, whether they are industry leaders or they are academicians or they are thought leaders or they are young students who are embarking on a career in the pharmaceutical sector, I think we need to be also aware that new challenges are emerging. There are lots of evolving trends that we are seeing, and possibly nothing could have brought this evolution or these trends to our notice, to the notice of the popular mind as dramatically as the COVID pandemic did. So for 2.5 years, the world in different countries at different times came almost to a standstill. There was huge economic loss. There was loss of life. We still don't have a therapy for COVID. We had to undergo huge vaccination drive. Huge financial human resources had to be diverted from other parts of the economy to maintaining life and ensuring the support to the poorer sections of society who were, impacted and had to suffer hardship because of one disease. So I think that was something that brought home to everybody at every level, whether it is the highest policy makers or the poorest family in a society, the importance of being able to meet these challenges. And for Indian pharmaceutical sector, it is again important that we should not rest on our laurels. We should not say, oh, we are now pharmacy of the world. We don't have to do anything else. Because all of you would be aware, since you are thinking people, you are intelligent students of the sector, that there are so many new trends that are coming in. Even what this curriculum you are studying needs to be updated almost on an annual basis, I feel. because we have seen the transition from the traditional chemical-based pharmaceuticals to the biologics and biosimilars. We are seeing more emphasis on protein peptide-based therapies, stem cell therapies, genetic manipulation, and there is a new appetite for natural products like the Ayush products, which I would classify as maybe natural products or phytopharmaceuticals. And we are also now, in fact, wanting to explore marine therapeutics as a new area for development in our country, and the NIPER at Kolkata will be coming up with a center of excellence for this. So there are emerging trends that all of us have to be aware of, even as we celebrate the strength of Indian pharmaceutical sector, of which all of you are a part. In order to be able to address these challenges, be able to respond to these trends, and ensure that we retain our global leadership, more and more now, innovation is becoming essential, innovation based on research. So it is not sufficient to be able to expand our generic capacities and competencies, but simultaneously, we should be investing in innovation. And the reason why we have in the department, all of us collectively with all the directors of NIPERs, taken a decision that we need to ramp up the research activity in all the NIPERs, bring together all players to ensure that innovation becomes the hallmark of Indian pharmaceutical industry and pharmaceutical sector. So while generics have a great importance in ensuring affordability and manufacture at scale, innovation is also going to be equally important, in fact critical, in the years and decades to come. And that is why I do believe that focusing our research efforts, and more importantly, again I would refer to Professor Gupta ji, collaborating our research efforts is going to be critical. What cannot be done by individuals can be done by a team that we can always see. And our team is spread not only in 7 NIPERs, but our team is there in DBT, our team is there in CSIR, our team is there in ICMR, our team is there in the IITs, our team is there in AIMS, our team is there in the industry. In the industry, we have innovators in startups, we have the large Indian pharmaceutical industries, we have the OPPI, which is foreign MNCs who are working in India. So there is a huge team that has to be brought together in a collaborative spirit. If as a country we are able, we will be able to transform our pharmaceutical sector into a innovation-based sector. And I think that is necessary to meet both our strategic objectives and our societal goals. So strategic objectives is to capture value, is to go up on the value chain, gain global leadership in emerging areas, continue to provide employments, and societal goal is to ensure that our patients have the benefit of the most advanced therapies, most advanced products at rates that are affordable in our country. So these are the 2 objectives, both the economic strategic objective as well as the societal goals that we need to work on together in a collaborative effort through research and innovation. So priority areas is something that we have been discussing among the NIPERs and within the department, because obviously, every individual researcher or faculty member would have areas of interest, and the nature of research is to go deeper and deeper and deeper into one problem that consumes you, that crazy passion that Deepak ji talked about. But it is also important to keep national priorities in mind when we talk about doing research in a collaborative way across public sector institutes and with industry. And if I can just try to list some of these priorities, because there are students here who would be pursuing research, there are researchers who would be, looking for a career for themselves. First, I would say our priority is drug security. So we have to ensure that we have the self-reliance in manufacture of APIs, bulk drugs, in some of the advanced therapies where we still import a lot. So drug security in terms of pharmaceuticals is one priority. I see Dr. Murthy looking at his watch, so I'm going to hurry up. So second one would be addressing neglected diseases, including neglected tropical diseases, and also emerging issues like AMR. Many of you would have seen reports of how antibiotic usage in India is on the rise, so that would result in AMR in the coming years. Third, I would say, is evolving opportunities in the form of biologics, gene and cell therapies, phytopharmaceuticals, natural products. Fourth is again something that NIPER should be encouraging students to look at, and that is advanced manufacturing techniques, including CFT, including the use of robotics in manufacture, because when that comes in a big way, India will lose its low-cost advantage. So we need to be working on that. And the final priority that I can list today, I'm sure there are many, is the integration of digital tools, whether we are talking about data analytics, AI, ML, in the entire cycle of drug discovery and development, clinical trials, clinical management, and pharmacovigilance, that is post-marketing vigilance. I think these are some of the areas that we do expect will be important in pharmaceutical research in the years to come. And this is what I would like to share, particularly with researchers and students here. When we come to the NIPERs, we have been encouraging the NIPERs, as I said, to increase their research output, focus their efforts. And to that, I must say that NIPERs have responded. They have come out with a common research program which themselves they have created, focusing on 4 areas and about 16 APIs, if I remember correctly. And we have also, as a department, tried to support with equipment and support to events such as this. The role of NIPERs in providing skilled manpower and research resources to the pharmaceutical sector cannot be overstated. For a while, we have not had board of governors. So as secretary of the department, I have been signing all the degrees and doctorate degrees that have been awarded for the last year or so. So I have had the opportunity to see the GPA of the students here in NIPER. So I must say that yes, it is very encouraging. I rarely see a GPA less than 7. So it is very encouraging, yes, of course. And most of the GPAs of over 9.4 are from the girls. So that is also very, very encouraging. But I must just sort of caution our students, our youth here, that it is necessary to keep updating your knowledge. You know, you cannot become an industry leader if you only depend on textbooks and lectures and laboratory work. There is a need to, what we used to call, reading around the subject. Go out into the information arena that is available and try to update your knowledge, because what the employer is looking for is not your GPA of 8.5 or 9.4. That is not what the employer is looking for. Employer is looking for your strong fundamentals. Employer is looking for your problem-solving skills. Employee is looking for your ability to identify opportunities, apply innovation, think ahead of the curve. So these are also areas where researchers should apply themselves if they are really to contribute and not merely become another cog in the machine. If we are to make a difference as NIPER, which we are have designated as institutes of national importance, on par with other institutes of national importance like IITs, IIMs, AIMS, we have designated NIPERs as the institutes of national importance. To live up to that designation, I would urge the student community here to continually try to improve and update themselves through their own efforts so they can add value. Recently, we have also issued the guidelines for innovation and entrepreneurship, and I hope that that will encourage faculty also in NIPER to tap their entrepreneurial skills, their entrepreneurial abilities, and become closer to the industry in sense of being able to respond to problems and issues and come up with good solutions. This international conference that has been organized is one effort to bring about collaboration, to bring attention to these priority areas that have been identified, to encourage researchers, faculty, students to become more active in terms of research output and research effort. The reason why we have started this in NIPER S.A.S. Nagar is because it is the eldest child among the 7 NIPERs, but this is going to grow into an annual, if not biannual event, with each NIPER taking its turn every year. And I would encourage that NIPER should work and behave like a family in terms of taking this forward. I also encourage active collaboration across departments. We already are seeing that with Bairak here, with Manish ji here, who has been very generous in sharing resources, thinking, and information with both DOP, that is Department of Pharmaceuticals, and NIPERS. I must thank you for that generosity. And also we have had speakers from DST, CSIR, IITs, as I mentioned. I would also like to welcome and appreciate the participation of industry colleagues here. We have members from all industry segments, all industry associations here. The idea is that industry-academia linkage should not remain a slogan or an empty motto. It should translate into projects, and it should be based on mutual respect and complementarity of skill sets and efforts. So I do look forward to more participation from the industry. I would like to end by saying that I wish that NIPER PHARMACON, as the name has been given, christened, will grow into an international event which will be a very prestigious event for India. These things start from small beginnings, but they have the potential to grow into a very, very prestigious and impactful initiative. All of you will remember, maybe 5 years, 10 years down the line, that you were there at the first NIPER PHARMACON, but each one of us has to contribute to ensuring that this initiative is a success and it is aligned with the strategic goals and the societal goals that I have been mentioning several times in my talks with you. So again, I would end with thanking all of you for your participation, congratulating the organizing committee with the leadership of Dr. Panda, handing over the baton of the next NIPER PHARMACON 2022 to whichever NIPER is ready to take it up in 2023, and welcoming and encouraging industry, all other departments to continue to be a part of this initiative. Let us treat it as a common public good that we are creating. Thank you so much.
Thank you, Madam. Now we move on to the prize distribution ceremony, and it is time to declare the winners of the oral and poster presentations which were held during the 3-day conference. So may I now invite our chief guest, Ms. Aparna, to distribute prizes to the winners of the oral presentation sessions. So for the drug discovery for cancer chemotherapeutics, the winner for oral presentation is Vishal Rajdev, NIPER Guwahati. I invite Madam to distribute prizes to the winners of the oral presentation. For emerging opportunities in advanced drug delivery systems, the winner is Pooja Todke from IICT Mumbai. Drug discovery for antimicrobial resistance. The winner is Rushali Raka from IMTech. Immunotherapy, biological pharmacogenomics, and precision medicine. The winner is Sandeep from NIPER Mohali. Development of key intermediates and APIs. The winner is Indraneel Chatterjee from IIT Ropar. Phytopharmaceuticals and nutraceuticals. Winner is Ruchika Maurya from Nabi. Therapeutic. Therapeutic approaches for central nervous system diseases. Winner is Bhupesh Vaidya from NIPER, S.A.S. Nagar. Thank you, ma'am. May I now invite, yes. May I now invite Professor Y.K. Gupta to distribute prizes to the winners of poster presentation, please. Yes, it is this way. Huh? I'll call one by one or so may I? So the winners of the poster presentations, P1, Pharmacoinformatics. The first winner of Pharmacoinformatics session is Anish Gomatam from Bombay College of Pharmacy. Anish Gomatam from Department of Pharmaceutical Chemistry, Bombay College of Pharmacy is the first winner of the poster presentation. Second winner is Harish Kumar from Department of Pharmacoinformatics, NIPER, Mohali. Third winner in the poster session, in P1 session, is Sonia Kumari from NIPER, Mohali. Now moving on to the P2 session winners, medicinal chemistry and process chemistry. The first winner is Upma Gulati, NIPER, Mohali. Upma Gulati from NIPER, Mohali. Second position is won by Pankaj Gupta, again from NIPER, Mohali. Third position is of Arnab Chaudhary from IIT Ropar. Arnab Chaudhary, IIT Ropar. I think he is not present in this session. So moving on to the P3 session, natural products and traditional medicine. The winner is, first position is Ravi Adi Narayan Somatini, NIPER, Kolkata. He is from NIPER, Kolkata. Second position is Fahima Narjesh from NIPER's S.A.S. Nagar. Fahima Narjesh from NIPER S.A.S. Nagar. Third position is taken by Amardeep Singh from NIPER Ahmedabad. Amardeep Singh from NIPER Ahmedabad. I think he's not present for this session. So moving on to the P4 session, pharmaceutical analysis. The first winner is Aditya Sharma from NIPER Guwahati. NIPER Guwahati. Aditya Sharma from NIPER Guwahati. NIPER Guwahati. Aditya Sharma from NIPER Guwahati. Second position is won by Gigal Shah from NIPER Ahmedabad. Gigal Shah from NIPER Ahmedabad. Moving on to the third winner is Rupali Mahajan from NIPER Hyderabad. For the P5 and P6 session, pharmacology and toxicology, the first winner is Anuradha Kesharwani from NIPER Hajipur. Lokesh Sharan from NIPER Kolkata is second in position. Third position is taken up by Mohammad Rihan from NIPER S.A.S. Nagar. Mohammad Rihan from NIPER S.A.S. Nagar. Yes, sir? Yeah. Now moving on to the P7 session, pharmaceutics. The winner is first position, Sheetal Yadav from NIPER Mohali. Second position, Reena, NIPER Mohali. Reena, NIPER Mohali. She is not present, it seems. So moving on to the third position, Bharati from NIPER Ahmedabad. Now moving on to the P8 session, Biotechnology. The first prize goes to Pradyut Kumar Roy, NIPER Mohali. Okay. Second position is Krishna Raja Muthuraman from Shizuoka University. Krishna Raja Muthuraman from Shizuoka University. Third position, Sahil Verma from NIPER Mohali. Now moving on to the P9, P10 session, Pharmacy Practice and Clinical Research. The first position is taken up by Amol Bisain, CDRI Lucknow. Amol Bisain, CDRI Lucknow. He is not present. Second position is taken up by Saeed Kashif Ali from NIPER Guwahati. Third position, Nadela Monica from NIPER Guwahati. Now moving on to the last session, which is P11 medical devices. The first position is taken up by Pulugu Priyanka from NIPER Ahmedabad. Pulugu Priyanka from NIPER Ahmedabad. जी मैम. Second prize goes to Indra Memdi Chorus from Shizuoka University. Third and last prize is for this session is Monica Chopa Dandi from NIPER Ahmedabad. Monica from NIPER Ahmedabad. With this, we have come to the end of the prize distribution ceremony, and now may I request the dignitaries to take their seat, please. May I now request Professor Panda to present a memento to the chief guest, Ms. S. Aparna. Thank you, sir. May I now request Professor Panda to present a memento to Mr. Deepak Behri. Thank you, sir. May I now request Professor Panda to present a memento to Professor Y.K. Gupta. May I now request Professor Panda to present a memento to Dr. Manish Diwan. May I request Dr. Manish Diwan to please come on the dais, please. Thank you, sir. May I now invite Professor Arvind K. Bansal, Organizing Secretary, NIPER PHARMACON 2022, to propose the vote of thanks.
Good evening, everyone. I am here to perform this pleasant duty of proposing the final vote of thanks. NIPER PHARMACON 2022 generated an enthusiastic response from academia and industry alike. The deliberations during the 3-day event were meaningful and would trigger collaborative activities. We are very sure of that. I would like to profusely thank Madam S. Aparna, Secretary, Department of Pharmaceuticals, for her guidance and support to the event and gracing today's event. Her presence is highly motivating for the entire NIPER family. I would like to thank Professor Y.K. Gupta for gracing today's function despite his busy schedule, and Mr. Deepak Behri for his inspiring talk to the students. I would especially like to thank the directors of other NIPERs, Professor Murthy, Dr. Shashi Bala Singh, and Dr. Ravi Chandran, for their presence throughout the event and inspiring the students to take up next level of research. We would like to put on record our thanks to OPPI for acting as knowledge partner for this event, and our sponsors Amway, Canara Bank, Merck, Raj Analytical for their support to the event. I would be failing in my duty if I don't thank the speakers for their presence and participating in the deliberations. And a big thank you to the delegates who have come from all parts of the country and outside the country to participate in today's event. I would like to mention that we will be posting a feedback form on the site of NIPER PHARMACON, where I request the delegates to provide their feedback. With this, I thank you all for participating in PHARMACON 2022. Thank you.
So, P7 Pharmaceutics, Reena, who stood second position, from NIPER S.A.S. Nagar. She is invited on the stage to take her prize. Veena. We can have that later. So sorry. I think they have taken it. Okay, we'll move on to. With this, we conclude the NIPER PHARMACON 2022 conference. Thank you everyone for being here for this occasion. All the delegates are requested to collect their certificates from the certificate committee desk in NIPER Convention Center lawn. Thank you everyone, and now I request all of you to join with us for the high tea outside. Thank you.